I'm Craig Camille. I'm the regional coordinator, Southern African regional coordinator for the African BioGenome Project. And it's my great privilege to warmly welcome you all to the 2025 Southern African Regional Workshop of the BioGenome Project. Whether here in person or online, your presence attests to the shared passion and commitment we have to advancing genomics on the continent. I would like to begin by expressing my sincere gratitude to our President and CEO, Prof. Tobeko Ntusi, who's the President of the South African Medical Research Council, for his unwavering leadership and vision in advancing science excellence in Africa. We also acknowledge and appreciate the efforts of the previous hosts of the African BioGenome Project workshops, whose groundwork has paved the way for rich discussions and collaborations, and we will continue that in that vein here this week as well. So my gratitude also extends to our incredible local organizing committee for their unwavering support and commitment to making this regional workshop a success. In particular, I'd like to thank Samira Gour for the efforts during the past year planning this event, and she has really gone above and beyond to make sure that we have an event that we can be proud of today. Also, I'd like to thank Karen Finnan from our Communications and Marketing Department for assistance and support during this planning process. Robert Ganesh and Ian Salamo from the Conference Center for all your help to make sure that we have a conference center that is, so I can agree with me, this is a very inviting venue, and so we thank the Conference Center for that. Our IT team, thank you Dwayne and Alex for all the help in getting our IT sorted out. To make sure that we can have an excellent online experience for our online attendees as well. And then, of course, we also like to thank God, who's always hovering in the background and making sure that everything is well coordinated all the way from the UK. And then we also have a list of sponsors. I'm not going to go through a whole list, but we would like to thank all our sponsors for being here today, and also for their involvement with all our regional practical workshops that will be happening over the course of the next week. We have an incredible lineup of guests and keynote speakers, and we'd like to thank each of them for taking the time out of their busy schedules to talk to us in the next 2 days. I'm also thrilled to have several of our postgraduate and early career scientists who will be sharing their work with us. And this is a good opportunity for all of them, these early career researchers, to network and to learn from experts in the field. So the Africa BioGenome Project represents a transformative initiative with an ambitious vision to build capacity and knowledge within Africa and to support sequencing and analysis of all life on this great continent. By fostering collaboration among scientists, institutions, and stakeholders, the project aims to unlock the vast genetic diversity Africa presents, and enabling groundbreaking research, conservation, and sustainable development. So this 5-day workshop is a crucial platform for connecting the regional scientific community. And we have an inspiring program ahead of us that combines seminar-style sessions with hands-on practicals on biodiversity, genomics, and the latest scientific insights. So these sessions are designed not only to educate, but also to empower you with skills and tools to advance your own research and institutional capacity. The theme of this year's Southern African Regional Workshop is Advancing One Health through Biodiversity Genomics, Innovations for Healthier Southern African Ecosystem. So this underscores the importance of an integrated cross-sectional collaboration to address critical challenges such as biodiversity loss, climate change, and emerging health threats. And these challenges that demand innovative genomic solutions and sustainable approaches grounded in African leadership and expertise. So as we embark on this workshop, let us embrace the opportunity to learn from each other and to share knowledge, to inspire action and to build lasting networks that will drive scientific excellence and social impact. And together we can propel Africa to the forefront of genomic research and biodiversity conservation. With that, I would like to declare the 2025 Southern African Regional Workshop of the Africa BioGenome Project open. Unfortunately, the MRC President and CEO is not available at the moment. He may join us a little bit later. So I will move on with the program and ask our next speaker, Professor Anne Mugai, to be ready for her presentation. Before she presents, I'll share a short bio. So Anne is a professor of genetics in the professor of genetics and is the Deputy Vice Chancellor, Academic Affairs and Research at National Defense University in Kenya. She was a member of the WHO Advisory Committee on Governance and Oversight of Human Genome Editing between 2019 and 2021. She is currently the chair of the African BioGenome Project, an African project that seeks to characterize 10,000 African species in 10 years. Furthermore, she is a member of the Bioethics Council and has previously been a member of the Kenyan Commission for University Education, the African Technology and Innovation Incubators Advisory Committee, the Kenya Plant Health and Inspection Services, and the Animal Genetic Resource Advisory Group for Africa, and holds an MSc in Plant Science, as well as a PhD in Population Genetics and Molecular Biology from the Jomo Kenyatta University of Agriculture and Technology in Kenya. With that, I'll ask Dr. Mugai to share the screen and start the presentation. Thank you.
So, good morning, South Africa. I hope I'm audible.
Yes, we can hear you online.
Thank you. Thank you very much. Let me first begin by thanking the local council of the African BioGenome Project workshop in South Africa for inviting me to make this presentation. And I'd like to sincerely congratulate Professor Craig and the entire local organizing committee for the successful launch of the workshop series today in SAMRC. My name is Professor Mugai. I'm the current chair of the African BioGenome Project. And my role here is to tell you more about the African BioGenome Project. And I've chosen to title Unlocking Genetic Diversity for the Sustainable Development of Africa. So this is my outline as I will make the presentation, make through the presentation. I'll be able to touch on these topics. But African BioGenome Project was conceptualized in 2019. And between 2019 and 2021, a few scientists, African scientists, were beginning to ask themselves a question: when is genomics and biogenomics, genomics and biotechnology and engineering going to take off in Africa? And between 2019 and 2021, we were looking at the framework, and this was actually the brainchild of Dr. Thangod Ebenezer, who's a very active member of the AfricaBP Steering Committee. And in 2021, by 2021 June, we had established a framework, and we launched in June after having a general meeting with African scientists, stakeholders. And in March of 2022, we published our vision what we were seeking to do. And there we told the world that we are going to sequence 100,000 species on the continent. And very, very quickly, we didn't expect the reaction that we got, but actually the reaction was overwhelming, and people were asking us, how are we going to do it? How are we going to fund it? And very quickly we were informed it will cost over $1 billion over the next 10 years. And the question was, should we be spending this money on sequencing efforts on the continent, or should we be using it for other priority areas like provision of health, provision of sanitation, fighting diseases and everything? So but we were very clear that if we want to solve those other problems that will come in the future, and we're actually facing it now, the place to start is actually through sequencing our genomes on the continent. But why Africa? And I could ask you the question, why not? Africa is home to the world's most diverse, most unique, not only animals, but also the ecosystems. We have very, very unique endemic species which are found nowhere else in the world. And we have a very, very large pool of Africans working in Africa, doing genomic science, doing state-of-the-art science that can impact global science. So we need to take the bull by the horns and be able to start innovating and spreading our innovations to the rest of the world. And through this, our discoveries will impact agriculture, will impact medicine, will impact conservation, starting from wherever we are here on our continent. But we need to leverage on this indigenous biodiversity, which we have realized is very, very unique, and it has traits that can be able to help the world in various areas, including plants that are disease resistant, plants and animals that are disease resistant or pest resistant. They are tolerant. And now with climate change, plants that can be able to withstand stressors, animals that can be able to stay longer periods without water, are the other sort of plants and animals that we need. But we have to ask ourselves, do we know what these plants are? Do we know what they have? And that is where the challenge is, that many of these plants have not been fully documented. They have not been fully characterized. And the sad thing is that they're already beginning to get lost through the extinction processes that we are seeing happening with us already. So it is a race against time, and we need to start now. We don't have time to wait for others to do it for us. We recognize that every region has its challenges. The scientists in Asia are dealing with their challenges. The scientists in Europe are dealing with their challenges. So we can't sit back and say, oh, wait, let's wait for the scientists from Europe or the scientists from China to come and help sequence our genomes for us. No, we have to do it ourselves. And we are the ones who better utilize the benefits, and we understand why we need to sequence what. And also remember that when scientists from outside come to do science on the continent, they have their priorities, which might not be fully aligned with ours. And therefore, when we do it at home, we are able to align our own priorities with our greatest needs and our greatest challenges that we need to solve. So the African BioGenome Project is set out to do exactly this. It's a large-scale initiative that is aimed at sequencing and catalog the genetic diversity of Africa's flora, fauna, and microbiodata. And it's a continental effort. So we're using scientists from all corners of Africa. It is led by African institutions and led by African scientists. And this is something that is very, very unique, and something that we are very, very proud of. So the goals of the African BioGenome Project are to coordinate all non-human genome sequencing initiatives. We're not sequencing the African human because there are other groups that are doing it, and they're doing it very well. So we're not seeking to do that. But we want to initiate, develop, and support any sequencing effort that involves a non-human genome sequence. So plants, animals, microbes, eukaryotic microbes. So we're not dealing with any prokaryotic genomes, but any eukaryotic genomes we're interested in. And our goal is to sequence 100,000 genomes in the next 10 years. And then we need to deploy this data to inform policy, to inform innovations that will actually help us produce more food, and also provide that data to be able to answer questions on which organisms are getting extinct and which ones we need to prioritize. And we hope that we will be able to contribute this sequence data to homegrown regional biobanks that are located within the African continent. So what is the importance of genomic research in Africa? It is very, very key to answer this question. I mean, we are now facing so many threats. Not only are we as Africans eroding our environment, but now the climate is changing. So on top of that, we have habitat destruction, we have illegal trade of animals and plants, we have pollutions, we have overgrazing. And therefore, we need to be able to answer questions very, very quickly. Some of the questions we're asking about which genomes should we sequence first, which genomes are very, very important for us. And these answers can be provided by genomic data. And what can genomic data do? Providing data-driven solutions, rapidly providing answers, creating an understanding of what is happening. We're talking about climate change, but are we understanding what that means to our livestock? Do we understand what it means to our soils or to our maize? We don't really understand what stresses are happening to our crops. And therefore, genomics and bioinformatics can be able to provide these answers. And then we also need to understand what adaptations are already occurring in these plants and these animals. And again, this is something that genomics can be able to provide. And using these answers, we'll be able to provide effective conservation strategies, effective breeding strategies, and effective responses to the climate change we're already experiencing. So to do this, we need to strengthen the collaborations within the African continent. As I mentioned before, African BioGenome is using African institutions and is using African scientists. So we need to know who these African scientists are, where they're at, what are the institutions they're located in, and what infrastructure is available in those institutions. I think one of the things that has happened very positively for Africa is that we have very, very strong North-South collaboration. So we have very strong European, Asian, North American partners. But the partnerships within Africa itself are not very strong. So we need to be able to build these partnerships, and that's what Africa BioGenome Project is aiming to do. And then use existing infrastructure and leverage that to be able to do and drive our genomic sequencing and bioinformatics efforts. You see, if an institution like SAMRC has invested, and it has invested quite a lot on genomics and bioinformatic infrastructure, we as African BioGenome Project should be able to leverage on that infrastructure and ensure minimum duplication efforts so that the cost of investment is actually fully realized for the African continent. And of course, through bringing this network of scientists together, there'll be a lot of experience and best practice sharing, and therefore this will help other institutions follow and adopt what has worked elsewhere. So to be able to sequence 100,000 genomes on the continent, we will generate about 203,000 gigabases of data. And the question is, who will analyze this? And we do aim to analyze this on the continent. We don't aim to send any of our sequences outside for sequencing. So we're sequencing on the continent and we're analyzing on the continent. And so we realize that it will take a very, very long time to be able to analyze all this data if we don't address the infrastructural gaps that are present and also the capacity, the scientists who will be able to analyze this on the continent. So that's what Africa BioGenome Project is aiming to do, to see where these infrastructure gaps exist and also to quickly start building capacity in genomics and bioinformatics capacity. And that's one of the aims of this workshop, to give scientists hands-on experiences, not only through talks, but also in the lab itself. So to address these infrastructure gaps and training gaps, we launched the African BioGenome Project, Open Institute for Genomics and Bioinformatics, to equip scientists, students, and local institutions with the needed skills and the capacities to be able to drive genomics and bioinformatics science. And we want to have quality genomic and bioinformatics scientists, quality. And we also want to have more of them. So improving the quality and the quantity to be able to drive the key and necessary science on the continent. And we're also remembering that we as established scientists will not be here forever. So we need to bring people in now, young scientists in now, who will be able to drive this science on the continent when we're no longer there. So we need to develop early career scientists in genomics, bioinformatics. But now when we started sequencing, we realized that there are other areas like computer science, like taxonomy, the actual infrastructure construction, that we don't have these strong scientists on the continent, or at least as many of them. And therefore, we need to be able to drive this development as we go forward. And then we also need to train them, but not train them arbitrarily, but also retain them in African institutions. I think brain drain has been one of the ills that has affected the continent, where we train scientists on the continent and immediately we're not able to provide them jobs and therefore they leave. So we need to harness these skills and retain them in the continent. And it is only through doing this we'll be able to have homegrown research projects and homegrown research innovations. And this will be able to solve our problems actually locally. So as we do that, maybe let me focus on the milestones achieved. I think one of the biggest milestones we can say as African BioGenome Project we've been able to do is to actually sequence on the continent. I think when we started out, there was very little belief that it's possible to sequence high quality whole genomes on the continent, but we've been able to do this. And also through African BioGenome Project, we've been able to train hundreds and thousands of African young established career scientists on the continent. And this series of workshops started in 2023. And we have them regionally in West Africa, in North Africa, in South Africa, like the one that is happening, and in East and Central Africa. So West Africa workshops happen in May. The North Africa workshops happen at the end of the year, around October, November. South Africa workshop traditionally happen in September, and the East and Central Africa workshop traditionally happen in August. So during these workshops, we have 2 days conferencing style and presentation style workshop. So that's what's going to happen today and tomorrow at this particular workshop. And that will be followed by 3 days hands-on practicals. And here we can see practicals, images of practicals that happened in 2023. This is a practical session that happened in Kenya by Inkaba. This is a image that on the right is an image by Separations, which happened in South Africa in 2023. This is a CRISPR-9 workshop that happened last year at the African Genome Center in University in Mohammed VI Polytechnic in Morocco. And we have registered tremendous success. Our 2024 publication is in press, and look out for it. It should be coming out soon. But in 2023, we were able to train more than 3,700 African scientists. Of these, 408 received hands-on practical experience. 40% were female, so a good number of young females in Africa interested in the area of genomics and bioinformatics. And we were targeting young early career scientists, but we had a range of scientists who were able to benefit, including established scientists and industry players. And we had representation from over 49 African countries, meaning that we are actually reaching all the corners of the continent. So we published the manuscript in 2024. These were the findings of our workshop. And this is something that we aim to do every year so that others can build on the best practices that we have established and also learn from some of the challenges and how we have been able to address any challenge that we came across. In addition to that, we have established fellowships for African scientists. And these fellowships are conducted together with industry players, with university partners, or even with projects. So any additional funding we receive, we're able to plow it back to the scientists, young scientists who have already gone through a workshop like this and are already domiciled in African institutions. And what they do is that they apply, they receive recommendation from the institution, and it's very competitive. So for instance, we held this partnership in 2024 where we had a 3-month fellowship for what we see as emerging genomic leaders in Africa. We shortlisted 4 young African scientists, each from all the regions of Africa. And we sent them to the Institute of Genomic Biology in University of Illinois in Urbana-Champaign in the US. And they were also able to spend some time in an African institution. Pictured here, you can see the young scientists meeting the editor of Nature magazine. So imagine if you're in your early 20s and you meet the editor of, a very renowned journal like Nature, and you go discuss publications, it's most likely that you will start publishing there. So these are some of the experiences we give them, and we offer various types of fellowships sponsored by various partners throughout the year. So I want to leave you with some food for thought. Number one, we have been able to show that not only are you able to sequence on the continent, but you're able to generate very, very high quality genomes on the continent. But we also have to ask ourselves whether we as Africans are to blame, because we partner with our partners from elsewhere, and we believe that the project we're doing is African-led. And I want to pick the quotation from Dr. Thangod, who says, an African-led project is not one where the PI has a primary African affiliation, but rather where 100% of the research is performed on African soil. So sometimes we are guilty of sending sequencing out there. We're happy to see our name on a publication somewhere in the midst of many other scientists. But we have to ask ourselves whether that project is really African, if 100% or 99% of the work was actually done outside the continent, and the only 1% of the work that was done on the continent was collection of the sample. So the other food for thought is that training of early career scientists needs to go hand in hand with mechanisms of retaining on the continent. So we need well-paying positions in African institutions. We need good infrastructure so that they can be able to have job satisfaction. And we also need these young career scientists to be able to grow and get promoted and also develop themselves. So we also have recognized the very, very important role that AfricaBP Open Institute regional workshops are playing, that we are able to bridge capacity gaps in young African scientists in genomics and bioinformatics. We are able to see where the genomics and bioinformatic gaps are and build this capacity. And we are also able to inform and influence the curricula that can be able to taught and used in these African institutions. But we need to remove barriers that are impeding the growth of science in Africa by going fully open science. So going fully open science, not only are we talking about publication here, where we need to remove publication fees because these are barriers to the growth of science, but we also have to remove the barrier to access to training. So we in African BioGenome Project have been able to provide training for free. The young scientists do not pay a shilling. And we realize that providing fees, charging fees in this type of trainings is actually a barrier. So if we can be able to get institutional support and also support from our partners, then we're able to provide this training for free. And we thank the exhibitors, we thank the sponsors, we thank the institutions that have been able to bring this workshop together, because then we are able to provide everything for free to the young early career scientists. So I want to end there and tell you, please kindly visit our genome, the African BioGenome website. Please follow us on Twitter and X and LinkedIn, and we will be able to see what is coming, what we're offering and what the new developments are. And I thank you very much for your attention. Thank you.
For that thought-provoking and informative presentation on the work of the African BioGenome Project. I think what you have presented here shows us the power of this project, not only by providing training which is most important for our future scientists, but also the importance of having African-led research being done on African soil. I think that is a powerful message, and it's something that we will champion from our region, and we know that this is something that is close to the heart of the African BioGenome Project. Unfortunately, Prof. Ntuzi is still not been able to join us, but Dr. Shane Murray from Diplomic has kindly agreed to have a presentation moved a little bit further into this, closer into this session. So we'll just be loading the presentation and then get going with the keynote presentation. So while we're loading the presentation, I will share a short bio. Dr. Shane Murray. Dr. Shane Murray holds a PhD from the University of Edinburgh in plant and molecular biology and has over 25 years of experience in research, lecturing, and project management. She has worked previously for the Agricultural Research Council, University of Cape Town, and the Center for Proteomic and Genomic Research in South Africa. Her current role as the genomics coordinator at Diplomics, Shane leads the National 1K SA project and collaborates closely with genomics partners labs across the Diplomics network. So as soon as the presentation is loaded, I will hand over to Shane to talk to us about the 1K SA project.
I think, Craig, we might need to share the screen because we can't see the slide for Shane.
Hi, good morning. Thanks very much, Craig, for the introduction, and thanks for the invitation to give a few minutes talk today. I'm going to be talking about the 1K SA project.
Sorry, Shane, we are only seeing the presenter view. So is there a chance we could please see the other view, which is the opposite view?
Is that okay for everyone online?
It's still showing the presenter view. Maybe we can just carry on. So I suspect you might need to the box at the bottom. Maybe are you sure you don't want to click that? Not the video, but the box. That way it gives all the presenter view, the audience view, and not. Otherwise, we can just carry on. Another way to do this is to convert the slide to a PDF and then present the PDF, if that's okay.
How about now, thank God?
Still the same thing. So let's carry on. Let's carry on.
Excuse my notes. Okay. So yeah, I'm going to start off with by just giving a little bit of background to Diplomics. So Diplomics stands for Distributed Platform and Omics, and it was set up about 7 years ago by the South African Department of Science, Technology and Innovation through the South African Research Infrastructure Roadmap, or SORIA, and it's hosted by the Center for Proteomic and Genomic Research in Cape Town. So there are 3 pillars to Diplomics, the genomics, the proteomics, and metabolomics pillar, and then also bioinformatics. And my role in Diplomics is that of the coordinator for the genomics pillar. So Diplomics has got two objectives. The first of these is to build a world-class omics infrastructure. So that's to make sure that the labs are in South Africa equipped with the relevant omics infrastructure. And the second objective is to ensure and to enable access of students and researchers to that infrastructure. And the second objective has become more important to Diplomics over time. So as I said, the first of the actions to do the first objective were to buy and to place instruments, to make sure that those instruments were serviced and maintained correctly, and to ensure that technicians received advanced technical training. But over time, this is now expanded to enabling access to students and researchers. So we've expanded our training and research program. In the last year, we've reached over 40 different workshops and over 1,000 different people across South Africa. We've also employed what we call spatial transformations, that's reaching out to smaller and more rural institutes that are engaged in omics research across South Africa. And we've also started what we call a high visibility, high impact projects. And the 1KSA project falls under this category. So what do we want to achieve by building a high visibility, high impact project? We have got four main ambitions here. First of all, it's to use the project as a vehicle for developing methods for doing research and research services. There's a very big training component to what we are doing. So we want to train as many students and researchers as possible in the new techniques that we are optimizing and developing. We are very much engaged in open science activities. It's becoming a requirement from our funder that open science needs to be considered with outputs. So we've taken that on board and we've educated ourselves around open science and deploying those high visibility, high impact projects. And then the fourth aspect is developing a community of practice. So developing a community of users who are skilled in using the different techniques for that particular project and others for research as well. And so about 18 months ago, we launched the 1KSA project. So 1K stands for 1,000 genomes of biodiversity importance in South Africa. So the project is very much aligned to a lot of the objectives of AfricaBP that we just heard about from Prof. Mungai. And in this project, 6 of our partner labs in the genomics pillar elected to become part of this project. They're all represented here on the slide. So in addition to the SAMRC genomics platform, we also have SIAD, the South African Institute for Aquatic Biodiversity, which is based in Eastern Cape. We have a lab at the University of Pretoria. We have Sengen, which is just down the road. It's a plant genomics specialist lab. There's the lab at the IMBM at UWC, also just down the road. And then we have a training lab at Diplomics, which is used for training, but we also have a dedicated section of that lab for the 1KSA project.
Sorry, Shane,
yes.
They can't see your slides online.
Oh, dropped. Okay.
I think just convert the slide to PDF. Just save it as PDF and then present the PDF, please. Yes, it looks better now. Perfect.
Okay.
Thank you.
Thanks. Okay, so this slide is an outline of the 1KSA project. We have a dedicated website for the project, 1ksa.org.za. Please go and take a look at it. All the information that I'm going to present now is available on this website. So 1KSA is a community project. So members of the community can nominate for their species of interest to be sequenced under 1KSA. And the species nomination form is available on the website. The species nominations are then considered on a monthly basis by the steering committee. And once the requirements have been established and that particular species has been accepted, the sample contributor is then sent a sample submission form. The projects preferably would like to receive DNA, but as there are quite a few DNA requirements, we can assist with DNA extraction if necessary. After delivery of the sample to one of the 6 labs, and typically that would be the lab that is the closest to the sample contributor, DNA quality control and library prep take place, followed by whole genome sequencing. And so for this project, we elected to go with Oxford Nanopore sequencing because of the long reads. The nature of the long reads allows for a de novo genome assembly more easily. And as part of this project, we placed several P2 solos across the country in these different labs. The problems are so whole genome sequencing then takes place on a P2 solo, P2 solo, and then we make use of the national infrastructure, the NICUS infrastructure at the Centre for High Performance Computing to carry out the draft genome assembly, and then the data is backed up and stored on another NICUS facility, DeRisa. We have also developed data sharing and data access policies, and it's possible to apply via the website for access to data if there's a species that you are interested in. We also highlight the species that have been sequenced in the project by generating what we call a species card. This is in effect a report card. There's an example here on the slide, and I'll show some more later on, which basically gives some of the important features of the species, why it's important to sequence it, and some of the high-level metrics from both the sequencing and the draft genome assembly. I've noted on the slide the little red cap sign. So we make use of the research database red cap to receive all the sample information, the metadata, the permitting. We work closely with the biobanks in South Africa, and so information about the accession number from the biobanks is captured in red cap. We also use red cap to record all the different metrics, both the sequencing, the library prep and sequencing and draft genome assembly steps. And this allows us to then extract information that we need for reporting purposes later on. We also make use of Zenodo to move data around from the different sequencing instruments to the CHPC. We also write our methods and our protocols, and we publish them on a 1KSA community site on Zenodo. And we also use the GitHub to record our bioinformatics workflow. And both the Zenodo and the GitHub links are available via the website. So this is a one-stop shop of the project. So I mentioned that method development is a big component of this project. And if you look at the center of the slide, these are the 5 different core aspects of method development. So looking at the DNA quality control, making sure that the DNA reaches all the quality metrics that are required for Oxford Nanopore sequencing, the library prep, optimizing library prep to get the most sequence data at the next step, the sequencing step, and optimizing sequencing to also ensure that we get optimal sequencing information. Data management has been a big aspect of this project. We had to start from scratch in terms of figuring out how to manage the data. Professor Guy spoke about keeping all the data on a continent. This is very much aligned to the 1KSA project as well. We were quite determined to make use of the national infrastructure for this project, and so we had to figure out how to do that. And this is an ongoing process. We're constantly tweaking it and trying to optimize it. And then finally, the genome assembly. We carry out a draft genome assembly workflow. It is very much just a high-level draft of the genome assembly, and it's really to collect QC metrics to ensure that we have sufficient sequencing data for the data is usable for scientists down the line. So on the library pregnant sequencing side, we've developed a decision making tree that guides us through the different steps of the library prep process. So first of all, we do have metrics that we publish on the website about how we would like the DNA to be when it's submitted. But we take a pragmatic approach, and if this is a very precious sample and it's not possible to get better quality DNA, we have developed a number of cleanup steps that can be used to try and get the DNA in a better shape for long-read sequencing. We then look at the average fragment size, and if it is bigger than about 40 kb, we fragment the DNA a little bit. And this is just to allow those long pieces of DNA strands to pass through the oxonanopore pores easily for sequencing. And if the DNA is a little bit smaller in size, then we don't carry out that fragmentation step. The next step is if we look at the expected genome size, and we make use of the genomes on a tree tool to estimate the genome size. And if the genome size is less than a gigabase, then we will consider using the native barcoding kits from oxonanopore. So that's only if we've got more than 2 micrograms of high molecular weight DNA, because this will allow us to make good libraries and also to multiplex them on one flow cell. If the amount of DNA is less than 2 micrograms, or if the genome size is bigger than 1 gigabase, we then use the ligation kit, which allows for 1 library on 1 flow cell. In the early days, when we had very poor DNA and we were starting out poor DNA, we would then use the short fragment buffer from oxonanopore if the DNA fragment size was less than 3 kilobases. But we hardly ever see that anymore, and most of our libraries are now built using the long fragment buffer, and in which case the DNA fragment size is greater than 3. And then if the expected size of the genome is less than 3 gigabases in size, we will then sequence on 1 flow cell. If it's bigger than 3, then you know we're in for multiple sequencing reactions and we need to use more than 1 flow cell. And we also employ a wash and reload in order to get up to 20% more sequencing data out of the flow cell. So this has been an incremental process over the last 18 months, and it's still an ongoing process. We tweak this protocol all the time and try and aim to get a much better output of sequencing. On the draft genome assembly side, we've also employed molecular development sort of thought process here. And this is, if we do it, the pipeline has been built in Nextflow for running on the CHPC. And this is the high-level steps in the pipeline. So we use Dorado for base calling. This happens during the sequencing run in most cases. And then there's a quality assessment step. We use KMC, which generates KMO analysis. And a single KMO peak will indicate then that we have sufficient coverage to continue with genome assembly. We use nanoplot and nanofilt to generate QC metrics and to filter and trim when necessary. And then initially we used the long-read oxonanopore optimized assembler fly, and then that was followed by evaluation with QUAST and BUSCO. BUSCO standing for benchmarking universal single copy orthologs. And we also employed, along with fly, a polishing step, which first meant mapping the reads back onto the draft genome assembly using Minimap and SAM tools, and then polishing with a Recon tool, and then this was then followed by another evaluation step. However, earlier this year, oxonanopore published an ONT optimized version of HiFiSm, which is a runtime efficient long-read genome assembler. And we've recently integrated this into our pipeline as well, and we're getting some really good results using this tool as an alternative to fly. So we've also developed very recently a decision-making tree around which assembler to use. And I'm going to take you through this briefly here. So I mentioned the KMO peak is one of the first QC steps. We need to establish that we see a KMO peak, which indicates that there's sufficient coverage in the data set. If on the right-hand side of this image, if there's no KMO peak, then we will do a bit of filtering and trimming. We've established some metrics which can be used. Sometimes too much data doesn't work, and we have to filter a little bit. If there's a KMO peak, then we can proceed to the genome assembly step. But if there's no KMO peak, then we will do more sequencing until we start see that KMO peak coming out. So once the KMO peak has been visualized, if the genome is a big genome, it's bigger than 3 gigabases in size, we will then proceed to the high-fiism step. In the guidelines that Oxford Nanopore initially published, they stated that high-fiism would be suitable for a sequencing input, and I'll come on to what that means later on, greater than 10 kilobases in size. And if that was seen, then we would proceed with high-fiism. But if the sequencing input was less than 10, then we would do some more filtering and trimming in order to try and generate a data set that had a better sequencing input. If the genome size was smaller than 3 kb, we still use fly, especially in situations where the sequencing input is less than 10 kb. However, we have been testing high-fiism on non-optimal sequencing. By that I mean an input less than 10 kb, and we're getting very good assembly. So we're very happy with the fly results, and it's really been a game changer for this project, especially in the plant space where the genomes are really big, and we had hit all-time problems with CHBC with genomes up to about 5 gigabases in size. So this has been a recent new development project. Very good. Okay, so as I mentioned at the beginning, we've developed terms of use for 1KSA, and we try to use open access and open science principles here. We have terms around data access, use, and publication by the sample contributor, and we are starting to think about the 1KSA samples as being a conservation species versus a bioeconomy species. And in the case of a conservation species, it absolutely makes sense to get that data out as soon as possible so that people can use it and can generate sort of value out of that that can be used for conservation of an endangered species. On the other hand, we're also aware of the fact that because of South Africa's great biodiversity and lots of bioeconomy uses of the species, that releasing those genomes into the public domain very early on could create IP issues. And so we've developed some policies around requesting an embargo for that data to remain private for longer. But we do ask that after 3 years, the sample contributor can have up to 3 years to reanalyze that data, add additional data sets to it, that data is typically then uploaded onto the European Nucleotide Archive. We also have developed data access, use, and publication policies. These are all on our websites. We've set up a data access committee that will evaluate a request for access to data and also a data transfer agreement template. We've developed an acknowledgement statement, which is on our website, and we ask that all 1KS sample contributors use that acknowledgement statement when they publish. We also ask them to track any intellectual property that they might develop using the genome. And as we would like there to be a small portion of that future IP that would feed back to genomics, biodiversity genomics education in South Africa as a benefit sharing mechanism. A big part of our project is visibility and awareness, and I mentioned already the species cards. There are 3 different examples. One is the Wooders whale, which is the only inshore whale found exclusively in South African waters. This was contributed by the University of Victoria. Slickworm, which is an iconic plant in South Africa that was sequenced by Sengen. And then Halloon on the left-hand side, which is the national fish of South Africa, and this was sequenced by Sire. So we use the species cards to raise visibility around the genome that's been sequenced. We also make videos. We have a number of short videos that are accessible on our website about key interesting species. And then we also produce stickers. And the equivalent sticker for the good as well, speckworm and the Halion are here. And for those of you who like stickers, I've brought quite a few of them with me. Just come and talk to me at tea time and I'll give you some stickers. So we've also integrated training into the 1KSA program. We had been supporting library prep and sequencing training through genomics for a number of years, and we follow the principle of train the trainer. So, if possible, we want to train somebody who's going to go on to then train other people in their home laboratories. We've run 8 library prep and sequencing training workshops over the last 2 years, and reached, just in this instance, more than 100 participants. For the last 2 years, we've supported the AfricaBP Southern African Institute with workshops at the University of Pretoria. And this year again, we will be running workshops now at the training lab at Genomics, because we're running the workshop this year. For the first time in May of this year, we ran the first 1KSA Genome Assembly workshop. These are some pictures from that workshop, and we'll be running that again also for AfricaBP next week. So I mentioned that we use open science in the 1KSA projects, and these are just some of the ways in which we are applying the open science principles. So by using open methods, we have our website, brings attention to the methods we're using. We keep records in RedCap. We publish our protocols on the 1KSA Zenoda community. We use open resources. We enable access to sequencing through the 6 different labs, through the sample contributor nomination process. We use national informatics resources. We have a data access policy and a data management plan for 1KSA as well. And we also promote training. We promote open source through our GitHub workflow that describes the 1KSA genome assembly steps. We promote open data through our 1KSA data management plan, through our data access committee, we promote visibility to our species cards, and we also request that the data is uploaded to the European Nucleotide Archive after 2 years. And then finally, open access, we promote sequencing where possible through open access journals. So this is the landing page of the 1KSA website, for those of you who haven't seen it yet. On this web page, we record our key metrics on a monthly basis. So starting on the left-hand side, as of the end of last month, we have now created 91 species cards. We have completed 102 assembled genomes. So the difference between these will soon be translated into the production of the species card. Lags a little bit behind. We've sequenced nearly 150 genomes, and we have 177 samples that have been received. We also record on our website the number of species that have been nominated, and that currently stands at 379. And we want to try and make it 500 by the end of the year. So to the South African researchers that are listening in, please get your species nominations in as soon as possible. And then we also record here the number of bacterial species that have been sequenced. So our partner at UWC is a bacterial expert. Their mandate is to sequence directly from their biobank, which is also at UWC. And so they've sequenced now just over 500 genomes from that biobank. I'm just going to take you through some of our results, and by having RedCap, we are able to extract some of the results. So on the slide, on the left-hand side is the sequencing read N50. So this is for our first 100 or so genomes that we sequenced. And so for those of you who don't know, the N50 is the size of the smallest read in this case, where it and all the reads larger than that make up 50% of the data. And so this graph gives an indication of the read and input size on the x-axis, and then the counts, so the number of species in the 1KSA project that had that particular read input. And the yellow line is the average. So the average is sitting at about 7.5 kb, which we are happy with. And you'll see it in some cases we're getting really long sequencing entropy. And as this project is going on and our methods are improving, we're seeing that there's a shift to much longer sequencing intervals. On the drafting and assembly, the graph slide. So here our content input is in the megabase range, which we're also very happy with. And again, the yellow line is so completeness of a genome is evaluated through the BUSCO tool, so the Benchmarking Universal Single Copy Orthologs tool. And this is just a breakdown of the results that I showed on the previous slide. In all cases, the single and duplicated percentage of genes is way above 95%. So we are happy that we have most of the genome represented. Just quickly, some examples. I mentioned Illumina, which is the national fish of South Africa. This is the data that appears on the species card. So a quick indication of the importance of the species, the output from the ProMethine sequencing run, the drafting and assembly output, so the content input is listed here along with the BUSCO score, the name of the sample contributor is also listed, and then also the distribution of this particular animal system as well. There's another example. This is elephant, which had actually been sequenced before, but not using long read sequencing, which is why the steering committee agreed for this organism to be included, and this was nominated to the University of Victoria. Okay, so just in terms of what impact is 1KSA having. So it's a proudly South African project from start to finish. Everything is happening in South Africa, from sampling, DNA extraction, sequencing, draft genome assembly, and also data storage. In the first year of the project, so this was sort of earlier this year, about 6 months ago, we had sequenced 100 eukaryotic species and more than 500 bacterial species. It's an example of a distributed project. So we've got 6 labs across the country that are working on this project. We have developed a community of practice. So these 6 labs, there might be small differences in the way they approach things, but by and large, they're using the same protocols, which is important for the community of practice. We have also generated a lot of experience in nanopore sequencing in country. And now there are plans afoot to do a similar project using human samples. And this is very much a community project. In the first year, we had 45 South African scientists nominating eukaryotic species for sequencing in 1KSA. And these 45 scientists came from this huge diversity of different organisms. Organizations as shown on the slide. Right. I think that's my last slide. Oh, this is my last slide. How can you get involved? So first of all, if you are interested in using whole genome sequencing in a project, maybe you're a PhD student starting out, you can consider nominating your species for 1KSA. Our requirements are that it has to be indigenous to South Africa. We require high molecular weight DNA, but again, if you're struggling with that step, we can provide some guidance and support. All the permits and ethics and transportation permits need to be in place. That's the responsibility of the sample contributor. We also request a lot of metadata for the sample. And this is because we want to tie in the sample that is sequenced with biobank samples with the future view of maybe using or adding 1KSA data to a South African instance of GBIF, for example. And you also have to agree to the terms of use. You may find that the species that you're working on has already been sequenced, in which case you can request access to data, and you can do this from a different part of the website by filling in the application form there. You can spread the word about 1KSA. You can go back to your home institutes and tell your friends and colleagues about this project and tell them about how they can nominate samples for sequencing. And you can also attend the 1KSA training. So for people who've signed up for the library prep and sequencing training later this week and the draft genome assembly workshop next week, you're already on your way towards that. Okay, and with that, I just would like to thank all 6 labs in the 1KSA project. It's a great project. We work really well together, and I would just like to acknowledge all the hard work. Thank you very much.
Thank you, Shane. Before we go into tea, we will have a focus session. But I'm just going to give an opportunity for about a 5 or 10 minute question and answer session for our 2 speakers this morning, for Anne and for Shane. So if there are any questions that you would like to pose to any of the speakers this morning, this is always open now for questions. And thank God, if there are any questions online, would you please also just mark that for us. Do you have a question out there?
Thank you for your talk. I was wondering about the cost of the sequencing. Thank you so much. Hope everyone can hear me. So I was just wondering about the cost. I know there's been a dramatic decrease in the cost of sequencing. If I was interested in having my organism interest sequence, roughly what is the cost to do that?
So first of all, for all samples that are accepted by the project, currently there's no cost. So your cost would be the DNA extraction, the sampling and the DNA extraction, and querying it to the lab if that was required. But all the sequencing, all the DNA QC, the library prep and sequencing, that's all provided for free by the project, which is why we've also provided the terms of use. So it's not just giving you the data; it's got to also be used by the people under open science principles and so on. What we have been able to do with Oxford Nanopore is negotiate bulk discount arrangement as well. So that's the power of where Diplomics has been able to get involved. So it's we're benefiting on both sides. So Diplomics buys all the reagents and then provides the labs for the reagents for sequencing. What we've also seen as an extra impact is that these labs now are able to offer services outside of 1KSA. So we've helped those labs establish protocols so that they can now get to their own sustainability from services as well.
Thank you so much.
Yeah. Okay. Hi, Craig.
Do you have any questions online, thank God?
Yeah, I think I can't see any question, related question. Okay, I have one question from online participants for, yes, I think they're asking how they can get training for the 1KSA practicals. Yeah, but I also have, in addition to that, I have a question I wanted to ask, but more of a comment and to allow Shane to kind of explain to the audience, many of which are attending from several African countries. And if one of the things we encourage in the AfricaBP is for scientists at the national level to set up their own national biodiversity projects, for instance, 500, 1,000, any number. So Shane, could you please in the next few minutes explain to the audience listening, there are over 200 online and more will join later. What was the experience like in kind of establishing this at the national level, particularly, how did that happen, and how did you get the scientists at the local level, the buy-ins, the institutions? That is one question. Another one is I can see there's a heavy interest in ONT. How does other players in terms of genomics technologies also be leveraged upon to get this data to, for instance, additional levels? And are some of this also chromosome scale, when you have like omnic and long read? So, all this perspective will be helpful to everyone listening on the call today. And hopefully we'll have some scientists who go back to their countries and, try to provoke actions at the national level.
I hope I can remember all those points. But how did 1KSA come about? So in the early days of the permits, we had what we call the technology showcase opportunity. And so quite early on, we decided to do an Oxford Nanopore showcase. And so the labs on the genomic side who wanted to be involved, we all provided them with a frequency, which is a smaller sequencing instruments. The Promethean hadn't come about at that point. And we tasked them with testing the frequency on typical DNA samples that they saw in their service environment. And that kind of that was a working group we set up, and that pretty much set the scene for 1KSA, because it emerged out of that there was a lot of interest in biodiversity sequencing. So I suppose you could say in some respects 1KSA started out as organically. We spoke to our partners, they were interested in biodiversity sequencing, and at that time then the guidelines were made available by Oxford Nanopore, and we were able to then discuss with the local representatives in South Africa about getting the starter packs out. That was still in the day where you could get starter packs for the Promethean. So, our view from a diplomics perspective was about generating an omics infrastructure, but then we added the 1KSA as kind of a structure to a vehicle for training, community development, and so on. So that's kind of the background to why 1KSA came about. Your comments, I think, about a lot of interest in Oxford Nanopore sequencing. I think broadly what we've seen in our journey with Oxford Nanopore as well is that in the early days there was a lot of comments about the error rate being so much worse than other sequencing platforms, but that's really improved a huge amount. The long reads really are useful for a de novo genome assembly. So it's the right fit for a biodiversity genome assembly project. It's relatively cost effective. There's a lot of data you generate. It's a medium output platform, which I believe is more suitable for South African and African context, where research budgets maybe aren't that high and people do want to just do a little bit of sequencing, not necessarily a lot. And what we see now is that not only are we getting better and better sequencing NFTs as we tweak and improve the different steps in the library prep part of the process, but high-fiism, as I mentioned, is really helping a lot in terms of getting near chromosome level genome assemblies. And for many downstream applications, that is fine. For a conservation species where you may be wanting to do population genomics, that's probably fine to have a draft genome assembly that's near chromosome level, but not a reference genome per se. And maybe just one last comment is that from our funders' perspective, we have to draw quite a careful line between providing support for research and research infrastructure. And so it's very easy for us to do a draft genome assembly under 1KSA because that's a research infrastructure that people can use to go on and do research with. And it's again, and also part of our reason for doing just the draft genome assembly is we wanted to reach more sample contributors, so the budget would just go much further based on that. Is there anything I missed out in the comments?
I think you've covered everything, Shane. Thank you. So we've run out of time for Q&A. We're going to do a group photo. So we'll just get Alan, who's going to set up with us to do a group photo. Then also, for those of you who haven't received your name yet, your name tagged it, Samira has been front. And also, I think all of us would please just sign the registration registry so that we can arrange for certificates of attendance following the conference. So I'll hand over to Alan. And then after the photo, we'll go in move into tea, and we'll start our next session at 11 o'clock, just so that we are on time, on schedule again for the people online. Okay.
So for the online participants, just to mention that they are going for a group photo and they'll be back in the next 10-15 minutes.
How are you? How's it going? Really? Oh, it's not
We're really interested in using high-dimensional phenotyping or phenomics to uncover the mechanisms of disease biology. And particularly, we're really interested in the biological mechanisms of genetic variants and mutations that are associated with both neuropsychiatric and neurodegenerative disorders. And we've been really interested in working with Element on their new technology because morphology is only one measurement. We have recently published this work on what we call neural painting, which is simply taking cell painting and then applying this to neural cell types. It's really easy in the lab, but the processing and analysis can really take a long time, and that feature interpretation that you get from that always requires follow-up experiments. So you have to then go measure RNA, you have to measure protein, and then try to understand which ones of those give rise to the morphological changes. And so the AVD24 platform allows us to capture all three of these simultaneously. And so we set up a pilot experiment with the AVD24 team where we took iPSCs from our lab, we differentiated these into both astrocytes and excitatory neurons. We grew these cells on the Teton flow cells. We would fix them, and then we sent them to Element for processing, part of the technology access program. Firstly, we're able to take the single cells and cluster them using the RNA expression transcripts with the AVD neuro panel. But here, we actually get better accuracy, and we get the added information of kind of the RNA expression. So here again, we're just taking these clusters that we found using the gene expression information, but then layering on here values for the morphology features that seem to discriminate our cells. But one could easily imagine now going into each of these clusters and doing different kind of regression analyses to sort out whether there are specific proteins or transcripts that would associate with these morphological characteristics so that we can begin to draw linear relationships between protein and RNA activity and how cells change in response to certain contexts. Doing 1 million cells in a run is feasible, right? And having the data to come back after simply overnight, effectively, is really incredible. Sometimes I think it's hard to kind of grasp your head around rather than waiting weeks or months to get information on 1 million cells. You can kind of have it right away. The actual experimental part of it is incredibly easy. You just need to grow cells and fix them. In a system like this, where we can use many different dimensions to measure, at least in vitro, when things begin to change, that gives us a better sense of maybe what's causing disease rather than what is caused by disease. And so doing everything simultaneously, I think, could be really powerful.
Welcome to the 20-year anniversary video of AfricaBP, Africa's genomics company, here from our headquarters in Makelnek, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of AfricaBP emerged not far from here in Pretoria in the year 2000 while I was doing research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so. The startup was only possible with the substantial direct foreign investments, mainly from shareholders from Germany and Switzerland, as well as the startup loan from the State Secretariat for Economic Affairs of Switzerland. So what was actually our idea in 2000 to start a biotechnology company in South Africa? The aim was to locally produce oligonucleotides which need to be custom made, and therefore by local production the turnaround time dropped from 2 to 3 weeks for imported oligos at that time to 1 or 2 days. This of course allowed to accelerate research output for our customers that apply PCR amplification and DNA sequencing in their research. And more recently, it allowed uninterrupted essential supply during the pandemic. In addition, we aimed to offer Sanger DNA sequencing services. The aim was here that outsourcing DNA samples for sequencing will allow our customers to save the investments in capital-intensive sequencing instruments. 20 years later, we still do all these services, but of course the technologies in life sciences have significantly evolved in the past 20 years, and so has AfricaBP. A big thank you to all our customers, our dedicated current and former employees and commercial partners over all these years, and of course our investors that took the risk and made this possible in 2002. Please enjoy this video about the different aspects of AfricaBP. Let us know your comments, suggestions or questions about AfricaBP in the comment section below. We strive to even be better in the next 20 years.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation. MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DNB-seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of few companies in the world that have the ability to develop and mass-produce low, medium, and high-throughput clinical gene sequencers from GB to TB. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, BIT products, and low-temperature storage products, with a strong focus on the upstream of the industry. MGI operates in more than 100 countries and regions, serving more than 2,800 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
QIAGEN.
Sample to insight.
Introducing the newest member of the QuantStudio family, the Applied Biosystems QuantStudio Absolute Q digital PCR system from Thermo Fisher Scientific. This fast and easy-to-use digital PCR solution offers high-quality data with minimal hands-on time. Sample prep is identical to qPCR. Simply load your reaction into the plate and insert the plate into the instrument. All components of digital PCR, including reagent compartmentalization, thermal cycling, and data collection, are performed on the single standalone instrument. After loading your plate, start your run and return in 90 minutes for sensitive, accurate, and precise quantification of your targets. The quantitation accuracy for any digital PCR system is driven by the amount of sample that is analyzed in a reaction, and while some digital PCR platforms can waste up to 75% of your sample, the QuantStudio Absolute Q system consistently analyzes over 95%, so that you can see the whole picture every time. At the heart of the QuantStudio Absolute Q digital PCR system is the novel microfluidic array plate technology. This technology was developed for maximum consistency and accuracy by minimizing waste and maximizing reagent distribution into microchambers. And all you need is one plate and one instrument. The MAP16 digital PCR plate provides reliable consistency, high accuracy, and an easy workflow. After loading your sample into the plate, the instrument performs automated reagent distribution into fixed microchambers. Each reaction remains independent during thermal cycling. 4 optical channels allows you to analyze more targets per sample. As the only digital PCR solution with the capabilities for pre- and post-PCR background subtraction, the QuantStudio Absolute Q system employs sophisticated rejection of non-PCR artifacts, offering high confidence in true positives for every microreaction. The MAP plate is designed to run up to 16 samples in parallel, with the flexibility to run as few as 4 at a time to match experimental needs without wasting partial plates. The intuitive QuantStudio Absolute Q analysis software enables users to easily change and define experiment-specific parameters, name and track experiment details, and monitor the run from an easy-to-understand user interface. After the run is complete, return to view the results and apply powerful analysis software. QuantStudio Absolute Q Digital PCR System from Thermo Fisher Scientific. Experience the future of digital PCR at thermofisher.com/absoluteq.
We're really interested in using high-dimensional phenotyping or phenomics to uncover the mechanisms of disease biology. And particularly, we're really interested in the biological mechanisms of genetic variants and mutations that are associated with both neuropsychiatric and neurodegenerative disorders. And we've been really interested in working with Element on their new technology because morphology is only one measurement. We have recently published this work on what we call neural painting, which is simply taking cell painting and then applying this to neural cell types. It's really easy in the lab, but the processing and analysis can really take a long time, and that feature interpretation that you get from that always requires follow-up experiments. So you have to then go measure RNA, you have to measure protein, and then try to understand which ones of those give rise to the morphological changes. And so the AVD24 platform allows us to capture all three of these simultaneously. And so we set up a pilot experiment with the AVD24 team where we took iPSCs from our lab, we differentiated these into both astrocytes and excitatory neurons. We grew these cells on the Teton flow cells. We would fix them and then we sent them to Element for processing, part of the technology access program. Firstly, we're able to take the single cells and cluster them using the RNA expression transcripts with the AVD Neuro panel. But here, we actually get better accuracy and we get the added information of kind of the RNA expression. So here again, we're just taking these clusters that we found using the gene expression information, but then layering on here values for the morphology features that seem to discriminate our cells. But one could easily imagine now going into each of these clusters and doing different kind of regression analyses to sort out whether there are specific proteins or transcripts that would associate with these morphological characteristics so that we can begin to draw linear relationships between protein and RNA activity, and how cells change in response to certain contexts. Doing 1 million cells in a run is feasible, right? And having the data to come back after simply overnight, effectively, is really incredible. Sometimes I think it's hard to kind of grasp your head around, rather than waiting weeks or months to get information on 1 million cells. You can kind of have it right away. The actual experimental part of it is incredibly easy. You just need to grow cells and fix them. In a system like this, where we can use many different dimensions to measure, at least in vitro, when things begin to change, that gives us a better sense of maybe what's causing disease rather than what is caused by disease. And so doing everything simultaneously, I think, could be really powerful.
Welcome to the 20-year anniversary video of AfricaBP, Africa's genomics company, here from our headquarters in Mackleneck, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of AfricaBP emerged not far from here in Pretoria in the year 2000, while I was doing research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so. The startup was only possible with the substantial direct foreign investments, mainly from shareholders from Germany and Switzerland, as well as the startup loan from the State Secretariat for Economic Affairs of Switzerland. So what was actually our idea in 2000 to start a biotechnology company in South Africa? The aim was to locally produce oligonucleotides which need to be custom made, and therefore by local production the turnaround time dropped from 2 to 3 weeks for imported oligos at that time to 1 or 2 days. This of course allowed to accelerate research output for our customers that apply PCR amplification and DNA sequencing in their research. And more recently, it allowed uninterrupted essential supply during the pandemic. In addition, we aimed to offer Sanger DNA sequencing services. The aim was here that outsourcing DNA samples for sequencing will allow our customers to save the investments in capital-intensive sequencing instruments. 20 years later, we still do all these services, but of course the technologies in life sciences have significantly evolved in the past 20 years, and so has AfricaBP. A big thank you to all our customers, our dedicated current and former employees and commercial partners over all these years, and of course our investors that took the risk and made this possible in 2002. Please enjoy this video about the different aspects of AfricaBP. Let us know your comments, suggestions or questions about AfricaBP in the comment section below. We strive to even be better in the next 20 years.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation. MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D MGI has developed a full range of patented technologies, including DMB-seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of few companies in the world that have the ability to develop and mass-produce low, medium, and high-throughput clinical gene sequencers from GB to TB. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, BIT products, and low-temperature storage products, with a strong focus on the upstream of the industry. MGI operates in more than 100 countries and regions, serving more than 2,800 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
QIAGEN. Sample to insight.
Introducing the newest member of the QuantStudio family, the Applied Biosystems QuantStudio Absolute Q digital PCR system from Thermo Fisher Scientific. This fast and easy-to-use digital PCR solution offers high-quality data with minimal hands-on time. Sample prep is identical to qPCR. Simply load your reaction into the plate and insert the plate into the instrument. All components of digital PCR, including reagent compartmentalization, thermal cycling, and data collection, are performed on the single standalone instrument. After loading your plate, start your run and return in 90 minutes for sensitive, accurate, and precise quantification of your targets. The quantitation accuracy for any digital PCR system is driven by the amount of sample that is analyzed in a reaction, and while some digital PCR platforms can waste up to 75% of your sample, the QuantStudio Absolute Q system consistently analyzes over 95% so that you can see the whole picture every time. At the heart of the QuantStudio Absolute Q digital PCR system is the novel microfluidic array plate technology, This technology was developed for maximum consistency and accuracy by minimizing waste and maximizing reagent distribution into microchambers. And all you need is one plate and one instrument. The MAP16 digital PCR plate provides reliable consistency, high accuracy, and an easy workflow. After loading your sample into the plate, the instrument performs automated reagent distribution into fixed microchambers. Each reaction remains independent during thermal cycling. 4 optical channels allows you to analyze more targets per sample. As the only digital PCR solution with the capabilities for pre- and post-PCR background subtraction, the QuantStudio Absolute Q system employs sophisticated rejection of non-PCR artifacts, offering high confidence in true positives for every microreaction. The MAP plate is designed to run up to 16 samples in parallel, with the flexibility to run as few as 4 at a time to match experimental needs without wasting partial plates. The intuitive QuantStudio Absolute Q analysis software enables users to easily change and define experiment-specific parameters, name and track experiment details, and monitor the run from an easy-to-understand user interface. After the run is complete, return to view the results and apply powerful analysis software. QuantStudio Absolute Q digital PCR system from Thermo Fisher Scientific. Experience the future of digital PCR at thermofisher.com/absoluteq.
Thank God. We are going to just give the in-person participants 2 minutes to get back into the venue. And in the meantime, next presenter can start sharing the screen.
Yeah, so that will be Lukman, right? If I'm right? So, Lukman, are you on the call, please? Could you please share your screen? Your slide, rather, on the screen? You can share now, Lukman. You have the co-host right as well.
Good, thank you so much. Thank God.
I am trying to find you so I can spotlight you. Lukman. Okay, don't worry. I found it. I found you. Okay, go ahead, please.
Okay. One moment. Sure. Am I good now?
Yes, please carry on, Lukman.
Okay, great. Thank you so much. Thank God, and thanks everyone. It's an absolute honor to be here.
Sorry, Lukman, it looks like they're introducing you already on site. So.
Give me a short bio, if you don't mind.
Yeah.
Okay, thank you. Yeah, welcome back after tea, and so we're going to start our next session. And the first presenter in this session is Mr. Lukman Aruwarama from Imo. Apologies because I really think I messed that one up. So Lukman is the chairman of the board chairman of AfricaBP in West Africa, and serving as the managing director for 10 years. He's obtained his BSc in biotechnology management from the Pearson Institute of Higher Learning in South Africa, and he's got a master's in African Studies from the University of Ibadan. He's currently running his PhD in African Studies at the same institution. He's also the founder of the Malembe Hospitality Group and the owner of Malembe Lodges. Lukman is also the founder of the Malembe Foundation, an NGO advancing scientific and cultural education across Africa by providing support to the scientific and research community focused on changing the narrative on scientific output in Africa. So with that, Lukman, I'll hand over to you.
Hey, good morning once again. An absolute honor and pleasure to be here, and thank you for the opportunity to speak about a project we had worked for over the past 2-3 years. And thanks to AfricaBP as well for this revolutionary platform that we hope will, just transform how we do genomics and perhaps science in Africa. So my name is Lukman Arora Mimo, and today I'll be speaking about the project we are currently working on. We've made a bit of progress here and there, and it's on the hi-fi sequencing of African biodiversity, and with specific focus on our experience with our pilot study on the Africa star apple. But before I get into it, I'd like to maybe share a bit of insight on who I am. And the reason I do this is just to show that I'm by no means a core scientist. Beyond my BSc in biotech management, I've actually never spent time in the lab doing core research or even managing and coordinating any scientific research as such. And what I have really is the interest in just science, conservation, and nature. And of course, my strongest affiliation to science is the fact that I work with a genomics company in AfricaBP, and of course, I have entrepreneurship interest as well. And the reason I shared that brief insight into who I am really is to, just put it out there that if we have the right intentions on how things should be done and, filling the gaps around some of the issues across the continent, one needs not necessarily be a professor or even a PhD holder to drive some of these. Of course, this is by no means not acknowledging the importance of core scientists, because ultimately they do the work. However, the idea need not come from core scientists as such. So the motivation behind the project really for me was around, because I've worked in the science space over the past decade, I've realized that the motivation for science need not always be the need to advance one's scientific career progression. Ordinary individuals like myself who just have interest in science would motivate scientists to, do really interesting work, especially if one has specific interests and identify specific gaps. And of course, I've always been a proponent of localizing scientific effort. Over the past few years, I've been involved in other projects like setting up a scientific radio show, just enlightening the average citizenry in Nigeria on basic scientific concepts and giving them the opportunity to ask questions live on radio. And of course, there's a need for stronger collaboration across Africa between the scientists. And ultimately, science is built on data and capacity. And I think the more data we have out there, the more, the stronger the likelihood that African scientists will actually do really interesting work. So for this project, I live in a city called Ibadan in southwest Nigeria. And a few years ago, I came across this really interesting bird. It's called the Ibadan malimbe. As the name suggests, it's an endemic bird species to Ibadan, Nigeria, and I just felt like it's a really interesting bird. Obviously, the fact that it's endemic makes it extremely rare and probably endangered, because if the few habitat opportunity it has to exist gets lost, which is a very strong possibility, and chances are this bird could be lost forever. So I thought, is there anything we could do to, just steps towards ensuring the long-term existence of this bird? So I got in conversation with Dr. Aoyemi, who is the forest manager at IIT Ibadan. IIT is the International Institute for Tropical Agriculture, and they've been doing really amazing work over the past 5 or so decades on just overall food security across the continent. So I got into conversations with the forestry manager on, can we sequence the genome of this bird? He found it really interesting, and after lots of deliberation, we got to the realization that capturing this bird during the process of sample collection, there's a real risk that this bird could get harmed or even die. And we thought, is that really how we want to, conserve a very rare and perhaps endangered bird species by exposing it to the risk of losing its life? So we thought we do not necessarily have the capacity to embark on this project. However, we would like to explore other equally important or perhaps even more important African indigenous species that are exposed to equal level of threat due to forest loss and habitat degradation. So we thought we sort of highlighted, we listed out a few crops. Many of them are indigenous to Africa. Actually, all of them are indigenous to Africa. And then we tried to intersect these crops between economic importance, cultural relevance, food security, and medicinal application. And then, of course, referencing the IUCN list, we also try to, shortlist those species of endemic crops which are near threatened, or even endangered. And then we came up with a short list of 23 crops. And then we went further to identify, because we knew that we needed the funding and support to sequence 23 genomes. Obviously, that's a lot of genomes and would require significant funding. So we thought, for us to get future funding, we needed to do a pilot, and we sort of came to the conclusion that the white star apple was the right candidate. So if you are West Central or even East African, chances are you would be familiar with this interesting fruit. It has different names depending on which country. In Southwest Nigeria, it's called agbalumo. In the East, it's called udara. And every country has its name. And it's quite an interesting crop in that it's obviously an important source of livelihood, economic importance, in that whenever it's in season, you typically find market women sell and derive livelihood from this, and it has medicinal uses as well. And more importantly, the genome of this crop has never been sequenced, so there was near zero genomic resources publicly available. So we honed down on this and thought this is an interesting crop to sequence. So of course, we formalized our partnership with IIT, because IIT Forestry had the nursery where these interesting crops were being conserved within their premises. So we signed an agreement with the International Institute of Tropical Agriculture to work on these 23 crops, starting with a pilot of the Africa star apple. And this is just a bit of an insight into the importance of these crops. It has really important traditional medicinal uses, economic importance, cultural relevance, etc. So, and then we obviously swung into action. For us to do the sequencing, we needed to obviously collect fresh samples, which IIT was gracious enough to supply. And then once the samples were collected, we then moved on to the high molecular weight DNA, which was important and critical for the genome sequencing step. And for these, so this is the nursery where the sample was collected, and this in the tube is the high molecular weight DNA. Obviously, this was a really important learning process for us, because at our lab in Nigeria, we had never done anything like this. So there was a lot of trial and error. And eventually, thanks to the persistence of the laboratory team and support of IIT, we were able to successfully extract the high molecular weight DNA. And this DNA was sent to the AfricaBP NGS lab in South Africa. So the for the hi-fi sequencing, the PacBio Revio was used. And then after a few years into the project, I got into conversation with AfricaBP through Thank God. And then, you know, we sat on the data for close to 2 years because we just didn't have the capacity or the again, this genome had never been sequenced, and because there was no reference genome, we had to explore. And, you know, so Thank God suggested the use of the Omni-C technology for supplementing the hi-fi reads, which worked quite well. So the short read, the ONSO was used. ONSO is a platform from PacBio as well, was used for the Omni-C, and we generated really interesting data from there. So and then we were able to assemble the genome thanks to the support of Michael from IIT in Kenya. So he did the sequencing, the genome assembly and annotation, and we're able to achieve 97.5% genome completion, which was quite good for as a reference genome standard. So like I said, we had challenges with the annotation and assembly, and then I got in talks with Thank God, I think it was late last year. And then together we assembled a team from primarily IITA and had a few other institutions. And, you know, we're able to, well, this was led by Michael from IITA Kenya, and we're able to proceed to do the assembly of the genome. And I'm pleased to inform you that you know, this work is pretty much done. We're also going to be publishing the results of our work in the next few months. But in between, we also did a few other things. So one we did was we had a fellowship, because again, the reason why the project stalled for a while was because there was no capacity to, well, we didn't have the right capacity due to funding and just overall capacity availability to do the genome and annotation and assembly. So as part of the program, we organized a fellowship in collaboration with IIT and AfricaBP, where scientists from 5 countries, in total 10 scientists, were trained on the annotation and assembly using the Africa Star Apple as a reference genome. And we hope that over the next few years, should we need to do similar projects in the country represented, we'd be able to have we'd have the local capacity to make this happen. And we hope this would then in the long term prime, you know, the critical mass aggregation of bioinformaticians who can then do more of such interesting work across the continent. So we've consolidated this project into the indigenous tree sequencing project for Nigeria. And, you know, while we've been able to do the first pilot, we still plan to do 22 of such indigenous tree species sequencing in Nigeria. And hopefully we'll see where this goes and see if we're able to expand across other countries on the continent. So just to summarize, my admonition is that interesting work could be done without necessarily having major funding. The funding for this pilot project was zero, and all of it was funded primarily by myself with the support of AfricaBP giving us significant discounts, and, you know, and we were able to make this happen. So sometimes, yes, funding is a major gap, but with the right intent and drive, it's possible to do really interesting work. And of course, the importance of collaboration. Without the collaboration between Kaba, Malimbe Foundation, IITA, and AfricaBP, I'm not sure would have been able to make this happen. So again, an admonition to the scientific community, let's collaborate very strongly. And this collaboration hopefully will drive local capacity and local content. And when all these pieces fall into place, it's a lot easier to get the required funding to do more interesting work. So I'd like to appreciate the collaborating institutions, Malimbe Foundation, Nkaba, IIT, and AfricaBP, and specifically these people without whom we wouldn't have been able to make this happen. So thank you so much once again for the opportunity. And that's my email address. If anyone is interested in reaching out or collaborating, we'd be happy to. Thank you.
Thank you, Lukman, for that excellent presentation. So we'll move on to our next speaker. Our next speaker is Professor Mark Baxter. So Mark is the head of the Tree of Life program at the Wellcome Sanger Institute. He leads a major initiative to generate and analyze reference genome sequences from thousands of species across the Tree of Life. His work focuses on sequencing eukaryotic species, particularly in Britain and Ireland, through the Darwin Tree of Life project, and contributes to several international genome projects. Professor Baxter's research spans evolutionary genomics, especially of neglected non-model organisms, including parasitic nematodes, and explores genome evolution, symbiosis, and biodiversity patterns. He was formerly a professor of evolutionary genomics at the University of Edinburgh and is a fellow of the Royal Society of Edinburgh. His program aims to unlock genomic sequence of life on Earth to aid the understanding of biodiversity conservation and evolutionary processes. So with that, we will hand over to Prof. Baxter. Thank you.
So can you hear me?
Yes, please go ahead, Mark.
And you can see my slides?
Absolutely.
Brilliant. Okay. So I am so sorry not to be with you in the auditorium. It's a personal disappointment that I'm not actually there in person. I'll say something personal to start off with. I was involved with many other people in the UK in the 80s and 90s in the anti-apartheid movement. I was arrested for protesting in front of the then apartheid South African embassy. And it's always given me great pride and joy to see South Africa free of apartheid. So that's my politics. I come from a very open and very, I hope, equitable background. But for my science, I've ended up in this position of amazing responsibility. So I run the Tree of Life program at the Wellcome Sanger Institute. And our goal is to sequence everything. So we're part of the Earth BioGenome Project. And our goal is to partially do the sequencing, but really mostly enable and promote the sequencing of all life on this planet. So you'll all understand that the planet is undergoing a biodiversity crisis because of the climate emergency and because of increasing pressures on wild landscapes because of anthropogenic habitat destruction. And we think that sequencing genomes has to be part of that solution. I'm a genome biologist. Of course, if you ask me what the solution to something is, I will say sequence it. That's what you do. If you ask a carpenter what should be done, the carpenter will produce the hammer and will say, let's build something. So I'm a sequencer. I think we should sequence life for the future of life, which is the strapline of the Earth BioGenome Project. So the Earth BioGenome Project is a collaboration, a network of networks, and its idea is to try and promote genome sequencing throughout the world. And we are now in what we call the start of phase 2 to try and build this digital library of life, this idea that we can sequence everything and use those data for a huge number of different projects. So we had just published last week a paper in Frontiers Biology which is announcing what we think we might do for phase 2. So we haven't even got to the end of phase 1 of Earth BioGenome Project, but we're heading for phase 2. So what do we think will happen in phase 2? We think acceleration of sequencing must happen. And what we've heard this morning already from Nigeria and South Africa suggests that that is happening. Those are amazing projects producing beautiful data for indigenous species. So both the 1K South Africa and the Nigerian trees, awesome. But what we want to do in the future is move from sequencing 1 or 2 or 3 or 100 to sequencing tens of thousands. So we propose that for phase 2, we should sequence 150,000 species, which in the end will contribute about 1 for each genus of species, and also the sequences of many species of conservation, economic, or biotechnological importance. We also hope to build for phase 3, which is sequencing everything, all 1.67 million species on this planet, by collecting about 300 species. So this means we're going to have to really motivate and mobilize a huge team of taxonomists and parataxonomists based in and sequencing in the biodiverse rich nations of this world to prepare for phase 3. So there are huge challenges in this project. One is that we don't have the sequencing capacities in all the biodiverse nations of the planet. And so we have to do 2 things. One is build capacity, and the other is build capability. And that's what you've heard from the first 2 major talks this morning, is about building capacity and capability in Africa, and that has to be repeated across the globe. And when we get there, I mean, imagine we're there and we have 150,000 genomes. What we're going to have, well, as has all been presented, we'll have new tools for monitoring and conservation of biodiversity. We'll be able to track species in space and time. We'll also be able to track the genetic health of species. So, for example, deciding whether 2 populations of endangered species should be brought together and bred to enhance the genetic health of that species, or whether they should be kept separate because they contain or have unique adaptations. So we'll have new tools for monitoring and conservation, and we can imagine all sorts of things: remote monitoring, automated monitoring of air and water for the DNA of species we're interested in, but also new ways of identifying and declaring species. So we're going to do lots of species discovery. The other thing we'll get, and this is really important, is diverse new biologicals and biomaterials. So nature has solved the problems of living on this planet millions of times. Each species is a successful experiment in how to live on this planet. And we can learn from those successful experiments, whether it's by taking a disease resistance gene that's present in a wild crop relative and breeding it into the crop, or whether it's taking a biomolecule that's made by species. For example, fungi make lots of antibacterials and antifungals, or spiders and moths and butterflies make silks, and using those molecules as part of a new bioindustry. So thinking about for Africa, what this means is that you have the ability to step straight into a new bio-inspired industrial future, not reliant on oil, that develops new medicines, new bioproducts for your economies. And for me, I'm a scientist at base. I'm a genome scientist at base. We will get an astonishing new understanding of life. Currently, our idea of what a genome is based largely upon model species: yeast, mouse, human, Drosophila melanogaster, the fly, Synrhabditis elegans, the worm, Arabidopsis thaliana, the plant and the only plant. But as we sequence new genomes, we find astonishing things out about other species and about the biological, genetic, mechanistic toolkits that they have for surviving. And we get an understanding of how this amazing planet with its amazing diversity has evolved. So as I say, what we want to do is build an open lending library of life. So a library where you can go to the librarian, the INSDC, European Nucleotide Database, and request the genome of the species you're interested in and read the whole genome from beginning to end, complete, high quality genomes. So that's what we're going to get to. So at Tree of Life at Sanger, we're involved in a whole host of projects. We help the Earth BioGenome Project with informatics and other things, but the Earth BioGenome Project doesn't actually do sequencing, it coordinates projects. Our major project is we call the Darwin Tree of Life Project, and that aims to sequence all of the eukaryotes, the organisms with nuclei that live in Britain and Ireland. So we're aiming to get to 8,000 species by 2026. That's only a year away. Currently, we're at 3,000, so we're getting there. We have some other projects which are funded to look at different components of biodiversity. The Darwin Tree of Life is a regional thing, and we also work with the European Reference Genome Atlas, which is all of Europe. We also have projects which have a taxonomic focus. So Project Psyche aims to generate reference genome sequences for all the Lepidoptera, the butterflies and moths of Europe. That's about 11,000 species. We're currently just over 1,000, so we're doing really well with Project Psyche. And that will feed into both evolutionary biology, climate change biology, but also helping agricultural systems, because moths are often, and butterflies are pests of the horticultural system. We have projects which are based around ecosystems or modes of life. So the Aquatic Symbiosis Genomics Project has sequenced corals and sponges and deep sea clams, many species which live in symbiosis with other microbial organisms. Here we're sequencing both the macrobiont, the host, and the microbionts, the bacteria and protists that these species live with. We also work in the agricultural area or the agroecosystem area, the project called AEGIS, which is Ancient Environmental Genomics Initiative for Sustainability. The idea of this project is to use ancient alleles that we can sequence from sediments and soil samples, which go back 40,000 years or more, to understand what the genetic composition, not just who was there, but what genes they have, of crops and cropping systems way back in the past. And so we're sequencing a number of genomes to offer modern references to map those two. Okay, so we're involved in a whole bunch of projects. The one that's most advanced is the Darwin Tree of Life project. So when we came to think about how the Tree of Life should sequence things, we decided we should sequence everything, yes, but there are ethical and political and legal issues about sequencing species from around the globe. We could have gone to sequence we could have suggested sequencing all of the things on the ICN red list. But those are your species, not ours. Those are species which, yes, they're the patrimony of the world, but they're also endemic to particular areas, Africa, wherever. So we decided instead to focus on our own nations and on this little archipelago of islands off the west coast of Europe, which is home to the human nations of Britain and Ireland, but is also occupied by about 70,000 species. So we decided to focus in on Britain and Ireland and the seas around. This gave us access to about 40% of all the taxonomic families on the planet and about 70,000 species. And the beauty is that in these islands, where organisms exist, where they can be found, is really well known. So it meant we could collect really quite efficiently. And so we put this forward as an idea that we should sequence locally, but think globally. So we're going to develop tools and the approaches and the protocols to sequence genomes of any species as efficiently as possible, and do that locally on our own species and make those data available to the world. So thinking globally in terms of making sure the data are open access. The Sanger Institute is not a biodiversity organization. We do sequencing. And so we've collaborated with biodiversity organizations such as the Botanic Gardens at Kew and in Edinburgh, the Natural History Museum in London, the University of Cambridge's Wytham Woods field site, which is a lovely ancient forest, ancient oak woodland, with Nature Scott, with Marine Biological Association, with the Zoological Society of London, and also with culture collections like Cabby, where they have lots and lots of, especially fungi and protists in culture. And then we collaborate also with analysis teams at the University of Cambridge, Edinburgh, Oxford, and also EBI, the home of the European Nucleotide Archive. All of this has been funded by the Wellcome Trust, backed up by local funding for each of these organizations. So it's been an amazing collaboration, a true partnership, where yes, the Sanger's done most of the sequencing, but actually it wouldn't have worked if all these other people hadn't been involved. And what we've invented, so our aim was to produce high quality genomes. So the Earth BioGenome Project has defined a reference quality genome. The full definition is at the link down below, but the basic story is that the contig N50, the raw contigs that come out of the assembly, have to be 1 megabase. So at least half the genome has to be in contigs of a megabase or more. The scaffold N50, which we get by linking those contigs together, usually with high C data, the chromatin conformation capture data, has to be approaching chromosomal. So we like to have more than 95% of the sequence in chromosomal molecules. And we have to have it very high quality, so less than 1 in 10,000 errors. So that's a Q40. So that's 10 to the minus 4. There's an explanation as to why it's 40 and not 4. So 6C Q40 is what we're aiming for all our species. Sometimes we don't make it, sometimes it's really hard, but that's what we're aiming for. This is how we do it, and I'll just quickly go through this. We acquire a specimen. This is our biodiversity partners. They identify it, and we also do a DNA barcode of that specimen so we can track it through the system, but also so we have an instant assessment of the taxonomic accuracy. So this really requires engagement from taxonomists. Once you've got all the easy to identify things, you really are in the expert realm, and it's really important that we work with those experts and get them engaged in these projects so that they see benefit for themselves and their work from our sequencing. We then do DNA and RNA extraction, and as has been mentioned, this can be very difficult. It can be very complicated. It can be hard to get the DNA free of contamination. The snail that's on the picture here comes with lots of mucus, lots of slime. Slime co-purifies with DNA in most preparations, so I have to get rid of the slime. We then do genome and transcriptome sequencing. So we use mostly PacBio for our long reads. We also use about 10% of the time we use ONT. We do high C, which is chromatin conformation capture. I'll come back to that in a minute, which is a way of linking the contigs together to make scaffolds. And we also do RNA-seq just for annotation, not for functional genomics. We then publish all our data as it comes off the machines. Before I've even looked at, before any of us have looked at it, you can see it publicly on our Tree of Life QC website. And that gives us data quality assurance, but also tells people that we are progressing with their species of interest. We then do a primary assembly of long reads, just as you heard in the South Africa 1K project. We then scaffold with high C. This is an important step which allows us to get to chromosomes. We then curate each genome. So each genome is looked at by a human being, aided by AI and clever software programs that basically corrected. And we're doing about 20 to 50 corrections per gigabase of assembly as we pump these genomes out. And then we submit it immediately and openly to the ENA so that the data are available for all. So the process from sample acquisition to ENA submission should take weeks. Actually, usually on average, it takes about 6 to 9 months because there are always interesting problems. And also we have a lot of species going through our queue. The genome is then annotated in Ensemble. Genome is then annotated in Ensemble to get gene sets and repeats. We then publish those gene sets and repeats and all the metadata through an open data portal, for example, the Darwin Tree of Life portal. And we also publish a little genome note. So this is a small note which says this person collected the sample, this person identified it to species, these people did the extractions, these people did the sequencing, these people did the assembly, and this is the project who did it. And this is an open access genome note which is available to all and gives credit. And then we're building this library of the Tree of Life, and then things are open for analysis. So this is a series of genomes which we've assembled just recently. So this is from the last short while and published in Wellcome Open Research. These are high C plots or chromatin conformation capture sequencing. This gives you a picture of which bits of the DNA are next door to each other inside the nucleus. And it so happens that chromosomes usually are wrapped up in little microdomains inside the nucleus and only interact with themselves. So the butterfly map on the top right is a beautiful example of this. Butterflies sequence relatively easily. And each of those dark squares is a chromosome. The sequence in the middle, so this is about a 500 megabase genome. And each of those little squares is a chromosome. And the chromosomes are interacting with each other and usually not with anything else. The ones at the top are the sex chromosomes, and you'll see that the W chromosome has weird interaction with everybody. It's very repetitive. But you can see different species have different patterns of high C. I point out the flowering plant at the bottom left, where you see lots of lovely X's in the boxes off the axis. And that's because the plant chromosomes have a particular conformation in the nucleus such that the arms of the chromosomes interact with each other, as do the telomeres, as do the centromeres, in very independent ways. Sometimes the high C isn't very good. The moss in the bottom left hasn't got very strong signal, but it's still enough to tell us that this is a good assembly. And we can do it even from difficult samples. The deep sea clam, this comes from hydrothermal vents. This deep sea clam had to survive being dragged up from 3,000 meters down before being preserved, and yet we're still able to get a reference genome out of it. I'll just illustrate by a couple of really things I think are really exciting. So this is the biggest genome we've assembled to date. It's the biggest genome anybody's assembled anywhere. It's a hemiparasitic plant called the European mistletoe. There are mistletoes all over Africa, so they'll be related to this. Most of them have large genomes. This one has a wonderfully large genome. It's 94 gigabases. That's 30 human genomes. 30 human genomes. Those three little pink dashes show three human whole genomes, 3 gigabase genomes, lined up against chromosome 4, which is 12 gigabases long. And the cross pattern here is precisely this pattern I was mentioning, where the centromeres of each chromosome interact around the nucleolus, and the arms of the chromosomes dangle into the, if you like, into the nuclear cytoplasm and interact with each other. So this sequencing this genome was fun because it broke every software tool. There's a little trick that software engineers use saying, how big do I think the sequence is going to be that's going to fit into this this particular variable in my program? And previous to this, the biggest chromosome anybody ever thought they'd analyze was about 2 gigabases. We had to rewrite the programs or get the programs rewritten so it would work with genomes with chromosomes up to 12 gigabases. So it does not now. We can also sequence tiny things. So most life on this planet, unlike us, is absolutely tiny. It lives in the spaces in between soil grains and the sediments, and also those tiny little flies that are so annoying. So those organisms have very little DNA per specimen, much, much less than is acceptable by the long read and high C technologies. So we've been developing with Chris Laumer in the Natural History Museum methods for getting genomes out of tiny things. And this is one of them. This is Odontocalamus thalassophagus. It's a predatory nematode that lives in the muds on the beach. It's a relatively big nematode at 2 millimeters long. It's got something like 1,000 or 2,000 cells per animal, so only about 2,000 copies of the genome. We're able, using an amplification method called PIMS, to generate reasonable data, to get a reasonable contig N50, not quite a megabase. But then we can take 22 individuals, just 22 of this nematode, and get high C data and turn that into chromosomes. So this is the first chromosomal assembly of a nematode from its group, the Inoplia. But just say we now think we can sequence almost anything using our special methods. So this technology is available to all. It's a set of clever PCRs, but so we think we can do anything, whether it's giant genomes, tiny species, we can do them all. The exciting thing for us, and it was mentioned in the South Africa presentation, is that when we sequence these species, we're pulling them from the wild. And so they come along with their own collection of what we call the cobiome. And so that's things in their guts, the microbiome, things that are parasitizing them or pathogens on them, so other bacteria and protists that live on them, and also anything that happens to be sticking to the surface. So usually we try and avoid, for example, sequencing the gut because we want to sequence the whole organism, but sometimes we can't avoid it. This is an example of a sponge, Spongia lamella, the elephant ear sponge. Sponges have very strong organization, but they also are associated with hundreds of bacteria. They live intracellularly and extracellularly. So it's not possible to sequence the sponge without sequencing its microbiome. And in this case, we have a chromosomal assembly of the sponge in 10 nuclear chromosomes. And we also have the mitochondrion, of course. And then we also have 127 microbial genomes. So these are bacteria from across the bacterial tree, really very varied. And 45 of these are circular, which are the blue dots on the tree there. And 85 of them are high quality as judged by gene completeness compared to representatives from their group. So they're usually all undescribed bacteria. Some of them we know what they are. But from this one sponge, we got 128 genomes, if you like, 129 if you count the mitochondria as separate. So we always try and assemble everything that's in the sample. And so while we've submitted 3,000 genomes from Tree of Life, we've actually also submitted about 12,000 bacterial genomes. So we're harvesting everything, not just eukaryotes. So just last month, the middle of last month, we celebrated our 3,000th reference genome assembly, and we sequenced everything from protists, including algae and parasites, through simple plants, angiosperms, flowering plants, crop plants, and all the different kinds of animals, from sea jellies to vertebrates, mollusks, annelids, and of course, lots and lots and lots of arthropods. Arthropods comprise about 60% of all the species on the planet, and we've sequenced about 40% of our genomes are from arthropods. You'll see a particular concentration of red bars there on Lepidoptera across to the far left. That's because we have a particular project looking at genome evolution in Lepidoptera. This also includes the Psyche genomes from Europe. So that's 2.7 terabases of genomes assembled and made public. So all these are available on ENA. And we generated 511 terabases of PacBio Hi-Fi, 50 terabases of ONT, 2,000 terabases of Hi-C, and 78 terabases of RNA-seq data. Again, all made public. We celebrated this by producing this absolutely horrible picture. No, it's a wonderful picture with each one of those little pictures, which are from phylopix.org, representing the species we sequenced, and they're present on the picture in approximate abundance according to what we sequenced. But we're really proud of having got to this 3,000 genomes step. We're increasing our sequencing rate all the time. So this year we hope to sequence 1,400 genomes in total. So we hope to get to something like 3,500 this year, 1,500 of which will be produced this year. And so this exponential increase is just what we need across the world. We're making all our protocols open. We're making all our approaches and policies open, just like the South African project. And so hopefully you will all join us in generating 3,000, 30,000, 300,000 genomes from Africa. I get to talk, and unfortunately I don't get to be with you, which is a great, as I said, a great personal disappointment for me. But I really have to thank everybody at Tree of Life, at Sanger, all of our collaborators across the many, many other projects we're involved in, but especially the people who do the collection and identification. Their expertise is absolutely essential and unrivaled. And then we are really strongly supported by Sanger scientific operations teams who run the sequencers, especially the people who run the long reads, who've dealt with some of the most horrible samples they've ever seen. And we're funded by a large number of people, mostly the Wellcome Trust, but also Novo Nordisk, the Moore Foundation, UK Research and Innovation, and the Horizon 2020 DEFRA collaboration. Thanks very much. I'll stop the share.
Thank you, I think we've learned a lot about the science of life. Our next speaker is Prof. Brighton Samatanga, who is a molecular and computational biophysicist and life sciences entrepreneur who founded the Biotech Institute in Harare, establishing next-generation sequencing, clinical diagnosis, agroanalytics, and bioinformatics laboratories, and developing a cancer genomics bioinformatics platform to advance precision health in Zimbabwe. He has previously served at the University of Leipzig, first as a senior research scientist, and then a full professor of physics for biological and soft materials, with research focus on single molecule spectroscopy and elucidating CRISPR-Cas9 gene editing mechanisms. He holds a PhD in molecular biophysics from the Swiss Federal Institute of Technology and executive MBA from Quantech School of Business, and is a member of several organizations such as the German Biophysics Society. So I welcome Prof. Brighton Samatanga to share his screen and talk to us about
Craig?
Yeah.
So I think Samatanga speaks next tomorrow, if I am correct. So we have Alexandra. We need to go to Brazil now. So is that correct? Is that what you have on your?
No, that's not what I have on my.
Because Samatanga is not on the call. So I think he speaks tomorrow, Tuesday, at 3:40 South African time. So what I have here, maybe it might be different. I have Alexandra Alexo, if that's correct. Is Samatanga on site now?
Is that what is the program say now? The program you have if
Yes, I'm here.
Okay, you're on site. All right.
Okay, no, it's fine. It's fine. We'll see if Alexandra Alexio is on site.
Yeah, he's on the call.
Then we will, and we'll just check that. Maybe I'm still working from an old program. Okay, so then we'll move on to Alexandra from Brazil. So let me introduce him as a speaker. So Dr. Alexandra Alexia has graduated in biological sciences at the State University of Campinas, Brazil. He has a master's degree from ecology and is from the same university. He also has a PhD in zoology. He also has from the University, Louisiana State University in the USA. He has a lectureship in evolutionary biology at the University of Helsinki and is currently leading the environmental genomics group at the Institute of Technology in Vail, and coordinating the genomics of Brazilian biodiversity initiative. He has experience in the field of genomics, animal genetics, and zoology, and is working mainly in the following areas: evolutionary biology, population genetics, molecular systematics, taxonomy, subjects on which he has produced over 350 publications. And this is in communications and journals and books, newspapers, magazines, and in events in Brazil and abroad. So with that, I'll hand over to Alexander.
Hi, everyone. Thank you very much for the invitation to talk to EBP Africa. It's a real honor to share our experiences with our African colleagues. I will try to.
Before you continue, is it possible for you to put it in present the view, please?
Yeah. So can you hear Alexandra?
Hi.
Yeah. Craig, can you hear him on site? Alexandra, he's speaking now.
We can hear him on site, but the slides we are seeing.
Okay, Alexandra, do you want to put your slide, make it a slideshow?
It's already there.
Yeah, it's showing like two columns of slides and 2 rows. So I suspect you might want to maybe remove it as column of slide or take it as so that it fits the page. one way I usually do this is that I just export it to PDF, and that way you can just share it as a. So I think that is also one thing we can do. So it's showing us 2 columns and then 2 rows with so many. So it's not showing us a single slide.
Okay, do you want me to convert to PDF then?
Yeah, convert to PDF and
Okay.
then share your screen and then present it as a PDF. I think that would be better.
Can we try stopping the display?
Okay.
Yeah.
Can I share me? I don't know if I do. Can I share my screen and also do the
No,
PDF?
stop sharing. Then save your slide
Okay.
as PDF. You just to save as PDF, and then open the PDF and present.
Okay.
Yeah.
Gotcha.
Just stop sharing, save your slide as PDF, and then open the PDF. So when you save it, it will tell you whichever thing you see is what we are going to see. Then you check whether you're fine with it.
Okay. I will convert it now.
Yeah, we'll just wait for you for a few seconds here.
Oops. It's... Okay. Got a PDF file.
Yeah. And is it showing us one full, when you open it, is it showing us one full, it's not showing us columns, right?
No, I guess not.
Yeah. Okay. All right. That's fine. Yeah.
Can you see it now?
Yeah, perfect. This works fine. All right, carry on.
Okay. Well, thank you again for the invitation to be talking to you. So I'll be talking today a little bit about the Brazilian the Genomics of Brazilian Diversity Consortium and what our experience has been with doing genomics in the Global South. So why genomics matters? I think we are in an audience that knows really well, why genomics matter. But it's all about how society perceives the importance of genomics. So it's actually now a very powerful scientific tool and public policy instrument that needs to be shared with policymakers in general, particularly in biodiverse countries like Brazil and most African countries. This is also important because it can uncover the full potential of these organisms in terms of not only allowing us to conserve them, but also to develop biotechnological applications that turn out to be very, very important. I had a slideshow here that, of course, is not showing because of the PDF, but it doesn't really matter. Another point is having a grip on genomics nationally can really be important to launch several biotechnological applications and contribute to the bio-revolution worldwide. That's why it's a matter of national interest to really have genomics as a very well-developed discipline in many countries, particularly countries that are really, really biodiverse. Another reason why genomics matters is that from now on, it will be necessary for countries to report not only on species extinctions, but also on genomic variability, genetic variability. How is it going with the years? Do we have enough genetic variability to adapt to climate change? Questions like that are now very important because it's part of a CDB targets that have been recently developed. So it's another reason to we also need genomics to monitor species genetic diversity through time worldwide. So we all know that even though genomics is a very powerful tool, doing genomics in scale is very challenging. You need a lot of financial resources. You need to establish partnerships, public-private partnerships. You have to have governance over, the different aspects of producing genome sequences, and also you need to cope with the pace of technology, which is really mind-boggling these days. So it means that you have to have access to very advanced sequencing machines, but also you need to have access to pipelines, to computational resources, and things like that, which are all not only very costly, but in general, as a whole, very challenging to really manage together. So that's how the Genomics of Brazilian Biodiversity Consortium came about. It's a partnership between ITV, a private research institute, and ICMBio, which is the National Brazilian Authority for Biodiversity. And it's GBB is divided up into two main hubs: the Environmental DNA Hub and the Genomics Hub, which in turn are subdivided into four sub-hubs, so to speak. The production of barcode references and the actual map-by-coding studies, which together are part of the Environmental DNA Hub, and the conservation and bioeconomy sub-hubs, which are part of the Genomics Hub. And of course, to support all that, you need a very sound data management. You need very sound data management solutions. You need curation of the samples and also the sequences that are generated. And you also have to have a grip on the analysis, which you have to do to not only come up with the genomes, but also to analyze them together. So altogether, GBB is an investment from the Vale company of $25 million between 2023 and 2028. And the main goal is to use genomic tools for the conservation of Brazilian biodiversity and promotion of the bioeconomy. Talking a little bit about ITV, the Vale Technology Institute. It's a non-profit research institute maintained by the Vale company, which is a mining company, a multinational mining company. A few numbers here. The institute is now just turned 15 years old. So here are a few numbers, but I won't stay too long here on this slide, just to show you a few things about ITV. And of course, ICMBio is a very strategic partner in this genomic consortium because it's the national Brazilian authority entitled to organize and to manage biodiversity protection in the country. ICMBio has a national scope. Brazil is a big country, so it's a very important partner because it has governance over the entire country, over all Brazilian biomes, for instance. It also entitled to the management of all federal protected areas throughout the country. It's also in charge of the national action plans for the conservation of endangered species. And it already congregates hundreds of researchers and also ICMBio itself has 15 research centers of its own focused in different taxonomic groups. And ICMBio also hosts the Monitora project, which is a long-term monitoring, biodiversity monitoring program across Brazil. And of course, in addition to ICMBio, GBB already has a network of over 110 partner institutions, which have collections, biobanks, and researchers who are specialized in different taxonomic groups. GBB is a WordPress, like VBP Africa, GBB is a WordPress genomic project. It's already associated with the EBP itself, with the VGP and other initiatives, genomics initiatives throughout the world. And this exchange is extremely important so we can all be on the same page when it comes to producing and analyzing genomes. Just to give you an idea about the lack of genomic data for Brazilian species, this is a figure from a recently published paper by our group. And you can see here the small dark green circles. They show here just the number of nuclear genomes available for Brazilian vertebrate species. You can see that it's a really minor portion of what's available, which is mostly mitochondrial data, as shown here in the light green circles. And for some species, you already have mostly mitochondrial sequences, but whole genomes are unavailable yet for most Brazilian species. And the data available so far, particularly on the whole genomes, this data is produced mainly outside of the country, like as shown here. For all vertebrate species that occur in Brazil, only a minor part of the genomes are really produced in the country. And in B here, we have just the situation for the endemic Brazilian species, which is, again, most of the genomes are produced elsewhere. And I think there are many reasons for that, but I want to share with you here this table from this paper of ours that I just mentioned, and it shows the cost of doing genomics in Brazil. It's really, we end up paying more, up to 3 times more to do genomics in Brazil than our US colleagues, for instance, are paying in their own country. And this, of course, varies across, depending on the reagents that you buy, but it's a situation where makes it really, at least on the surface, look like, okay, it's not really competitive to do sequencing in Brazil. But this is a well-known pattern. So as our colleagues here have shown, they already pretty much nailed down the situation that when you do genomics in the Global South, because most of the reagents are not really from the Global South, they come from the North, there's always the middleman. So we have to pay more for getting the same supplies. But on the other hand, here in this map at the very bottom, it shows that there's a really high positive correlation between the number of sequencing centers available in different countries and the number of genomics projects that are carried out in these countries. Pretty much when you have a place to do your sequencing, you're going to have enough projects there to use up that demand. So a little bit about what GBB has produced so far in over 2.5 years. We have already 800 genomes from 450 species so far, and we are counting. Most of the genomes are actually shown here, super barcodes, which are going to address the lack of barcodes for supporting eDNA studies. And we also have a lot of resequencing for population level genomics. And we are also carrying out eDNA studies with in different protection areas across the country. Looking ahead, so far, there are 125 different projects under GBB, contemplating over 700 species of animals and plants. And here you have a little bit about their, on the left, their taxonomic distribution across the different groups, and also to the right, across different IUCN threatening categories. So actually, most species are actually being sequenced are of low concern, because they are the ones that actually we need super barcodes to support their detection by eDNA studies. We are also producing, we have the target of producing 80 high reference genomes at the chromosomal level. And here's pretty much what we have done. This is already outdated, but it tells you, it shows you, and also there is a comparison here with humans at the bottom of the left column. But actually, this is just the beginning of the production of several reference genomes at the chromosomal level for Brazilian species. So going back to the problem of sequencing in the country, why, if it costs more, up to 3 times more sequencing in Brazil, why do we have to sequence locally? And also, not only sequence locally, but I would say perhaps assembling genomes locally is actually even more important than sequencing locally. A few points here to consider. Sequencing and doing genomics locally, you can be sure that your sequencing efforts will be aligned with with the country's own agenda and priorities, which is very important. Along with sequencing and assembling genomes, you have to develop a lot of scientific structures such as biobanks, genome banks, sequencing centers, and open data platforms. And perhaps the most important thing, you are going to train human resources specialized in genomics and bioinformatics locally. And actually, this will support the development of the biotechnology industry, for example, not only producing genomes for the sake of it, but also you're going to end up with professionals that can be used in different areas supporting the biotechnology development. You can also strengthen governance over genetic and traditional heritage in your own country and your own continent. And of course, you're going to have to establish and to strengthen also national networks, which are all and should be connected to international scientific cooperation networks as well. And to give you an idea, just over 2.5 years, GBB has already increased 200% the number of genomes that are assembled in Brazil. Of course, it started with a very low baseline of just one, I think 10 reference genomes produced in the country, but it shows you that in little time you can accomplish a lot in terms of increasing the number of genomes done locally. Before I leave you, one important aspect to consider is, okay, how can we finance genomics globally, particularly in the global South countries? And I think GBB is an example that companies can also have their share in this effort. And I think this is really lined up with the EBP sequencing goals for the phase 2. I think the private sector has to be more involved, and we have to be really creative to find ways to finance genomics. But one important thing to consider is that you can gain a lot more. You can multiply the benefits of doing genomics if you do genomics locally, if you really involve the local communities in a very strong way. And I think this is one important message that I think I would like to leave you with that. Thank you very much for your attention, and feel free to get in touch. And thank you again for the invitation to talk to BP Africa. Thank you.
We are going to go into a panel style Q&A for our three speakers in the session. So they're all online. So we'll open up the floor for any questions to the speakers that were presenting this morning. Thank God, are there any questions online? I see you posted quite a few questions yourself.
Yes, absolutely. So let me see if there is any question before that. So I'm just gonna try to. I think one will be a comment, partly because we're involved. That will be the presentation by Lukman, and yeah, the genome is now finished up, and it will be published soon. And it's also so far, to the best of our knowledge, the first genome plant sequenced fully on African soil, in terms of plant having all the whole data using Hi-C and PacBio. So I think the second question goes to Mark. So Mark, I think you have a slide that shows so many projects that the Sanga TOL is involved in. So how many basically in total, and also what is the number of species that have been sequenced based on the DTOL project versus other projects? Because you mentioned, I also saw that on the social media the other day about the 3,000 genomes that have been sequenced by the Sanga Institute. So how many of these are like more of the national genomics program, and how many are also other collaborations particularly?
Thanks, thank God. So we have collected and have specimens in the freezers in Sanga for about 11,000 species. This includes about 8,000 for Darwin Tree of Life. And we have about 6,500 sequences in sequencing or completed. So we have a stock of species to sequence. We announced the release of the 3,000th genome in the middle of August, and that was just over 2,000 Darwin Tree of Life genomes, just under 400 Project Psyche genomes, and then the other genomes from a number of other projects. The interesting thing is that the number of other projects includes projects where we can sequence a species in 3 different projects. So we can sequence a sponge, which is part of ERGA because it's a European sponge, but it's also found in Darwin Tree of Life area. So it's also found in Britain and Ireland, and it's also a symbiont species. So we could count many of the species 3 different ways, but the official Darwin Tree of Life ones is just over 2,000.
Thanks. And also one of the things which again you do know, and also is you pointed it out in the slide, is as we sequence more species, the need for curation and publishing them rapidly need to increase. So how do we do that? Because I mean, are we going to use AI? I know that there are challenges to doing that, because curating it is one of those things that requires human intervention. But as we move to, for instance, in the phase 2, 150,000, that also becomes quite a lot. So can we have a platform, or is this, in terms of curation, whereby we could have like an AI system, you pop it in, and it may not be as high quality as human, maybe, but
absolutely. So those well, 2 things. Yeah. So on the curation part, absolutely, we believe that AI ought to be able to fix the errors introduced by the assembly programs. The assembly programs are all written for ordinary diploid organisms and actually focused on assembling human. So focused on assembling a genome with the sort of human pattern of repeats and segmental duplications and GC content. So as we move to weirder and stranger and more wonderful genomes than the human, then we are hitting problems. So AI will help, and we are already using AI tools at the Wellcome Sanger Institute to aid curation, but we're not there yet. The training data is what we're building at the moment, and we will train better AI tools to help curators. What we're using for at the moment is reducing the number of issues that the curators actually have to deal with. And we've found that actually training individuals in curation is a really rewarding thing. So we've had several curation workshops where we have trained up curators. And what we do is a dual curation. So these amateur curators, let's call them, these new curators, will curate a genome and will submit it to one of the experts, whether at Sanger or elsewhere, for validation. And that dual curation really helps the new curators, the trainees, to learn, but also distributes a load. So for Project Psyche, for example, all the Lepidopteran genomes are being curated by Psyche members across Europe, and that is a really powerful way forward, and that's something that drives success. In terms of publication, so at the moment we use genome notes, which are these short announcements in Wellcome Open Research. Wellcome Open Research is only open to Wellcome-funded organizations, researchers, and it also costs real money. We don't see that cost, but the Wellcome Trust is paying Wellcome Open Research to publish these. If you want to publish a genome note in one of the other kinds of journals, so for example in Giga Science or in
scientific data
or genetic data, or all these things, it costs you real dollars to do so. And that is a price on top of doing the sequencing. So in some cases, the price could be the same cost as doing the sequencing. And so we're moving towards the idea of building a truly open genome note publishing platform, which will be free to submit and free to view, so proper open access, but which will not include human or scientist formal scientific review. This is one of the problems we've hit with Darwin Tree of Life, is that we have now burnt out all the possible reviewers in the community. We have published nearly 1,200 genome notes, and there are people come up to me at meetings and say, please don't send me any more genome notes. I have reviewed 100, that's enough. Yeah, and I think we will get to that with everyone. So what the Open Genome Note platform will do is do a technical review based in computer. So is your data public? Is the genome as good as you say it is? Are the quality metrics meeting EBP standards? Have you made a statement about the ethics and legality of your sequencing? And we'll just give a badge of honor, genomes that pass, but the genomes will then be open to public review. So we'll have a comment system.
Yeah, thank you, Mark. So Craig, I do have one more question for Alessandra, if that's okay. Alessandra?
Before you continue,
okay.
Yes, it's here.
Online or in the room has
okay, all right, go ahead on the on-site participant here.
Anybody on site for the question? Or if not, then Thankard, you can continue.
Yeah, so I think Alessandra, I think this is more to you. You talked about the cost, right? So I understand the cost because we in AfricaBP we started from the bottom up, and it's a very grassroots, and then now bottom up approach. So I understand the cost when it comes to sequencing. So how are you guys managing it? Because when I look at the cost of sequencing in the US in that slide you showed, it's 3.3 times or more than what it is in Brazil. So how does it work? Do you guys still do that at that level of expense, or were able to leverage on some opportunities to bring down the cost at the local level or national level? And also, do you also manage to bring the government involved already at the ITV and the GBB program?
Well, thank you for all these very insightful questions. And well, it comes down to budget. So we have a budget that's strong enough to allow us to cope with the higher costs. However, I should say that over the past months, we have been able to negotiate with a local PacBio dealer much smaller costs than we used to have. So there is hope, I think, depending on the volume of your purchases, you may be able to negotiate. So I think this is good news. But I do think that we need to make a much stronger case to companies such as PacBio, Nanopore, and Illumina to find ways to get around the middleman, so to speak. But I don't think we have done nearly as much as we could do. And also, you also mentioned the costs of I really forgot about the second part of the question.
It's about do you have any investment from the government? Because looking at sustainability,
right.
You know, how has that happened for you guys as well?
Yes, so we have been involving the government, the Ministry of Science and Technology. But I mean, in terms of resources, their capacity is really limited nowadays. Just to give you an idea, the highest grant that they offer, for instance, cannot cover a PacBio machine, so to speak. So even though there are other ways of buying, sequencing machines, like modern sequencing machines, the fact is that not many people are applying for that, money. So there's also room for improvements. But I think to be honest with you, I think the academics in general, and I am an academician, so I can talk with from knowing the situation.
Yeah.
Academics tend to really look at their own project, and I think initiatives like ABP Africa, GBB, or the Sanger, they really congregate academicians, but actually nobody's really looking into their own research project. So I think as a whole, science funds in Brazil are really currently distributed under that kind of logic. So I give this money to one research, and she or he will do a great research project, but actually it's still restricted to those researchers on one job. I think that's why you don't have grant caps that are much higher.
Yep.
So I think some changes are also needed in that part of the structure as well.
Yeah, thank you, Alessandra, and thank you, Mark, and also Lukman. And hopefully, Mark, at some point we can talk about. I know we had a workshop earlier this year about where things will go. Hopefully, in the next 2030 by 2050, but we don't have enough time for you. I would have wanted you to talk more about it if our privacy allows to actually be public about it. But maybe we'll invite you at next time. Over to you, Craig, please. Christian, could you please stop sharing? I don't know if the on-site participant can hear me so that I can see them. We'll need to move to the next presentation. Christian Tiambo? Craig, are you guys there? So, Christian, could you please stop sharing so that Craig can take over and introduce you? I can't see them. Okay. Craig, over to you, please.
Oh, we were muted. You could not hear
Okay,
us.
we can. Yeah, I think we're moving to this. Yeah.
Right. Good afternoon, everyone. I have the pleasure of introducing our speakers for a very important topic linked that adds to what we have been discussing this morning. This session will focus on the ethical, legal, social, and policy associated aspects of the very important work that we are doing in AfricaBP. I have the pleasure of introducing Dr. Christian Tiambo this morning. If we can maybe start with Dr. Christian's slides so that I may introduce his bio. Can you please start sharing again, Christian?
Christian, please start sharing your slide.
So while he gets his slides ready, I would like to formally welcome Dr. Christian Tiambo. He's a scientist at the Center for Tropical Livestock Genetics and Health at the International Livestock Research Institute, where he leads the Cellular Resources, Functional Genomic and Biotechnology platform of the CTLGH. His lab activities focus on livestock reproductive technologies and precision breeding, biobanking management, using stem cell technologies. He also serves as officer for the access and benefits sharing of the Nagoga protocol and CBD protocol. So a very apt speaker to add to our program today. Christian, are you ready?
Yeah, Christian, please unmute. You can now unmute yourself. Click the unmute button. And you are also a co-host.
Yes. Yeah. Okay. Yes, please. Can you hear me? Right?
Yeah, go ahead, please.
Okay. So, yes, thank you. Thank you very much. Thank you, everyone. It's a great pleasure for me to be here today and for this session. Just to share with you a bit of what we have been working on integrating local and indigenous livestock genetic resource into the global biodiversity framework. So this is discussions that have been going on also in the different COPs and in collaboration with other national and international partners, just to ensure that when we are talking about biodiversity, all the biodiversity is well captured in the general discussion. So I don't know if my screen is moving.
It's not moving. We can't see the next slide. If you're in the next slide, Christian.
No, it seems to be. Okay, let me stop sharing and then I come in again.
Yeah. Another thing you can do is to convert it to PDF, but that seems to work. Technology, right?
Yeah. Yeah. Just, yeah, sometimes technologies can fail us also.
You can just save it as PDF, and then open this PDF and share.
Can you see my screen?
It's starting up.
I think there is an issue with the...
Yeah, so I think because usually the Microsoft slide, the PPTs can, for some reason, yeah, they work now. It's work now. Yeah, it works now.
Okay, perfect. Yes, so that will be a bit of the structure of my presentation. And as I said, it is a document that we also, that is available, it's publicly available, and what we are discussing, we share also with the CBD. And just to start by giving you a bit of the background on what we are calling local indigenous animal genetic resources for food and agriculture. And also to have in mind that from the CBD, generally when we are talking about biodiversity, we saw a lot of focus on crop. And even when we are talking about animal, people see immediately wildlife. And we need to understand that when we are talking about life in the global south in general, and particularly in Africa, it is both conventional and non-conventional livestock species. So here we are not talking only about the big five: cattle, chicken, pig, poultry, small ruminant, etc. But we also have many other species and breed which exists in the rural settings and which finally is what contributing to feed 85% of our local communities. These are mostly breed which are locally developed or naturally selected. With the expertise of our local farmers, etc. They are well adapted and sometimes with good productivity in their local environment, where they thrive very well, with low input system, less care, less medications, low input in terms of nutrition, etc. And it is not just about what is giving us milk, meat, and eggs. Of course, it is source also of medicine for some of the communities, some of the breed. It is the source of livelihood. So when we are talking about livestock, it's not just about the milk and eggs. It is also a source of income, source of livelihood. These are material or genetic resources which are at the origins of a lot of ecosystem services still supporting livelihood of agroecotourism. It is a huge part of our sociocultural heritage and identity, and even serving as spiritual values, and serving also as a farm-based recreation, sometimes sport and leisure. And some of those local animal genetic resources also are serving as educational value and aesthetics in our local communities. And this is the reason why they are also supporting agroecotourism. So when we are talking about biodiversity in the CBD and in the other fora, we should be able to understand that it's not just about what is in the bush, what is in the wild, but it's also what is around our gardens, around our homestead, etc., and contributing to a lot, to more than food, etc. So it is then very important at the level of the CBD to understand that when discussing about animal genetic resources and biodiversity, of course, we have to consider the environment, we have to consider the farmers, which are the base of it, but on top, it is the great diversity of these locally available genetic resources. And it's also very important to understand that whatever we are developing, whatever we are discussing in the Nagoya Protocol, in the access and benefit sharing, and the other global biodiversity framework, we should be able to have at the center of the discussion our local communities and their engagement. It is their livelihood which is most important at the end. It's not about the publications we are making. It's not about how many meetings we are attending, but it is how we are transforming the livelihood of these local communities. So it means moving from the environment that should be sustainably used and producing ecosystem restoration. Of course, with farmers, as we said, we should be able to conserve this greater diversity. We should also be producing benefit that should be equitably and fairly shared through the multicultural collaboration, multi-sectoral collaboration, multilateral collaboration, etc., to ensure that whatever we are putting in place in terms of policy, in terms of investment, in terms of sustainable use, it is to contribute to the livelihood of these communities. And this is really what is pushing us through what is called now the green recovery. Understand that this diversity of genetic resource is really subject to economic growth, to social benefit, as I mentioned, and to environmental sustainability. And the end goals, as I mentioned, of course, it is this community. As I said, it's not just about publications and very nice talks and participation to all the meetings. At the end, we need to ensure that whatever we are doing is improving livelihood and reducing poverty. Food security and sovereignty is key also for these local communities, and then it's coming biodiversity conservation. So if we don't put these guys at the center of what we are discussing in the global fora, we may not be able to have sustainable conservation, restorations, or benefit sharing, etc. And we need also to understand that it's not just the big five, as I mentioned, chicken, poultry, cattle, and small ruminant, etc., but these local and indigenous animal genetic resources has been at the center of the global livestock development. It means sharing that genetic material from different parts of the world has been contributing to what people are enjoying today in terms of meat, milk, and eggs, etc. And if we are talking about access and benefit sharing, we should acknowledge that contribution from the local and indigenous animal genetic resources. And here we are just taking an example of the black belly goat that originally went from Cameroon long time ago and has really colonized the whole world, going from Africa to Latin America, etc., through the slave trade, etc., and went back to Europe. And everywhere in the world today, you should be able to find the black belly goat, which is well adapted to most agroecological zones, etc. And you can find it in temperate areas, in very cold areas, as well as in hot areas. So this is just to demonstrate how some of these genetic resources are really contributing to global benefits around the world. But how do we make sure that the benefit is coming back to the real owner? And how do we make sure that they are receiving that benefit if this genetic resource is not embedded into the national and the global biodiversity framework? So from there, we need also to understand that the acceleration of genetic exchange will shape the future of livestock breeding. So it is important to understand that, of course, we need to facilitate, and this is the role of the Nagoya Protocol, facilitating sharing of data. There is no benefit if there is no sharing, if there is no research, etc. So ensuring access to a broad genetic base supported by appropriate regulatory framework will be fundamental in enabling us as scientists, as researchers, to develop livestock system which should be tailored for specific breed and specific ecosystem, and make sure that we are benefiting our indigenous people and local communities. So this is just a caption from the FAO exactly to demonstrate how about the second global assessment on animal genetic resources, presenting a bit the benefit coming from animal genetic resources. Of course, here the benefits and the trade. Of course, here you can see camel, chicken, poultry, dogs, cows, etc., and pig. But they are defining animal genetic resources as part of the 38 species and more than 8,000 breed of domesticated birds and mammals. But we should have also in mind that some of the indigenous breed that we are mentioning, which are also called sometimes non-conventional, may not be domesticated, but is what is feeding our local communities, carrying genes that also be supporting the conventional system. And what we want to focus on in these sections is the weak policies and institutions, and the importance to strengthen institutional frameworks and to be able to expand and diversify conservation programs, etc. So this is something that we need to have in mind when we are putting the discussion forward on the global biodiversity framework, understanding the traits from the local and regional animal genetic resources, habitat destruction, crossbreeding, and genetic dilutions, and sometimes importations of breed. As people mentioned, that it was very important to import to solve the problem of low productivity, to crossbreed, etc. But is that really the solution to conservation and to sustainable use of what exists locally? One of the major threats is also the climate change. And we can just see how in the years between 2020 and 2023, East Africa regions lost close to 10 million animals just in the East Africa parts, or in the east part of Africa and the Horn of Africa. If you evaluate this and look at the amount of the number of animals which survive also, you will see that most of the local and indigenous animals really subsisted or resisted to the heat wave, etc. And this is also means they contribute a lot to the livelihood and recovery mechanisms of the local communities. So it is very important to understand that we need to conserve them. They need to be embedded into the biodiversity frameworks, etc. But that will not happen if the policy, market, and market marginalization continue. Many national and international policy prioritize commercial livestock over indigenous breed, leading to unfunding of conservation effort, etc. And when we look very well on how livestock and these indigenous animal genetics are currently addressed in the CBD, in the Common Montreal Global Biodiversity Framework, you can see in Target 4, which explicitly recognize the importance of genetic resource for food security and ecosystem resilience. And this offers a new entry point for us to discuss animal genetic resources beyond wildlife in the biodiversity strategy. But when we come back now to our national biodiversity strategy and action plan in different countries, you will see that livestock and animal genetic resources is poorly captured in the biodiversity strategy and action plans. And even when we consider the access and benefit sharing of the Nagoya Protocol and their applications to the local and indigenous animal genetic resources, it is very limited. Livestock lacks multilateral system similar to what we have in the plant treaty, and which result in fragmented access and governance structure. This is also another aspect that needs really to be looked at from the global south perspective, etc. Those who are keeping at least 1/3 of the global livestock diversity. Another aspect, the financial mechanism. Currently, the global environmental facilities and the green climate funds allocate very small fractions of biodiversity funding to livestock conservation. And it means we are putting aside those 85% of people relying on this local livestock for their livelihood. How can we be talking about fair and equitable sharing of benefit if these people are not considered? And some of the limitations from the conservations of local animal genetic resources are, in fact, that absence of recognition and policy fragmentation. Sometimes even within the country, you will see the office regulating access and benefit sharing may be in another ministry which is not in charge of livestock, and sometimes taking their decision without considering what is really happening in the livestock sector. Mechanisms in plant breeding and development is not the same as in animal genetics, for example, and some decision when taken without considering those genetic resources will be less efficient. Another aspect is absence of the animal treaty, something that we need to look at, and we need to also try to advance this in the global biodiversity framework, and the weak operational synergy among the three conventions for land and the soil and climate and the biodiversity. Lack of comprehension of data, and thank God and others will really be telling you how we think about having a real database where Africans and global South countries also can be sovereign. And if we don't have that, the risk of genetic erosion and biodiversity is still very high. Same as if you don't have sufficient funding to support that, the risk of losing what is very important for a livelihood of our community is very high. So it is very important for us to try to look closely and compare what is happening in the plant treaty or in the plant genetic resource for full agriculture to the animal genetic resources, serving the same purpose: food, agriculture, livelihood of our communities. If you look at the policy aspect, there is no dedicated treaty for animal genetic resources as we have for plant resources, which really affect the governance of livestock and genetic resources in many countries and at the global or multilateral level. If you look at the aspect of benefit sharing, you will understand that livestock genetic resources are not fully covered in the ABS framework, etc., as compared to the plant treaty, the plant genetic resources, where we have the Nagoya Protocol, the plant treaty, with the strong multilateral system providing guidelines on the exchange and benefit sharing in crop. Of course, crop, conventional and non-conventional also, and the major crops are not falling exactly in the same basket, but livestock is really undermined. And the conservation strategy, in situ and ex situ, as I mentioned, the mechanisms are not the same. It's possible to halt at any specific time conservations of the breeding of plant, put them in the freezers and restore and resume the breeding program at any specific time. But there is no way you can apply that same system in animal breeding, which need continuous support. You cannot put an animal in the freezer as you want and bring it again on the to the farm immediately. So it means some of the methods that we are putting in the plant sector may not be working very well in the livestock sector, and that's also pushing us to develop specific national and regional gene banks for breed conservations and funding mechanisms, etc. And research and data coordinations, as I mentioned, also the need for a global database for tracking breed diversity, knowing exactly what we have, conserving them, and be able to bring them back in the breeding mechanisms to support restorations, livelihood, etc. I've already mentioned about all the international agreements like the breeding dynamics, as I mentioned, the comparison between breeding in crop and in livestock, the mobility and adaptations, ex situ conservation, etc., and the diversity of custodian group that are in plant and livestock, etc. Pastoralists, they have total different reality as compared to those who are in intensive production system. And if you don't capture them, some of the important breeds that exist only in the ASAL, arid and semi-arid lands of Africa and other parts of the world may be lost totally. And this is part of their livelihood that should be also lost. So the CBD COP 16 really marked a significant milestone, of course, in making sure that animal genetic resources are captured in the global biodiversity framework, but we still have a lot to do to advance this agenda, particularly the integrations of animal genetic resources in the GSI discussion, the establishment of the Cali funds and the dedicated fund mechanisms for the conservations of local and indigenous animal genetic resources with direct financial support for traditional breed and recognitions of indigenous people and local communities who are the real custodians of this genetic diversity. And these developments signify a progressive shift, of course, from the CBD to include livestock diversity in the food security and ecosystem resilience, but the discussion should go further to understand. And if we want to target the policy and solutions and action plan, we need to understand what needs to happen immediately. It is explicitly recognitions, as I mentioned, of livestock genetic resources in the CBD, in the UNFCCC, and the UNCCD negotiation, integration in the global biodiversity frameworks, and initiate a dedicated funding for livestock conservation. Of course, at the medium and the long term, we need to have also a panel of activities which should be deployed between national system, regional system, and the multilateral system. And at the micro level policy recommendations, if you go through the CBD and the communal biodiversity framework, we may understand that some articles need systematic review. Recognizing pastoralists, as I said, and their system as the biodiversity landscape, and their importance as custodians, as indigenous people keeping the genetic resources, which are very important to improve productivity of the conventional breed and improve sustainability, improve the livelihood of our communities. So Article 8, 9, 10, 15, 16, 20 need revisions in the CBD to include local and indigenous livestock genetic resources. In the communal biodiversity framework, you can also see target 10, 4, 13, 18, 19, etc. But we need also to ensure coordinations between different key institutions, the CBD, FAO, ILRI, etc. And we need to strengthen compliance with national system and international ABS regulation, etc. So this is what really needs our attention to ensure that we address some of the key issues we are preventing and accelerating the loss of biodiversity. And if we really need to have benefit coming back to the custodians of genetic resources, we need to understand that failure to act now and explicitly recognize and protect these genetic resources will lead to irreversible loss of genetics, and the climate resilience that these animal genetics are carrying will be lost or reduced, and it is a lot of missed opportunity for sustainable development. and also a failure to integrate this diversity in the governance. The global governance is not just a technical oversight, but also an issue of equity and justice. And this is why we are in this session right now. So what is at stake is that the livelihood of millions of people and smallholder farmers and pastoralists depend on this. And critical genetic diversity that holds solution for climate change adaptations and disease resistance is in future global food system. And we need also to understand that we need to balance ecosystem in ranchlands and grazing landscape, making sure that indigenous livestock contribute and understand that this indigenous livestock contribute not only to soil fertility, contribute to crop production, and all of us are depending on this crop production, hence depending on livestock diversity, locally adapted, and this is what is also leading to biodiversity conservation. So we need to understand that for the next COP, we need to all contribute to this agenda and include local and indigenous livestock diversity in the international conservation effort for food security, for climate resilience, and for us to face the global challenges that are threatening all of us. To be sure that if there is no conservations for these ones, whatever we are discussing at the global level will not be completed. So, and some of the next steps are here. We can discuss it later at the national level, at the government level, with funders, and also us as research institutions and conservation networks. So that is what I really wanted to share with you. And I hope we can have opportunity to discuss. Thank you very much for inviting us for this general discussion of AfricaBP. So thank God and the others, thank you very much. The floor is back to you. Thank you.
Thank you, Christian. We're going to go back to the on-site participant. I think they have the next presenter, and then just hang around, please, so that we can have a panel discussion afterwards.
Sure.
Over to you, on-site.
Thank you so much, Dr. Christian, for contributing. And we're going to hand over the mic now to Sally Kati, who is also from the International Livestock Research Institute in Kenya. Sally, are you ready with your slides? Can I introduce you? Oh, you're here in person! I'm so delighted to see you. So, do we have her slides queued up? Sort her out quickly. Introduce you quickly. Well, I'm going to introduce Sally Kati, who I saw already while her slides are being prepared. She's an ABS Research Associate at the International Livestock Research Institute, where she works at the intersection of biodiversity, genetic resources governance, and policy engagement. Her work supports inclusive, equitable, and science-driven implementation of the Rio Conventions with a focus on livestock genetics and traditional knowledge. She has contributed to national and regional policy dialogues, supported multi-partner consortiums, and is particularly passionate about advancing African perspectives in global biodiversity discussions. And we welcome you, Sally. Thank you.
Thank you so much for that introduction. Good afternoon, everyone. The colleagues here and online. Now, we've had some rich presentations, and I'm really excited to be coming after Dr. Christian Tiambu, who I happen to have the privilege to work with. So he's done the heavy lifting for me. So I'm going to try and keep this short, sweet, and useful. Now, I'm gonna try and start with a bit of an icebreaker. I don't know if online members can confirm if the slide is moving, so that.
Yeah, so I think we are seeing the slide as presenter view. So if you can let the IT team know, we are seeing it as presenter view rather than as, audience or something. So alternatively, I think we can convert it to PDF. Yeah, I can just convert it to PDF, then share the PDF.
Okay, so I'm gonna let them do that, but since the next slide was pretty much a question, I'm just gonna ask the question to the room and to the audience, and you can probably answer in the chat or in the room by showing and lifting your hands. So the question I have for you today, as a policy person in the room, my job is to poke holes, ask tough questions, and today that's what I try to do. I don't have all the answers, but my hope is that as we discuss, we're gonna get some of those answers. So I have a question for you. After the presentations you've been hearing, how much of Africa's genomic value do we think returns to Africa after, genomes have been sequenced and digitized? How much value do you think actually returns to Africa? By a show of hands, do you think it's more than 10%? Any hands more than 10%? I can see some faces, there's cringing and stuff. In the chat, please, do you think it's more than 10%? Do we think it's less than 10%? Please put your hands up. Some hands up. Some people are not sure. Most of the hands in the room went up here. So the answer is, we really don't even know. I don't know as well, because no one's actually tracking this, right? But what we do know, as was alluded to by Christian's presentation, is there's a Cali Fund that was launched. So in 2024, we went to Colombia and we had this revolutionary discussion at the Convention on Biological Diversity. And what we do know is the Cali Fund was launched to redistribute the benefits from digital sequence information. Now, as I was traveling from Nairobi to Cape Town yesterday, I decided to do some research, just so that the information I'm giving to you, is recent, not old. As of yesterday, what I have seen is that no funds have been contributed into the Cali Fund. 0 contributors, $0. Now, I have worked with scientists for the last 3 years, and I realize this is a numbers game. So anytime I come to talk to scientists, it's not about the law and the policy. I come with numbers so that you guys can actually listen to me and we don't fight. When we think about $0, 0 contributors, I think the silence in the numbers tells us everything we need to know. There's a fundamental gap, right? And that raises the question that we're going to be discussing today. And again, I don't have the answers. This is what I hope we discuss throughout the course of the week. Who owns the DNA sequence and who gains from the profits? The thing is, the reality of the matter is, if we can't even measure how much value returns to Africa, it's a strong signal that none of it does, right? Yeah, glad we're agreeing in the room and people are not looking at me. So, one thing I can say for certain is genomics and artificial intelligence is really rewriting the rules of food, health, and biodiversity systems as we know it. In fact, it's even changing how education and learning systems are. I digress, that is a long conversation I don't want to get into, but it's important for us to keep it in mind. Now, the global genomics market is projected to exceed around $62 billion. This is by 2030. And AI, in an article I was reading that was published by WHO, says that nearly $600 billion stands to be gained from traditional medicine using AI in 2025 alone. Those are some big figures, don't you think? Right. The thing is, what is clear is Africa is really a powerhouse when it comes to thinking genomics and biodiversity, but we're often excluded from the digital value chain. Now, Christian mentioned in his presentation the Montreal Global Biodiversity Framework, and as I promised, I'm not going to get into sections like he did. I'm just going to pick one. Target 13 talked about extending and explicitly including digital sequence information and traditional knowledge in the benefit sharing conversation. When you think about this, this is both an opportunity and a risk. Professor Anne in her presentation alluded to the fact that if we don't do things, people will do them for us. And I pose to you in the room, if we're not shaping the rules of how genomics is governed, they're definitely being shaped for us. I can see people nodding. That means we're doing a good job. So as we think about it critically, some of the things we've been thinking about in ILBI is researching with the end in mind. Now, when we look at the genomic value chain, I see this as a policy and lawyer person looking from the outside in. The value chain starts from farms, it moves to indigenous knowledge, then it goes to the labs where the sequencing happens, and then it goes to the global databases, and then it goes to patented products. For me, that's how I look at it. From the very onset of when the samples are collected from the farmer or the animals, and then it moves to what we find on the shelves, what we eat, what we buy, that's how I look at the value chain. Now, what the reality that we really don't discuss is at every step, there's loss of ownership. It just keeps moving. It keeps moving. For example, as sequencing now is being done within African BioGenome, we're seeing it being done in the continent. But before, it was always done outside the continent. And when we look at it right now, we can see the sequencing is happening here. But sometimes the IP capture, it's abroad, right? It's not for all scenarios, but I just want us to have an open conversation about this. Now, when we look at this, consider the fact that, just as Christian's presentation was saying, 90% of animal and plant genomic data is stored in databases outside, and often with no conditions for use. And I'm happy we've had some top-notch presenters, so I can reference them. I'm going to reference what Dr. Shane said. She's smiling at me. She talked about open as possible and as closed as necessary, which I think is something important that we need to be thinking about as we go into this. Now, some of the things we see is that we have so much data that's out there, right? And then because the context is not looked at, you find that traits in like Nguni cattle and Ankole breeds that are vital for climate resilience, they end up being overlooked. The worst part is we don't even talk amongst ourselves, right? I'll give you an example. So at ILBI, we have a project that's running with Nguni, the red Maasai sheep, actually. So we've had discussions on this project, and it was only until we started talking about doing a genomic reference map for cattle that we had conversations with national stakeholders, and we got to find out, hey, they're also doing work on red Maasai sheep. So there's a duplication, and we keep talking about funding and budgets and issues, but somehow we're still doing the same work because we don't talk, right? Now, if you think about it, the bigger question here becomes, unless we secure this ownership across the chain, we continue supplying raw data for other people while they capture the profits, right? Now, leveraging again on open as possible and as closed as necessary. I am all for open access, right? I believe it fuels science, but without safeguards, it becomes open extraction, right? Because open science doesn't mean free or uncontrolled. And unfortunately, from time immemorial, it's been open exploitation. I've been doing a little research, and this is one of the cases we're going to be discussing when we get into the LZ practical workshop for those of you who are going to be there. I've been looking at the San Julia case, and we all know this as a traditional knowledge kind of situation where we had an appetite suppressant knowledge that was commercialized without consent. It was only until years later that ABSC was negotiated. Ribos generates $600 million annually, right? And it's only recently that the Koi and the Sun have been receiving 1.5% of farm gate prices. I think that's a big question mark if you think about it. If you look at the numbers, again, you see people are nodding because if I came here with facts and cases, you wouldn't listen. But when I talk numbers, people listen. At COP16, it's even crazier. At COP16 in Cali last year, that's when indigenous people and local communities were recognized through establishment of a subsidiary board under the Convention on Biological The CBD has existed since 1992. Imagine in 2024 is when indigenous people and local communities are actually now recognized. Crazy, right? So when we think about the care principles as they remind us of the collective benefit and how indigenous authority must balance their principle, I think it's quite important for us to do this, because if we don't do this, we risk not being politically correct, and at the same time, we end up just sorry, I lost my train of thought. If we don't do this, we don't remain politically correct, and otherwise, open access then becomes another word for digital biopiracy. I really don't like that word, and it's not fairness. I can see some people debating, so it's still good. Now, when we look at the Kali Fund, something I wanted to actually discuss practically was this decision was adopted. It's called Decision 16.2 for those of you who want to keep reading. So it was the first global multilateral mechanism for benefit sharing. Now, explaining what the Kali Fund is supposed to do. So 1% of profits or 0.1% of revenue was expected from sectors like big pharma, biotech, cosmetics, and agribusiness. At the back of your mind, I wanted to bring that no one is exclusively a provider of genetic resources and a user. We are all both providers and users of genetic resources. So it's important for us to keep that in mind. Now, we've already gotten through the fact that as of yesterday, no contributions had been made to this fund, right? So keeping that in mind, this fund was earmarked for indigenous people and women. The contributions were supposed to go first. The first half of it was supposed to go to the women and the youth. This was a landmark decision. If you think about it, as a policy person, we were in the room, we were like, yeah, this is great. But now when you think about it, that no one has actually contributed to this fund, the reality of it becomes a bit jarring, right? When companies that could be contributing to this would be like AstraZeneca, Global Smith & King, they were approached to be front runners in this, but they declined to commit. Why? Industry bodies are giving big pushback against binding rules. This fund is currently voluntary, so people contribute as they please. Now, I want us to think about perspective, again, numbers. Last year, AstraZeneca earned $54 billion in revenue, and 8.7 billion of this was profit. GSK earned $40 billion in revenue and 4.6 billion in profit. If 1% of those profits flowed into the fund, this would mean that we would have around $80 million in the fund. Instead, this fund sits empty, exposing the gap between ambition and accountability. A quote I like to use often is, when ambition happens without accountability, we only have good intentions. So that's all it ever is. So now, as we move on, there's something that's constantly come up in Christian's presentation, which is against fragmentation and harmonization. What I want to echo here as we sit in the room and think about the research that we're doing with the end in mind, two things. Issues like climate change, if we contextualize this, challenges don't come to people siloed. For example, you don't face even in the room, economic shocks, climate change, loss of biodiversity. We don't face that in silos. You don't face biodiversity loss from January to March and then April. It all happens at a go. So the solutions need to be thought about with coherence. Now, so DSI, it's not only a CED issue, it sits across, you have WHO pathogens, you have plant treaty for crops. Christian actually mentioned that there's no animal treaty, which is an issue. Then you have BBNJ for marine genetic resources, which is beyond national jurisdiction. So you have fragmented rules. This brings about confusion, compliance failures, and missed opportunities for science. I'd like to think there's a challenge in this in the sense that people don't, the system, it's designed, it works the way it was designed. So the fact that we have a fragmented system and it's not working in harmony, it's because it was designed fragmented. So if we have a harmonized system or design a harmonized system, then it works. So the system is currently working the way it's supposed to. Now, scholars have urged, harmonized triggers and interoperable systems to prevent loopholes. There's a paper that was published by Amber Hartman Schultz and a group of scientists who we work with at the DSI Scientific Network, and it streamlines how we can ensure, the loopholes and fairness happen. But if you think about it, there's still a need for more. There's a need to break silos between climate, agriculture, nutrition, and genomics is no different. If regimes remain fragmented, we risk our voice being drowned out. And unfortunately, we have to champion that coherence as we're here in the room and talking about these solutions, and that goes for my colleagues online as well. When we think about the power dynamic, ethics, and consent in the digital age, consent has to evolve with technology. It's not about signing forms, prior informed consent. It's not about, going to the field and getting people to sign these forms. It's about whose knowledge and biodiversity is being digitized and reused. For example, again, the H3 Africa initiative pioneered a broad consent in genomic research, but digital innovation demands for us to do more. It demands for us to have Africa-led safeguards. Now, when you look at a $600 billion market being tied to AI is not going away. It's definitely evolving, and we have to have more safeguards. We have to have indigenous data sovereignty. We have to have free prior informed consent as non-negotiable. When we look at digital bioprivacy right now, it really just mirrors patterns of extraction in the old form. People are now not taking samples. Difference is now it's happening through servers and algorithms. People mine open African genomic data, run AI bioinformatics and patents, and we don't get the benefits. The scholars are calling this data colonialism, where the global south supplies data and the global north will capture the benefits. This was from a paper by Koji and Majaris. You can read on it if you want. For me, I want to contextualize it for the people in this room. How I see it happening, it's like we hand over selfies, essays, and assignments to apps, hi ChatGPT, without asking where it all goes. So why should it surprise us when African data takes the same trip, only it doesn't return to us in the form of Instagram likes or papers, it returns as patterns. The South Africa ABS agreements, the Sanhudia, the Kwesan, and the Rooibos really brought recognition, but we have to strive for a further shift in inequities. Otherwise, we would have significant conservation outcomes. So the solution has to be domestic. Why I say it has to be domestic is, if you look at some of the things that are happening in the global arena, we agree in terms of what should happen against biodiversity loss, climate change. We agree in principle. But the implementation of the interventions, it cannot be unison. We have to consider the context, right? And that's why African countries, they have to transpose these decisions into enforceable law. Let me give an example. Where I come from in Kenya, the Maasai community really are about their cattle. Like, it's cultural, and Christian actually showed like the 6 different layers of how we look at cattle or livestock. For us, like cows, it's equivalent to wealth. So to tell like a Maasai to reduce his herd so that he can reduce gas emissions, it's not gonna work. So we have to think about contextualized interventions. Institutions also need to be prepared to absorb non-monetary benefits, such as training tools and knowledge sharing, and we need to talk amongst each other. We have to have coherent systems, for example, that allow us to enable cross-sector data sharing, centralized oversight, and accountability through partnerships. Christian alluded to the fact that we don't have a centralized, federated database in Africa, where we have all this information that we're talking about here. It's something we need to think about. As I wind up, what I would want us to remember in the room is what we map actually matters, and what we neglect, it actually costs us our survival. Globally, you see our sequencing priorities are like on Holstein and Angus cattle, and then crops like maize, and if you look at even big 5 poultry and all those things. Meanwhile, Nguni, Ankole, and Fonio in West Africa, finger millet, sorghum in Kenya, and indigenous vegetables, they remain underutilized, right? What's even worse is we don't even know, like there's what we know, there's what we know we don't know, but then there's also what we don't know that we don't know. So it's something that, we need to think about. And Africa's diversity is important, and the African Orphan Crops Consortium shows us that we sequenced 101 species, but that's really just a fraction of our diversity, if you think about it. And now we're not calling them orphan crops, we're calling them opportunity crops. Don't call them orphan crops. It's important for us to understand that the foods and everything that we ignore today, it really could be what we need for our survival toolkit under climate change. So we have to prioritize mapping these local resources and integrating them into nutrition and climate innovation agendas. Now, what are our aspirations? Our aspiration is clear. As I mentioned, Africa has to be both custodian and beneficiary of its genetic resources. Science has to travel the full chain, from indigenous knowledge and from the labs to the patents to the products in the market, with the benefits flowing back to the community. We really have to ensure that that is actually what's happening on the ground. The other thing is numbers don't lie. If you forget anything I've said today, please remember Africa holds 55% of the world's biodiversity, 20% of its livestock breeds, yet 90% is what's stored in genomic data abroad. The rooibos industry generates $600 million, but the koi and the sun receive 1.5% of this in terms of farm-gate prices. The Kali Fund was projected to raise between $1 to $10 billion a year, yet no one has contributed. Meanwhile, global genomics market is expected to reach $62 billion. So I just want us to keep those things in mind and those figures in mind as we think about what could happen, what should happen, and what we can do. So transformation is really not about agency, it's about writing our own story and not being written into someone else's. And unless we write the rules and we want things to change, we'll remain sequence rich but power poor. And I know that's not the future I want. I would hope that it's also not the future you want. So as I close, what I would like to ask you today, one last time, is who owns the sequence, who owns the algorithm trained on it, and who owns the value? So I put this together as a spark conversation rather than to give you all the answers. And as a policy person in the room, I'll always insist that science has to guide policy. It can't chase it. And since I'm the only thing standing between you and your lunch, I will stop here and let you ponder over those things, over those little insights and framing remarks that I've done for BioInformatics. Thank you so much.
Thank you so much. Wow, what a wonderful, thought-provoking presentation. We really enjoyed your discussion. And I also want to bring back Christian.
Yeah.
We'll have a
So,
panel-based Q&A now.
yeah, do you want to stop sharing your screen, please, so that we can spotlight Kristen? Yes, Kristen is already in the spotlight. Okay.
I just want to have a look. I think this mic is working, right? We want you to talk some more. It's okay, I'll use this one. Okay. I'm already stopped sharing. Right, can you all hear us?
Yes, please.
Okay, fabulous. Right. Let's move to some questions for Christian and Sally. We've got the chat open here. You're welcome to type them, and you're welcome to also. You've got everybody there. Thank you. You're also welcome to raise your hand for some questions, please.
Yeah. So, I think I can go first if no one is going on site and while anyone is perhaps on the type. So I think mine is not just it's not kind of a question, but just a comment and also to provoke the discussion. So just a bit of a background that one of the things AfricaBP does, and is kind of enshrined in how we do things, is on ethics, how we kind of get the data and stuff like that. When we started out putting together AfricaBP 4-5 years ago or 6, the idea, I went around speaking to people, and it was very clear through the engagements that people don't want to be sample collectors. They don't want to be, especially the African scientists. They also don't want to be. They also want to be able to get benefits. With regards to their data, with regards to their sample, and that also influenced how AfricaBP is crafted or engineered in terms of this, and ensuring that all activities we benefit from it. So I do appreciate Christian's and Sally's discussions, and over the past few years we've been working together to see how we can build the awareness needed for these discussions on the African continent, and that includes things like having these discussions in workshops and also other avenues. But one question I think that would be to Christian would be, how do you envisage, because I'm also familiar with this. I'm not a lawyer, but I'm a scientist. I have background in genomics, software engineering, and others, but I also have interest in ethics side of things. And I try, one of the things we're trying to do in AfricaBP is, how do we get scientists to think as a technical specialist, and also as, for instance, an ethics specialist. So, but my question centers around livestock. Given that we already have several, I'm not trying to be biased, but it's just to kind of try to perforate some of those things, and let's have that discussion. Given that we already have several regulations, we have the Nagoya Protocol, we have the International Plant Treaty, we have the CBD upcoming, and also have several at the national level. How do you envisage the livestock treaty, which you just mentioned? How is it going to work with others, including the interoperability? Would that also mean that more countries would then need to, comply with it? Or should we be looking at, say, one big bang policy framework whereby we know this is it? Because one of the problems, especially with compliance, is there are just so many of these. Like each time I'm working with partners through AfricaBP, I try to look at so many of these framework and policies, right? I find it interesting, right? But sometimes, people want to say, okay, let's do this. So how do you see that working in terms of this campaign message, and whether also you've considered to have it like a policy brief? Because I think what you raised is an important point, that we have plant treaty, but we don't have livestock. I know, and I'm just hearing it for the first time. So it would be nice for you to expatiate more on that.
Yeah, thank you very much, Dangot. And it's also a very great pleasure to hear from Sally, the wonderful presentation. Yes, indeed, I think some discussion happened long before, but I think we just silence also at a certain level, because people were seeing plants different from animals, and somehow thinking the ownership was not exactly the same. But in fact, if the discussion with both type of commodity was pushed at the same time, we have not been in the situation where we are now. But what we need to do now is first for us and AfricaBP to continue sensitizing scientists and livestock scientists, for example, to understand exactly what it takes, and understand that whatever we are doing in terms of genomics, in terms of access, is in fact to develop products which are serving communities, and the benefit has to go back. And for us to have access, of course, we need to comply. So from scientists' perspective, we first need to understand exactly that pipeline, etc., not just from product development to the policy, from the sampling to the product development, etc., but no feedback mechanism. We need also to build the feedback mechanisms for our local community, custodian of genetic resources, to understand exactly what they are losing if they don't give access to the genetic material, etc. And now, of course, we have already a lot of regulations going on, etc., but we just need some time to write on what already exists. We may also write some articles in plant treaty may be working very well for animal treaty, for example, if we succeed develop that. But not all of them will be working at the same level, etc. Or sometimes need a little bit twist, because as I mentioned, if you are conserving plants, you can take seed today and put in the freezer and revive it in the next, I don't know, 10 years, and it will continue the work, continue germinating. But you never take an animals and put in the freezer and the next day it continue. So we need to understand that system, etc., and understand that we have some regulation, we have some background already, some legal infrastructure already exists, but we just need the willingness of our national partners, of scientists, of the multilateral system, etc., to embark on this. It will not be something that will happen overnight, but we really need to start discussing it and understand that even the plant treaty was not developed in a single day, the Nagoya Protocol was not developed in a single day, but we need to start the journey if you really want benefit to also return to the custodians of our genetic resources.
Yeah,
So,
thank you, Christian. So you also said something that I really, which is, I like how you said it, that there is no benefit without sharing. So I think it's something we really need, and which is why sometimes when I listen to the campaign messages for for some of those things in terms of let's get open science, I feel like we could potentially have a different message. And what you just said again is one of those things whereby there is no benefit without sharing. So if you want to get, and there's also no sharing without benefit. So those things have to go hand in hand. So thank you so much, Christian. I have one question for Sally on site, which is, as she was talking, okay.
Can we quickly give a chance to Tobias?
Okay, all right. Okay, Tobias. Tobias, could you please ask your question?
Tobias, you can unmute and ask your question.
Tobias, please unmute.
Hello.
Yeah,
Nice
go
to be
ahead, please.
here.
Thank you. Yes.
Yeah, thank you. I have been following. Yes, I have been following the discussion. With respect to, first and foremost, let me thank the presenters so far for their wonderful presentations. And the current discussion, particularly as raised by Adia Tangot and the last person that, is it Tambo or so? My contribution to this is this. African BP has done a lot in terms of raising awareness and then galvanizing all of us to think in the direction of protecting African resources, and particularly as it regards to making sure that all resources from African soil are conserved in terms of giving the sequencing and retaining our data within the African space. And that is why I think in terms of sharing and getting benefits, is it not time for what has been done by Tangot and his group to be done in terms of gathering some few experts who will look at all these international protocols and find a way of domesticating it as it concerns Africa and its resources? Then with that kind of idea, with kind of that what has been gathered in terms of looking at all the protocols, find a way to bring aspects that concern African resources, now write their own version, and now have a bargaining power to go to the international space and say, this is what we think the way we should go for returning, giving back from Africa and also returning benefits to Africa. In other words, what I'm saying is that let there be a think tank of experts that will look at all this in terms of ethics and all the protocols or international regulatory systems that will now project the African angle to the international regulators, so that there will be a balance that recognizes the input from African space. I think until you have a bargain, you have something to give, something to protect, something to say, this is my bargaining power, this is what I give if I must give my resources. then the rest of the world may not take us serious. I may not have been able to put it clearly, but then if somebody understands me very well, I want my own angle to it is that we should look inwards. There must be some scientists who also have some legal backgrounds, who will also put legal touches to the technical angle and make it a projection to the international regulators so that we have a bargaining power from the African space in terms of some of these regulatory systems. We should not guarantee a return to the African environment or to the African scientists or to the African system benefits from the resources that are taken away from the African soil. I don't know if I have communicated this improperly.
Yeah, thank you, Tobias.
Yeah, and I also want to give an opportunity. I see Sally nodding to when you spoke about a think tank and things. And maybe, Sally, did you maybe want to contribute to what Tobias was saying? Just being mindful of our time and the fact that we're waiting for lunch.
Can you hear me? Yeah. Okay. I think Tobias had some really great thoughts and insights, and I think these are.
I think you guys are mute.
Sorry, it's not working to do that. Sorry, can you start again? I'm sorry.
Can you hear me online?
Yes, please carry on.
Yes.
Okay. So as I was saying, what Tobias is saying is very important, and it's part of conversations we've had with Tango and Christian in different platforms. There really is a need for Africans, yes, to streamline and understand, the contributions that we can make on this global arena based on our unique concept and on our unique context and our unique, standing as Africans. But something we also need to consider, Tobias, is perhaps AfricaBP. Thank God, I don't know if we can coordinate and maybe be approaching or discussing with, African group of negotiators before they go for these kind of, conference of parties. It would be something helpful to be doing. The second thing is also in terms of these think tanks. Why I said in my presentation that research has to happen with the end in mind is so that we're able to influence these policies when they're being created. Because if we are not doing that, this is a hare and a tortoise, assuming the science is the hare or the tortoise is the policy. It can never catch up. It has to be focused, slow, and it has to be contributed from the science aspect. Because imagine this: if you have a renowned innovation or a renowned, contribution to something that will advance our global agendas towards nutrition or climate, be it whatever it is, or conservation of genetics, right? If you're not involved in discussing with governments from the onset, then you come later and have this discussion. It takes like 5 years before you get policymakers on the table again, and then another 5 years for implementation. So I don't know. Thank God, I think there's a loop there, but it needs to be from the onset and at the beginning that research is influencing policy. Thanks.
Yeah, I do. Can I comment?
Very quickly, thank God.
Okay, very quickly. So, yeah, how many minutes do you have, just so that I can be within time?
We're already 20 minutes over time.
Okay, maybe I can just leave that to Kristen. And yeah, Kristen, you want to say something?
Yeah, just to say to thanks. Are you getting me?
Yeah. Go ahead, please.
Yeah. Just to thank Tobias for that questions, and to also remind him that that's exactly the reason why we are requesting country to review again their national biodiversity strategy and action plan and make sure that livestock is included. And now, for that example, for the case of livestock, also with Sally and others, we have been working on what we are calling now the cattle consortium agreement, where we had a project that was working on 14 African countries regarding the genomics of African cattle, etc. And initially with ILRI, we're having bilateral agreement, but now we're having a multilateral agreement between the 14 countries, etc., to discuss exactly and try to align the multilateral system, try to align the CBD regulations with our national context, etc. And this is also the beginning of what we are calling here the think tank. Sally has been thinking about that for very long, just the support to go for it, etc. And the best entry point, as she said, is to work with the Africa group of negotiators to really position this plan, review the article, and see where each proposition is fitting. And at the end, we should be able to have a bargaining power and something to table at the CBD, maybe during the next COP, and say this is what African want if you want to move forward in the multilateral system and the TSI stuff. Thank you.
Yeah. So to that,
that's
just to
great.
mention that on the AfricaBP side, I won't go into much details. We're already working on this, and in the next few months, you begin to see some rollouts on it. So I wouldn't go into much detail. So, but whatever thing you just, all the things you mentioned now, we're already thinking about it, and Sally also is involved and others. So yeah, just know that
It's good
we're
to hear
yes.
this.
All right. Over to you.
Thank you.
On site here.
We have one question. If you quickly want to ask your question, and then that would be our last question for this session.
Can you guys hear me?
Yeah.
My question is for Ms. Sally. Thank you for such a beautiful presentation. It was really captivating. You mentioned, I think just paying off an attention for a little bit, you mentioned the Maasai tribe, and I wanted to ask that as someone who is from Kenya and may have been possibly exposed to those people, how do you, number one, go about dealing with farmers who may not be willing to put sustainable measures in place in order to reduce gas emissions? And you also mentioned that there should be contextualized interventions, and I wanted to ask, how would that look like for farmers who have such a large number of
cattle? All right. I don't know again if you can hear me online, but I will throw the technical part of that question to Christian. I'll answer from a policy perspective. So based on the work that we've been doing at AV, one of the things, when you look at even the figures on gas emissions, Africa is not really a huge contributor to those gas emissions. Now, I will use a different example, not the Maasai community, because in as much as we've been exposed to them, I can't comment on what I've not directly worked on. But there's a project I'm involved in where it's called the More Milk Project. So this is a project we're doing in Philby where you get to see real examples of how an intervention can help a community. So this project focuses on milk quality and milk safety for nutrition, right? What we do in this project is we're collaborating with the Kenya Dairy Board, which is a regulator, right? In this context, we end up going to the field to discuss with the community and find out from milk vendors in this business, why are you not complying to regulation? Because this is set up to formalize the informal sector, right? So when we go to the community, you find instances where a milk vendor will tell you, I'm not complying because maybe the licenses are more expensive than my turnover in a month. So you can understand, or someone will tell you, I need a fridge to store the milk, or the many pipelines or the instruments that are required to run this business. And they say, you don't start the business with everything. For example, in this room, if we decided we were gonna start selling pens, we're not gonna wait until we have like the bulk manufacturer and everything to have an intervention. You start in bite-sized pieces. So in terms of after understanding what the community needs are, we're trying to now co-author, right? Solutions and interventions that will help that community directly and push them into compliance. So what does that look like? It looks like going for sessions and asking the people involved, if you were the regulator, how would you deal with things? So basically what I'm trying to say is these intervention measures require conversations with community. And at the same time, it also requires technical people, i.e. in this room, to discuss local realities and try to channel that into solutions. And I believe by and large, that's what AfricaBP is trying to do.
Thank you. Thank you so much, Sally, for that contribution and for that question. I am mindful of our time today, and I am so energized by this discussion. I think we are going to return to these themes in so many discussions in AfricaBP. Again, like thank God made the commitment that we are already thinking about these issues and how we are going to incorporate them into core AfricaBP activities. And we hear your calls for a think tank. And I think as a community, that's definitely something that we will explore. I think you will all join me to thank our incredible speakers for this session. And we look forward to further engagements on these topics. Thank you very much, everyone. Breaking for lunch now. As I have it on my program, we will be back at 2:10. Am I correct?
Yes.
Thank you much, everyone.
We're really interested in using high-dimensional phenotyping or phenomics to uncover the mechanisms of disease biology. And particularly, we're really interested in the biological mechanisms of genetic variants and mutations that are associated with both neuropsychiatric and neurodegenerative disorders. And we've been really interested in working with Element on their new technology because morphology is only one measurement. We have recently published this work on what we call neural painting, which is simply taking cell painting and then applying this to neural cell types. It's really easy in the lab, but the processing and analysis can really take a long time, and that feature interpretation that you get from that always requires follow-up experiments. So you have to then go measure RNA, you have to measure protein, and then try to understand which ones of those give rise to the morphological changes. And so The AVD24 platform allows us to capture all three of these simultaneously. And so we set up a pilot experiment with the AVD24 team where we took iPSCs from our lab, we differentiated these into both astrocytes and excitatory neurons. We grew these cells on the Teton flow cells. We would fix them, and then we sent them to Element for processing, part of the technology access program. Firstly, we're able to take the single cells and cluster them using the RNA expression transcripts with the AVD Neuro panel. But here, we actually get better accuracy, and we get the added information of kind of the RNA expression. So here again, we're just taking these clusters that we found using the gene expression information, but then layering on here values for the morphology features that seem to discriminate our cells. But one could easily imagine now going into each of these clusters and doing different kind of regression analyses to sort out whether there are specific proteins or transcripts that would associate with these morphological characteristics so that we can begin to draw linear relationships between protein and RNA activity and how cells change in response to certain contexts. You know, doing 1 million cells in a run is feasible, right? And having the data to come back after simply overnight, effectively, is really incredible. Sometimes I think it's hard to kind of grasp your head around rather than waiting weeks or months to get information on 1 million cells. You can kind of have it right away. The actual experimental part of it is incredibly easy. You just need to grow cells and fix them. In a system like this, where we can use many different dimensions to measure, at least in vitro, when things begin to change, that gives us a better sense of maybe what's causing disease rather than what is caused by disease. And so doing everything simultaneously, I think, could be really powerful.
Welcome to the 20-year anniversary video of AfricaBP, Africa's genomics company, here from our headquarters in Makelnek, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of AfricaBP emerged not far from here in Pretoria in the year 2000 while I was doing research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so. The startup was only possible with the substantial direct foreign investments, mainly from shareholders from Germany and Switzerland, as well as the startup loan from the State Secretariat for Economic Affairs of Switzerland. So what was actually our idea in 2000 to start a biotechnology company in South Africa? The aim was to locally produce oligonucleotides which need to be custom made, and therefore by local production the turnaround time dropped from 2 to 3 weeks for imported oligos at that time to 1 or 2 days. This of course allowed to accelerate research output for our customers that apply PCR amplification and DNA sequencing in their research. And more recently, it allowed uninterrupted essential supply during the pandemic. In addition, we aim to offer Sanger DNA sequencing services. The aim was here that outsourcing DNA samples for sequencing will allow our customers to save the investments in capital-intensive sequencing instruments. 20 years later, we still do all these services, but of course the technologies in life sciences have significantly evolved in the past 20 years, and so has AfricaBP. A big thank you to all our customers, our dedicated current and former employees and commercial partners over all these years, and of course our investors that took the risk and made this possible in 2002. Please enjoy this video about the different aspects of AfricaBP. Let us know your comments, suggestions or questions about AfricaBP in the comment section below. We strive to even be better in the next 20 years.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation. MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DNB-seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of few companies in the world that have the ability to develop and mass-produce low, medium, and high-throughput clinical gene sequencers from GB to TB. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, BIT products, and low-temperature storage products, with a strong focus on the upstream of the industry. MGI operates in more than 100 countries and regions, serving more than 2,800 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
QIAGEN. Sample to insight.
Introducing the newest member of the QuantStudio family, the Applied Biosystems QuantStudio Absolute Q digital PCR system from Thermo Fisher Scientific. This fast and easy-to-use digital PCR solution offers high-quality data with minimal hands-on time. Sample prep is identical to qPCR. Simply load your reaction into the plate and insert the plate into the instrument. All components of digital PCR, including reagent compartmentalization, thermal cycling, and data collection are performed on the single standalone instrument. After loading your plate, start your run and return in 90 minutes for sensitive, accurate, and precise quantification of your targets. The quantitation accuracy for any digital PCR system is driven by the amount of sample that is analyzed in a reaction, and while some digital PCR platforms can waste up to 75% of your sample, the QuantStudio Absolute Q system consistently analyzes over 95% so that you can see the whole picture every time. At the heart of the QuantStudio Absolute Q digital PCR system is the novel microfluidic array plate technology. This technology was developed for maximum consistency and accuracy by minimizing waste and maximizing reagent distribution into microchambers. And all you need is one plate and one instrument. The MAP16 digital PCR plate provides reliable consistency, high accuracy, and an easy workflow. After loading your sample into the plate, the instrument performs automated reagent distribution into fixed microchambers. Each reaction remains independent during thermal cycling. 4 optical channels allows you to analyze more targets per sample. As the only digital PCR solution with the capabilities for pre- and post-PCR background subtraction, the QuantStudio Absolute Q system employs sophisticated rejection of non-PCR artifacts, offering high confidence in true positives for every microreaction. The MAP plate is designed to run up to 16 samples in parallel, with the flexibility to run as few as 4 at a time to match experimental needs without wasting partial plates. The intuitive QuantStudio Absolute Q analysis software enables users to easily change and define experiment-specific parameters, name and track experiment details, and monitor the run from an easy-to-understand user interface. After the run is complete, return to view the results and apply powerful analysis software. QuantStudio Absolute Q Digital PCR System from Thermo Fisher Scientific. Experience the future of digital PCR at thermofisher.com/absoluteq.
entries and see how far they've come. It's fully functional, and I didn't write a single line of code. You start by describing your idea.
Good. Can I just get confirmation that everyone online can hear me? Are we audible online?
Yeah, we can hear you.
Okay, perfect. Thank you so much. So welcome to this afternoon session, everyone. I hope you enjoyed your lunch. I'm gonna introduce our first speaker for this afternoon session while he shares his slides along. So our first speaker is from Dr. Rian Pierneef. He is a South African bioinformatician and a researcher with a passion for advancing genomics in support of health, biodiversity, and sustainability. His work spans microbial genomics, metagenomics, and data-driven approaches to understand complex biological systems. Dr. Pierneef is also particularly committed to building bioinformatics capacity in Southern Africa and to foster collaborations that bridge science, conservation, and public health. He's currently involved in initiatives that apply computational biology to real-world challenges in the One Health space. And he's also aiming to empower local researchers through training and technology. And so this afternoon, he's going to tell us a bit more about what he does and how he plans on achieving this with the bioinformatics and driven approach. So Dr. Pierneef, the floor is yours, and you're welcome to share your slides and start your presentation.
Great, perfect. Okay, if you could just confirm slides are visible.
Yes, please go ahead. We can see you slides.
Yeah, we can also see you in the venue.
Okay, fantastic. Thank you very much. So my name is Rian Pierneef, and thank you for giving me the opportunity to have a talk. So our health and the health of our environment and the richness of biodiversity are all connected in ways we are only beginning to understand. So today we follow the footprints of life from ecosystems to genomes on a digital safari through bioinformatics. So when we think of a safari in South Africa, we imagine lions on the hunt and elephants moving across vast landscapes and endless savannas stretching into the horizon. But today, I'd like to invite you on a very different kind of safari. Instead of following animal tracks in the sand, we'll be following digital footprints hidden in DNA. Now, this safari doesn't take place in the bush, but inside computers, laboratories, and data sets. Yet it tells us just as much, if not more, about the life around us. So it's a journey into the digital landscape of biodiversity, where every gene, every sequence, and every genome offers a clue to understanding, protecting, and celebrating the living world of South Africa. Now, South Africa is one of the world's greatest biodiversity hotspots, a region where life thrives in extraordinary abundance and variety. So think of the Cape Floristic region, a relatively small stretch of land that contains more unique plant species than almost anywhere else on Earth. Or the vast savannas, which provide a home for some of the largest animals, elephants, rhinos, giraffes, all species that inspire awe around the world. But biodiversity isn't just about what we can see. Beneath the surface, in our soils and our waters, and even within our own bodies, lies this astonishing hidden richness of life. Microbes, fungi, insects, and plants interact in ways that sustain entire ecosystems. So when we talk about biodiversity in Southern Africa, we're not just describing beautiful landscapes and iconic animals. We're talking about a living, breathing system of connections, both visible and invisible, that supports our health, our future, and our food. So what does biodiversity, or why does biodiversity matter? It matters because it underpins absolutely everything we depend on. It regulates our ecosystems from clean air and fresh water to fertile soils. It secures our food through crops, pollinators, and healthy livestock. And it shapes our life and our health by providing new medicines, by buffering against diseases, and by keeping this environment in balance. Without biodiversity, we lose resilience. Crops become more vulnerable to pests and climate change, and our ability to discover new medicines is reduced. Even the stability of our economies and society is shaken. So biodiversity is not a luxury, and it's not just there to attract tourists. It's our life support system, a critical foundation for survival. So protecting biodiversity means protecting ourselves. Now, this is where bioinformatics comes in. Bioinformatics is the science of turning biological data into knowledge. Every living thing carries a code, DNA. And modern technologies now allow us to read that code at an astonishing scale. But reading is only the beginning. So to make sense of billions of DNA letters, we need powerful computers and algorithms and databases. So in simple terms, bioinformatics is the digital equivalent of fieldwork. But instead of using binoculars or tracking guides to study animals in the field, we use computational tools to track genes, proteins, and entire ecosystems in data form. It allows us to zoom in on the tiniest details of life, while also zooming out to see the bigger patterns, connections between species, between ecosystems, and even between the past and the future of our planet. So in essence, bioinformatics is the science of building puzzles. After extensive fieldwork and careful laboratory preparation, we don't get a full genome handed to us neatly. Instead, we receive fragments, millions of small pieces of DNA sequenced, stored in a digital form on large servers. Now, our task is to reconstruct these fragments, put the pieces back together until a complete genome emerges. It's very much like solving a massive complex puzzle, except the pieces number in the millions and the picture on the box is often unclear. Once the genome is assembled, we can then place it onto the tree of life. Now, this tells us where an organism belongs, but it also tells us the evolutionary paths that it has taken. This winding safari on dirt roads that life has walked to arrive where it is today. Sorry. Where it is today. So on a traditional safari, rangers read the landscape. They follow footprints, they notice broken branches, and watch for patterns that reveal the presence of wildlife. On a digital safari, we do the same, but in a world of data. So instead of animal tracks in the dust, we follow genetic footprints across massive data sets. We search for hidden species, uncover relationships, and track biodiversity at a scale which was never before possible. In both cases, the goal is the same. We want to understand, to protect, and to ensure that life's incredible diversity endures. So let's begin the safari in the invisible world, or the world of microbes. When we sequence DNA from just a handful of soil or water, we uncover thousands of microbial species, many of which cannot be seen under a microscope. Now, these tiny organisms influence agriculture, shape water quality, and build resilience against climate change. Without bioinformatics, this hidden diversity would remain invisible to us. So one striking example lies in the Barberton Greenstone Belt, a site which is famous to geologists for some of the oldest rocks on Earth. But it's also a biological treasure. The microbial communities here may still be undiscovered, and they hold secrets about the origins of life and clues for surviving extreme environments, and perhaps even the potential for new medicines. So let's move on to some wildlife conservation. Now, genetics allows us to see stories of the survival that are otherwise invisible. So if we look at the African wild dog, one of our most endangered carnivores, by analyzing their DNA across southern Africa, researchers discovered something remarkable. Wild dogs are still able to disperse over vast distances. Now, this genetic flow shows signs of resilience, a species that, despite threats, can still connect its population across landscapes. Now, this knowledge helps conservationists design wildlife corridors and strengthen protection strategies. In contrast, we have the Cape vulture, where genetic studies have revealed that these birds have a very low genetic diversity and a shallow population structure. That means they are more vulnerable, less adaptable, and at greater risk from environmental change and human pressure. So together, these two examples show us how bioinformatics gives conservation a hidden map, identifying which species are holding on and which are in the most urgent need of help. So indigenous crops further hide a world of resilience and diversity that often goes unnoticed. By decoding their genomes, we uncover traits like drought tolerance, disease resistance, and exceptional nutritional value. Now, this knowledge allows us to develop more sustainable agriculture, support local communities, and adapt to changing climates. Without bioinformatics, these vital signs and the crop's hidden potential would remain invisible. So what does this all add up to? So bioinformatics bridges conservation, agriculture, and human health, revealing how biodiversity underpins the One Health concept, the deep connection between people, animals, and the environment. By uncovering these links, we open doors to innovation, from new biotechnologies and sustainable farming practices to ecotourism that benefits communities and ecosystems alike. So in short, seeing the unseen helps us protect life in all its forms. But there's one more critical element, and that is people. So to fully explore our digital biodiversity, we need trained scientists, local experts, and robust computational resources right here in South Africa. Investing in bioinformatics capacity is not just about advancing science. It's about empowering our region to lead in understanding, conserving, and sustainably using its own biodiversity. By building local expertise, we ensure that the discoveries of today become the solutions of tomorrow. So our safari has taken us beyond the savannah, deep into the code of life itself. Just as rangers protect wildlife on the ground, bioinformaticians safeguard biodiversity in the digital world. The footprints are there, waiting to be discovered, understood, and preserved. Our biodiversity is written in DNA, and bioinformatics is the key to reading it, learning from it, and protecting it for generations to come. So thank you, and the digital safari continues.
Thank you very much, Dr. Birnia. I'd ask you to please stay online till the end of the session because we'll have questions after the last speaker. So the next speaker is Ina Solas, And while she comes forward and gets her talk up and running, I'd like to introduce Dr. Anse van Coller. She's a senior scientist and bioinformatician at the South African Medical Research Council. And along with having a PhD in medical microbiology and immunology from Stellenbosch University, she also has extensive experience in next-generation sequencing data analysis, specifically genomic assemblies and developing bioinformatics pipelines. Her research spans across various fields of molecular biology, including virology, microbiology, and immunology. She's also led and optimized several NGS workflows for human, animal, plant, and pathogen genomics, leveraging high-performance computing for large-scale data analysis. And today she's going to be talking a bit more about the assembly of a very complex indigenous plant species.
Thank you for that introduction, Tracy. Just to confirm, I know you can see the slides online.
We can't see your slide, Ansia. So we're just seeing the file browser. So my suggestion is to convert the slide to PDF. Just save it to PDF and then present the PDF.
How about now?
Yeah, it works now. Yeah.
Yes, so okay, so yeah, thank you for the introduction, Tracy. I'm very excited to share with you guys today some of the work that we've been doing on helichrysum biodiversity in collaboration with AfricaBP. So just for a little bit of background on this plant, so the genus is named after the Greek words for sun and gold. That's because this genus has these characteristic golden yellow flowers, and also the word odoratissimum refers to the strong odor of the plant, which is why it is also commonly known as African incense. It also goes by poichut or impepel in some of the local languages. This is a very highly abundant plant, and you'll find it all throughout southern Africa, and it is a very popular traditional remedy. It grows all over in the highlands, a lot of southern African provinces and other countries as well. In fact, I also spotted quite a large grouping of them actually in N7 when I was driving in today. So it is of great cultural significance in South Africa. Most parts of the plant are often burned to help remove evil spirits, or can be inhaled to try and introduce good luck. It has a lot of documented medicinal uses, including treatment for heart disease, chest pain, but it can also have quite a calming effect. It can be used for treatment of cough, fever, and UTI, and so studies have shown that it likely possesses some antimicrobial properties. It's been used to treat psychosis, which could be due to the presence of phytocannabinoids, which has been found in other helichrysum plants. And because of the strong odor, it's also been used as a repellent and a perfume. So despite lots of studies into the helichrysum genus, no fully assembled genome exists for any of the helichrysum species. Lots of studies have found extensive variation in the genome size, the chromosome number, at the ploidy level within the genus. And it's expected that polyploidy is a very important evolutionary driver for the diversification of the genus, leading to a more broad ecological adaptability. So, for instance, there was a study that saw that plants growing on the side of a mountain were all diploid, whereas ones that grew further away, perhaps more dry area, was more likely to be hexaploid. But given that no assembled genome exists for any of these species, we then sought to sequence and assemble the genome of this plant using Nanopore Technologies and high throughput chromosome confirmation to capture high sequencing. So this might be a bit redundant. I'm sure a lot of you already know what is a de novo genome assembly, but just for the benefit of perhaps those that are not as familiar with these terms. So if an organism doesn't have a reference genome, it needs to be constructed in a de novo manner. So to use the metaphor that the previous speaker used, we're building a puzzle without really knowing what the picture is going to look like. And this process relies on alignment tools that align all the different reads to each other to identify overlaps and kind of build these into contiguous sequences known as contigs, which can then also be linked together in a process known as scaffolding to, at the end, generate a consensus sequence that is the reference of this genome. So this is a very computationally intensive process, and it can take quite a while. This computational intensiveness increases as well with the repetitive nature of the large genome size of some plants and add additional burdens on this part of the process. But in general, long reads such as PacBio, or ONT reads are very useful for de novo assembly because it can be easier to resolve some of those complex regions such as repetitions and structural variations. So we sampled the plant at Babylon Surin in the Western Cape, and then some of my colleagues were able to extract some really high quality nucleic acid, and then we did ONT sequencing on our P2 solo, thanks to the Atomics Monkey Exchange for funding this part. And then we did high-C and RNA sequencing on our MGI-Tech as well. So this is a very broad overview of the assembly workflow that we followed. Just highlighted the different data types that we used. So for the ONT data, we did the super accurate base calling to kind of just improve the quality of the base call reads. And we then did basic QC adaptive driven and filtered out all the low quality reads. So we only kept reads that had a Q score of 20 or above. And we also did some employee and genome size estimates using various different tools. We then integrated the high-C data, which we also QC'd and trimmed before doing a hybrid de novo assembly using HiFiism, where we incorporated the ONT and the high-C data to generate a draft genome. We then used the high-C data to perform scaffolding, to pull all of the context together. And throughout this process, we did a lot of QC to look at the metrics of the assembly. We then did manual curation to kind of paint the scaffolds into chromosomes, and then followed by annotation, which incorporated the RNA data. So for the ploidy investigations, so as I mentioned briefly earlier, so previous ploidy estimations have indicated that they are both diploid and hexaploid helicrystal odoritism individuals. And so like we got a hexaploid one. As you can see, the 6 kmer peak. So this does significantly affect the assembly. It brings the complexity up part of it. And we also saw that there's high level of heterozygosity within this plant, and with a really large estimated genome size potential over 3 gigabases. So I plugged it all into HiFiism, and it has the ability to phase into 6 different haplotypes, because we specified that this was a hexaploid individual. But for the purposes of this study, we'll just focus on the primary assembly, which is essentially just a collapsed version of all the haplotypes. So it removes some of the natural genetic variation, but it's a bit easier to get a more complete picture of the genome. So this is a pretty good quality assembly. It was still quite fragmented. So if you look at the number of contexts, it was about over 5,000. But it had a really good N50, which Shane described earlier. This is 50% of the genome is made up of contexts that are 12 kb or higher, so it's pretty decent assembly for a first draft. I also like to always look at the L90, which is showing that 90% of the genome is made up by 220 contigs. So there's a lot of large contigs and then a bunch of smaller ones. So this is because I didn't trim all shorter reads, so it is recommended in high pricing to use only reads that are 10 kb and above, but I include the smaller ones as well. So these do not integrate as nicely into the genome. Because there's this large number of really small small contigs, I tried to simplify the process by removing the ones that mapped to known mitochondrial or chloroplast sequencing, because I wanted to focus on building a nuclear genome. And then after removing the contigs that were 50% or more matching to the organelle sequences, we ended up with much less contigs, but without affecting the completeness of the genome. We had a BuzzCo score of about 98.4, which is very decent. So to integrate the Hi-C data, now I know a lot of people are not familiar with the Hi-C technique, so I thought it would be useful to insert this little slide here. So in a cell, the DNA is kind of coiled in on itself, and so we can exploit this by performing cross-linking, where we attach regions of the DNA that are in close spatial proximity to each other, and then when we extract the DNA from that, you end up with DNA fragments that contain bits that are not necessarily linear, but within close spatial proximity. When you sequence this library, you can get this Hi-C contact map, which shows which parts of the genome interacts with the pieces, and this is very useful for scaffolding. So we got pretty good quality Hi-C data with really good resolution, so it was able to pick up lots of contacts within close proximity and ones that go further spread. And I used this Hi-C for scaffolding, and you can see we didn't manage to reduce the number of contigs that much, but we definitely increased the N50 to make a very contiguous assembly. You can see here that all 90 went to 24. So the next big part was manual curation. So you heard Vanguard and Prof. Mark talk about this earlier this morning. So essentially, when you create this Hi-C contact map, it looks a bit messy at first sometimes, because there are sometimes errors that are introduced by the assembly algorithms. And so you can see, for instance, here where there are interactions off to the side, which means that this part of the genome probably belongs over here. So this is quite an intense manual process of breaking up scaffolds and moving them around so that they are in the chromosomal order. This is a very lengthy process, especially for a complex plant that we have here. So here is a view of the Hi-C contact maps of the draft assembly. You can see there's quite a bit of pieces off to the side. And even if you zoom into one of these scaffolds, you can see that it's quite messy on the inside as well. I have not had the fortune of any one of the genome curation workshops, so I had to phone a friend. So I did work closely with Sadiq, and he was able to assist with the manual curation of this genome into a 7 chromosome, which is typical for the species. And we have 7 large chromosomes along with about 3,000 or so smaller unplaced scaffolds. Yeah, this was quite a lengthy process due to the complexity of this plant, and I'm very grateful for Sadiq's help on this. So we did some annotation as well. So the purpose of what we were trying to do was not to identify which genes are associated with which biological compound, but rather just to describe what we're seeing in the gene in the genome in terms of gene content. And we were able to identify that there was around 340,000 transcripts, and there was a lot of repetitive elements in the plant genome. Most of these were transposable elements. So this is something that often happens in plants, especially polyploid plants, where you see there was likely some kind of whole genome duplication event in the past, and the plant has just held on to all these extra copies of genes spread across all the different chromosomes. And of course, this data can now be used for further bioprospecting to see if we can find what genes might be economically suited. So just to conclude, we were able to construct a high quality chromosome level genome for galactosin, and this would be the first chromosome level assembly done in Africa, where all of the work was performed in Africa. There have been other African-led initiatives for chromosome level assemblies, but most of these, all of these had some part of the sequencing done in another continent. So that's a great genomic resource which will contribute to future conservation, sustainability, and of course active compound discovery, and also be useful for breeding and the commercial development of this plant. I'd like to just acknowledge everyone who formed part of the team, and thank you all for your attention.
Okay, thank you so much, Dr. Ansia, for that wonderful presentation. Our last and final presenter for this session is Professor Clint Roeder. I'm not sure, Professor Clint, are you online?
Clint is not online. I can't see him online. He's not here.
He's not in person, but we are running a bit early. We're about 9 minutes ahead of schedule.
Yeah, okay, maybe perhaps we could maybe have a panel session, and then hopefully maybe he plans to join like 1 minute at a time, or right. So Clint is yes. So we can just have the
sorry, go ahead.
Yeah, we can just have the question and answer panel session between Ansia and PNF, and then perhaps Clint would likely join like 1 minute at a time or something.
Yeah, okay, that sounds good. So, Ansia, I don't know if you want to come up to the front because you're here in person. And then Dr. Leon, he's online. So, do we have any burning questions or comments in the audience first for either Ansia or Dr. Leon?
I'll come up here. Sorry, but in relation to the talk that I said, we are the traditional knowledge holders of the knowledge around the use of it as for medical purposes and so on. And have any discussions taken place with the traditional knowledge holders? And what's your plan in terms of publishing?
Yes, so there are traditional knowledge holders associated with this plant because of the medicinal uses thereof. It's spread across quite a few different cultures in South Africa and further north. We have not engaged with the traditional knowledge holders as of yet. So this is where there's a bit of a fine line where in the study that we were doing was not bioprospecting or like utilizing indigenous knowledge where we were just looking at it, putting it out there. But now I think the important conversation starts because when we publish this and this genome becomes publicly available, then someone might grab that IP. So something you get.
Maybe if I can just comment along those lines. So for one case, one of the things that we sort of thought through was when the data goes on the databases, that the title would be given one case, a genome, and then the name of the species. And we hope to be able to then more easily track how many times that data set is then downloaded and used. It would be fantastic if the users of the data could take on some kind of responsibility towards a future contribution towards a Kelly fund, should they develop IP. But it's all based on good faith, as Sally pointed out. So maybe part of the discussion is what can the broader AfricaBP community do to influence compliance in that area. Just putting it out there support.
Yeah. I think definitely integrated to the conversation would be discussions on non-monetary benefit sharing as well. So we reach out to these communities and we tell them what we found related to these points that are very interesting.
Do you have any other questions? Nobody. Do you have any other questions in the audience? So if we don't have any more in the audience, thank God. I don't know, can you monitor the chat on your end?
So I'm just trying to read the questions in the chat box. So one question says, is the high-C workflow, what types of cells are used for cross-linking, considering is de novo? Then what types of cells are used for the cross-linking? Okay, they are still the same question. So I mean, I don't know if you can answer it because you didn't. I mean, you're more in the computational side, but you may attempt to give it a go.
So we use the lead, so you just need fresh tissue. We use the lead, but technically you can use any cells. Can be used with a high any cell that contain DNA in the nucleus.
I think that's all of the question from the chat here.
I have follow-ups. Sorry. Do you keep the nucleus intact during that period of time? Initially, when you do the cross-linking. I can chat to you about it afterwards, don't worry.
Maybe chat with Samira.
Yeah.
But I think, yeah, in the beginning stage, you can breathe it as is break on the nucleus.
Okay. Okay, perfect. So,
So I was just intrigued when you were mentioning the geography of where a plant would be located and its ploidy. So what is the benefit of being exoploid if you're on the side of a mountain versus diploid if you're on top of the mountain for those plants?
I can't tell you exactly the reason for these plants, but I can guess in that having duplicates of certain genes affects the expression levels thereof, and that in a way that may be beneficial to the survival of the plant. Or perhaps if you consider that these plants are similar, they're located at different spots and they therefore may have the gold.
Yeah.
I don't think they jump from being diploid to hexaploid. I think that's something that stays within a specific population in a specific region.
Okay, no, I was just thinking, Shay, I don't know if you remember, there was a presentation at 1K SA about the ants. The ants on the anthill that are also have the expression based on where the anthill is. So I just find that quite intriguing. So do we have?
Thank God. Do we have Clint online yet?
Let me check and see if Clint. Yeah, Clint is online now.
Ah, great.
Okay.
So our final presenter for the session is Professor Clint Ritter. Professor Clint, you are welcome to start sharing your slides along while I do the introduction.
I'm trying to, thank you.
Okay, perfect. So Professor Ritter holds a doctorate in genetics with specialization in population and quantitative genetics. He's a registered professional natural scientist and is currently the president of the South African Genetics Society, as well as the chair of the Department of Genetics at Stellenbosch University. He has a primary research interest in understanding how various microevolutionary forces influences genomic diversity in animal populations and how this then in turn facilitates phenotypic development. And although his work is fundamental in nature, it finds application in the genetic management and breeding of domestic and wild animals. He currently leads projects with a specific focus on aquaculture and vermiculture for sustainable animal production systems, as well as conservation of marine living resources. And in the recent years, he has increasingly applied genomic technologies to answer such research questions and has also produced the first annotated genomes for indigenous economically important South African marine species, such as the abalone and the dusky carp. And today he'll be giving us a little bit more information about how he uses genomics for sustainable marine living resources. Over to you.
Thank you very much for the introduction. Thank you for the invitation to present here today. Can I just make sure is my screen showing? It says it is. I'm not too sure whether it is. Is my video on? It's supposed to be on.
Yes, we can see you and your slide, please. Carry on.
All right. Thank you. So I'm going to talk to you a little bit about my research and how do we apply genomics to manage our economically important marine organisms. So maybe just to start off with some little context on the ocean's economy, its importance is specifically highlighted by a number of national and international initiatives. I think we all know One Health, for example, where we can speak to animal health and environmental health and even human health from a marine perspective. And I'm pretty sure we all are quite aware of the Sustainable Development Goals, in which the so-called blue economy also features in a number of these themes. So the oceans remain largely unexplored, but it creates a lot of opportunity. So if you look at the graph here to my right, at the moment, we still use the oceans a lot for food production, but there's this vastly unexplored area in terms of non-food uses that might hold specific applications in pharmaceuticals, other biotechnological innovations, and the propensity to be more sustainable in our economies as well. So from the FAO, global production from the world's fisheries and aquaculture has reached an all-time high. But the important thing to note here, if we look at the graph, is that wild harvesting from the oceans have pretty much reached its limit. So it seems to be fairly stable and in terms of harvesting capacity. And of course, conservation of these harvested species and the sustainability of our fisheries is and remains a major concern. But the demand for seafood is not reducing, and the shortfall is being made up through aquaculture, so the farmed production of marine species. It is expected that aquatic animal production will increase by at least 10% within the next decade or so. And if we look at the graph here, we'll see steady increase in finfish production, crustaceans, and mollusks from the animal perspective. But of course, there's this major part that is also made up from algae production. But to meet all of these aquaculture demands, we have to have a good understanding of what constitutes a species and where do we have specific unique populations within species that may all be viewed as independent management units in terms from a production perspective. And then, of course, we all know that sustainable animal production is hinged on the genetic improvement of the traits of interest. And this in our conventional livestock has been done through conventional or classical selective breeding through artificial selection. But what makes marine organisms quite exciting is the incorporation of so-called new breeding technologies that can expedite the genetic improvement for traits of interest. So if I look at my own research, this slide that I'm showing you here summarizes my main research components. So I work on both elements of identifying species and populations, and specifically which evolutionary forces might be shaping genomic diversity on the one hand. And on the other hand, from a quantitative genetics or genomics perspective, is how does this genomic diversity impact phenotypic expression? In other words, can we identify genetic variation that ultimately leads to improved phenotypes in the organisms that we want to produce? So with all things in genetics and genomics, it often starts with identifying phenotypic variation. We sequence the genomes to identify and the gene sequence variants that might link onto these particular phenotypes. And increasingly these days, I'm also interested in how this impacts gene expression. So we've been doing a few transcriptomics, methylomics, and proteomics studies to really give us this systems genomics view of what are the drivers of phenotypic variation. And of course, if we understand this, then we can sensibly implement genetic strategies that can facilitate domestication, because we have to remember most of these marine organisms are still essentially wild and have only been starting the domestication process over the last few years, and then linking onto that, ensuring that the production practices remain sustainable. So we've been working on a variety of marine organisms over the last number of years. And I will highlight a few things from our yellowtail, abalone, and our cob work. I didn't incorporate a lot of the urchin work there, but we are doing some work on the sea urchins as well. Yellowtail, abalone, and cob are well-established aquaculture species globally. In South Africa, the abalone industry is the most developed industry, with the finfish industry a little struggling at the moment, but still holds vast potential for improvement. So one of the first things when we consider when we want to bring an animal into domestication under captive rearing and farming is, of course, to identify what species we are working with. Now, a dusky cob has a global distribution, as indicated here on the map, and at the moment it is recognized as a single species, but there are multiple genetically distinct populations all around the world. Yellowtail, on the other hand, has a much wider distribution and has been categorized into a number of species across the globe. The species that I've underlined here were all historically seen as a single species, Seriola lalandi, with a recent revision of the taxonomy created S. dorsalis in North America, and in Asia, Seriola aravitata. So what is interesting here is that here we have two species superficially very similar in their life history characteristics, although we do know for both dusky cob and yellowtail the individual populations all have life history and morphological differences that have been noted and well documented across the globe. So we've recently sequenced the dusky cob genome, and there is an available dusky cob genome from China as well. And we did a comparative analysis between the two conspecifics from South Africa and from China. And our evolutionary analysis indicate that yes, that they share a common ancestor, but there's quite strong support for these two to be separate lineages. And if we actually look at the divergence, we see as much as 8% divergence between the two genomes of the geographic conspecifics. So that essentially means there's only 92% similarity between the two genomes. Now, quite often people ask, so why is this interesting? And then I often refer to the example of the human-chimp genome similarity. Now, depending on who you ask, or which paper you might be reading, the human and chimp genomes share about 96 to 99% similarity. And these are two organisms that are seen as two species in separate genera. Now we have two genomes that apparently is supposed to be from a single species, and they only share 92% similarity. So digging into some of the functional elements, if we look across gene enrichment across the two genomes, the South African genome seems to be specifically enriched for immune function related genes, whilst the Chinese genome seems to be particularly enriched for transposition, sensory, and stress response genes. And as a pet interest of mine, we've also explored transposable or repeat elements across the two genomes. Now, it is interesting to note that the Chinese genome seems to be enriched for genes with a function in transposition. And if we look at the graph here, again on my right, you'll see that transposition has been quite active in the Chinese conspecific genome in recent history versus the South African genome, which saw a lot of transposition events in the past and a little less in recent history. So that might explain some of the divergence. And it might also hint that we might in fact have two separate species here rather than just having one species. We've done something very similar for the yellowtail genome. Again, we sequenced and assembled the South African yellowtail, Seriola lalandi, and we had a few genomes available from Australia, from China. These are two Chinese genomes, and then genomes from North America. The tree you see here is based on gene expansion and contractions, but perhaps what is more important for us to note here is that we have a 98 to 99% similarity between the genomes, and these are across all of the so-called yellowtail genomes that historically used to be S. lalandi. So if we again look at what these gene expansions or contractions might be related to, if you look at the South African genome, our initial analyses highlight some immune response genes, and motor activity genes versus, for example, the US genome that has a higher proportion of these gene expansions in sensory regions, but again, also immune response. So perhaps rather than having multiple species, do we perhaps have a single species across the globe with unique populations that have been differentially adapted for the various environmental circumstances in which the particular populations might find itself in. Now, again, these are preliminary results. So these are just our initial explorations, but it's hinting that genomics might be an important element in trying to resolve taxonomic uncertainties that might exist. We have to also keep in mind that marine organisms are many times older than most of the land-based organisms. So we expect their genomes to be a lot more diverged. We expect there to be a lot more diversity across the various taxa. So the two examples I discussed from cob and from yellowtail are very broad on a global scale identifying species, but in a similar way, we can use genomics data to also differentiate local stocks within a specific geographic region. So the South African abalone, which is a mainstay of the South African aquaculture industry at the moment, is known to occur across the South African coastline. And some of the initial population genetics work have highlighted that there's at least 2 separate genetic stocks. Then we overlaid some genomics work. So this was population genomics work from my own PhD many years ago. I don't even think that this work will technically qualify to be genomics level work these days anymore. But we had genome-wide SNP data to disentangle the neutral versus adaptive diversity across the coastline, just to make sure that we really do have separate stocks. So the east coast of the country has fairly warm water. The west coast of the country has fairly cool water. And then there's this secondary contact zone where the two populations mix. And what we do see using this whole genome-wide SNP data set is very strong genetic differentiation between the east and the west. And then with this hybrid zone on the south coast also clustering separately. So when we ran signatures of selection analyses, many of the loci that popped up at that early stage of the investigations seem to be associated with respiration, energy, metabolism, which again might hint that the population divergence that we see along the coast might indeed be linked to warm and cold waters on the west and east coast respectively. So that there is seemingly an element of adaptation happening here, which of course, given the various scenarios for climate change, is becoming increasingly important for us to understand what is conferring heat stress resilience. So we do have a initial draft genome of the South African abalone, and we have recently resequenced that draft genome for both a West Coast and an East Coast individual, and we are in the process of exploring regions of the genome where the West and the East Coast populations might be diverging. Some of our initial transcriptomics work did highlight differential transcriptomic patterns across the geographic regions in South Africa, where Saldanha Bay represents a West Coast population, Reed Point represents an East Coast population. So Saldanha will be cold water, Reed Point will be warm water, Witstrand is a South Coast population, and then we had a cultured population as well for comparison. So there seems to be some differentiation in gene expression happening, and we would like to link that onto what is happening at a genomics level. We don't quite have data for this yet, but hopefully we will generate data for this soon. So why is this important? So one, for fisheries and conservation management, if we understand what is a species and we understand where do we have localized adapted populations, then we can set the fishing quotas accordingly within and across national boundaries. Not that important for the South African abalone, but something that might be more important if we start talking about the yellowtail or the cob fisheries. Can also help us to design more effective marine protected areas. And then, of course, if we need to implement conservation breeding strategies for population augmentation, we would know from where to source specific wild animals to produce cultured stock that we can then release back into the environment as a initiative for conservation. From an aquaculture perspective, in terms of genetic improvement, having different species and having different populations might mean that we have to think twice before we just translocate animals from one region to the next, because if the genetic constitution of the populations are not the same, we might not maintain the genetic gains that we think we might have with a conventional selective breeding program from one region to the next. But it can also give us interesting opportunities to explore breeding strategies across a number of species or populations to potentially exploit hybrid vigor as a means to increase production. So if we move on to the second important thing that is required for the sustainable use of marine organisms, and this is in terms of genetic improvement of the traits that we are interested in, we need to keep in mind that most aquaculture species are still essentially wild, especially those in South Africa. We could potentially argue that maybe the abalone is on the verge of domestication. But what is also interesting, because they are still essentially wild, we see a lot of phenotypic variation. So the two pictures you see in this slide is essentially animals from not even the same population, but they are from the same family. So if we look at the abalone here at the bottom, these are all full sibs. And you can see just amongst the siblings how much phenotypic variation we can expect to see. Similarly for this, the picture of the cob here, these are full sibs. This is a smaller individual, this is a larger individual. They are all the same age, right? So there's a lot of variation that we can potentially exploit. And our initial estimates of heritability would suggest significant genetic component to phenotypic variation, suggesting that we can have a lot of genetic improvement with a selection program. So to explore this a little further, to really understand the genetic architecture of these production-related traits. We constructed the first high-density linkage map alongside our genome. We conducted QTL analyses and then mapped our QTL loci back to the genome to identify potential candidate genes. Now, what was interesting here is that the few candidate genes that we did identify, or not candidate genes, the few QTL regions that we did identify seems to explain a significant amount of the phenotypic variation, anything from 7 to 11% of the variation can be explained by a single locus. Now, those of us that's familiar with the theory of complex traits would immediately recognize this as either a major gene or that we might have a compounding effect at a single genomic locus of multiple causal variants. We saw something, so this is all on abalone. To further explore this size differentiation, we also did a comparative genomic analysis because size variation is quite distinctive between a number of abalone species. And identifying genes with a signature of selection across species with varying sizes, we also identified a few additional candidate genes involved in nervous system functions and gene regulatory functions, which suggests that size variation across the species might be a function of both how the organism perceives its environment and how it then regulates its genes. We did a very similar QTL analysis for the dusky cob. Again, we identify similar loci across the species, even though abalone is a snail, is an invertebrate, and cob is, of course, a fish, which is a vertebrate, but similar trends are being observed of these large effect QTL, and then if we map that back to the genome, we identify a number of candidate genes that are involved in biological processes that can explain size variation. So the general implications of this is then the undomesticated nature of most of our aquatic animals seems to have great scope for genetic improvement because there are still many major gene loci segregating in the populations. So if we compare that to some of our conventional livestock, many of these major genes have already been fixed in the commercial populations for many years, which might explain why our conventional livestock is not responding to selection as vigorously as what our aquaculture species seemingly can. But aquaculture species in itself creates some challenges that the conventional livestock industry did not necessarily have. So one of the constraints is that quite often we have a vast array of species, all with unique biological characteristics. Populations are often small, so the founding populations might be subject to specific founder effects, which can have a negative impact. Unlike most of our conventional livestock, we work with animals with a variety of reproductive modes. Most of our aquatic animals have mass spawning, which really makes it hard to sometimes record the pedigree information, but there are workarounds like using DNA fingerprinting to reconstruct the pedigrees. The other important thing that might hinder selection in these animals is, of course, their long generational intervals. For abalone and for cob, that can be anything from 3 to 6 years, where if you compare that to conventional cattle, did you lose my screen? Sorry.
Yes, you need to reshare, please.
Do I need to reshare my screen?
Yeah,
Can you see
yes.
my screen?
No, can't see it.
Okay, let me just reshare that quickly. Sorry.
Also, Professor Clift, I don't know how many more slides you have left, but we have about 1 minute remaining for the session.
I'm almost done. I'm
Okay.
almost done. Can
Carry
you
on,
see my
please.
screen now?
Yes, please. We can see it now. Yeah.
Okay. So a combination of marker-assisted selection and classical breeding can yield rapid and fast improvement. And then the array of candidate genes that we've identified creates interesting opportunities for gene transfer or gene editing technologies to expedite genetic improvement. So I need to acknowledge my students and postdoc working in my research group on all of these things that I've shared with you today, and all of my funders and the Africa BioGenome, as well as all of the farms that I regularly work with for the biological specimens. Thank you very much. I'm happy to answer any questions if there are.
Thank God, is your hand up or are you clapping?
Yeah, I think we have a question online or
Okay.
questions. Yeah. Shall we go first?
You can go first. Do we have just one though, in the interest of time, so that we can do the T-break and people can
Okay. So I'm gonna merge the two questions. So thank you, Professor Rode.
Sure.
I will have a question from Hewan and Rector. So Hewan is asking, given the complexity of abalone, right? Populations and breeding environments, how confident are you that the associations identified aren't driven by hidden co-founders rather than through genetic signals? So that is one question. The second question from Rector is saying, any collaboration between Sire Biobank and Stellenbosch University to curate extracted DNA sample of these studies? So is there any collaboration between
So maybe I'll answer the second question first. So at the moment, there's no active collaboration between my own group and SciAb. Of course, I am always open to collaboration. I think combining forces to answer the pertinent scientific questions of the day is always a better way to go. So if you are from SciAb and you would like to know more, I'm more than happy to drop me an email. Then the second question about the complexity, that is an interesting and very valid question to ask. So if we're just looking at sequence variation and the differentiation signals that we see, it is, of course, at this point a statistical association. So do we need to confirm this with functional studies? I think so. And that is certainly where the research is most likely going to go. The challenge, of course, with that is if I just look at the dusky cob or even the abalone, it's not the easiest organisms to work with. There's not established protocols for functional work. So we are investigating model organisms like the zebrafish or model organisms like the common garden snail to try and see if we can use that as a laboratory model for some of the functional validations that we need to actually confirm that some of these genetic signals. I hope that has answered your question.
Yeah, so I think the question was online. So I'm sure, Hewan, I'm sure you are satisfied with the response as well as Rector. So we can go back on site. Any question on site? Over to you, moderator.
No, we don't have any questions here on our side, thanks.
Yeah, so Clint, I think there are some chats going on the, so you can just please spend some time to respond to them. All right.
Sure.
Yeah.
Okay. Thank you very much.
Thanks so much, everyone. Thank you, Professor Roda. And thank you to all of the other speakers of the session. We have our tea break now, until 3:40. So for those online and everyone in here, we'll see you guys back here for the last session at 3:40.
We're really interested in using high-dimensional phenotyping or phenomics to uncover the mechanisms of disease biology. And particularly, we're really interested in the biological mechanisms of genetic variants and mutations that are associated with both neuropsychiatric and neurodegenerative disorders. And we've been really interested in working with Element on their new technology because morphology is only one measurement. We have recently published this work on what we call neural painting, which is simply taking cell painting and then applying this to neural cell types. It's really easy in the lab, but the processing and analysis can really take a long time, and that feature interpretation that you get from that always requires follow-up experiments. So you have to then go measure RNA, you have to measure protein, and then try to understand which ones of those give rise to the morphological changes. And so the AVD24 platform allows us to capture all three of these simultaneously. And so we set up a pilot experiment with the AVD24 team where we took iPSCs from our lab, we differentiated these into both astrocytes and excitatory neurons. We grew these cells on the Teton flow cells. We would fix them, and then we sent them to Element for processing, part of the technology access program. Firstly, we're able to take the single cells and cluster them using the RNA expression transcripts with the AVD Neuro panel. But here, we actually get better accuracy, and we get the added information of kind of the RNA expression. So here again, we're just taking these clusters that we found using the gene expression information, but then layering on here values for the morphology features that seem to discriminate our cells. But one could easily imagine now going into each of these clusters and doing different kind of regression analyses to sort out whether there are specific proteins or transcripts that would associate with these morphological characteristics so that we can begin to draw linear relationships between protein and RNA activity and how cells change in response to certain contexts. You know, doing 1 million cells in a run is feasible, right? And having the data to come back after simply overnight, effectively, is really incredible. Sometimes I think it's hard to kind of grasp your head around rather than waiting weeks or months to get information on 1 million cells. You can kind of have it right away. The actual experimental part of it is incredibly easy. You just need to grow cells and fix them. In a system like this, where we can use many different dimensions to measure, at least in vitro, when things begin to change, that gives us a better sense of maybe what's causing disease rather than what is caused by disease. And so doing everything simultaneously, I think, could be really powerful.
Welcome to the 20-year anniversary video of AfricaBP, Africa's genomics company, here from our headquarters in Mackleneck, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of AfricaBP emerged not far from here in Pretoria in the year 2000 while I was doing research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so. The startup was only possible with the substantial direct foreign investments, mainly from shareholders from Germany and Switzerland, as well as the startup loan from the State Secretariat for Economic Affairs of Switzerland. So what was actually our idea in 2000 to start the biotechnology company in South Africa? The aim was to locally produce oligonucleotides which need to be custom made, and therefore by local production the turnaround time dropped from 2 to 3 weeks for imported oligos at that time to 1 or 2 days. This of course allowed to accelerate research output for our customers that apply PCR amplification and DNA sequencing in their research. And more recently, it allowed uninterrupted essential supply during the pandemic. In addition, we aim to offer Sanger DNA sequencing services. The aim was here that outsourcing DNA samples for sequencing will allow our customers to save the investments in capital-intensive sequencing instruments. 20 years later, we still do all these services, but of course the technologies in life sciences have significantly evolved in the past 20 years, and so has AfricaBP. A big thank you to all our customers, our dedicated current and former employees and commercial partners over all these years, and of course our investors that took the risk and made this possible in 2002. Please enjoy this video about the different aspects of AfricaBP. Let us know your comments, suggestions or questions about AfricaBP in the comment section below. We strive to even be better in the next 20 years.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation. MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DNB-seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of few companies in the world that have the ability to develop and mass-produce low, medium, and high-throughput clinical gene sequencers from GB to TB. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, BIT products, and low-temperature storage products, with a strong focus on the upstream of the industry. MGI operates in more than 100 countries and regions, serving more than 2,800 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
All right, Greg, you're on mute.
Thanks, Angrad. So we will start our final session of the day. And in this final session, we will have two presenters. And I'll introduce our first presenter as he shares the screen, and I've got the proper time this time. So we send a warm welcome to Prof. Python Samatanga, who is a molecular and computational biophysicist and life sciences entrepreneur who founded the Biotech Institute in Harare, establishing next-generation sequencing, clinical diagnostics, agroanalytics, and bioinformatics laboratories, and developing a cancer genomics bioinformatics platform to advance precision health in Zimbabwe. He previously served at the University of Leipzig, first as a senior research scientist, then as a full professor of biophysics, of physics of biological and soft materials, with research focused on single molecule spectroscopy, and elucidating the CRISPR-Cas gene editing mechanism. He holds PhD in biophysics from the Swiss Federal Institute of Technology, an executive MBA from Quantic School of Business, and is a member of several organizations such as the German Biophysical Society. So with that, I would like to invite Prof. Samatanga to start sharing his screen, and we can get the presentation underway. Thank you.
Is Bright on site? Oh, because he's not online. He's not. I can't see him online.
He's not in the venue.
Yeah. So.
Is Gain on my tab?
It's not online. So let's see if we have Gain. But Gain is also not. Yeah, we don't have Bright online yet.
Okay, so maybe we'll give him another 5 minutes to join.
Yeah.
Otherwise,
otherwise
we'll adjourn until time for Kane's presentation.
Mm-hmm. So maybe let's give him 5 minutes. Yeah, he's still not on Zoom yet. Bright Samatanga. So I'm still trying to figure out if he joined with a different name. I don't think he joined with a different name.
And we just got confirmation from inside the venue that Professor Matanga is not going to be presenting today.
Okay,
So,
all right.
okay, so we'll wait until 4 o'clock for when Kane is online.
Yes.
And then we'll close off the session.
All right, let's wait for the last speaker then. So I think we'll just give Gain 6 more minutes to see if he'll join. So since he's the last speaker. Okay, Gain is here. Hi. Okay. Again, I'm just gonna make you a co-host.
Okay.
You need to unmute, Craig.
Our presenter for this for today is Professor Jane Wong. So Professor Wong is a professor at the University of Alberta in Canada, and he highlights that his research has gone through several phases for now, and it's entering its third. And the sole commonality between these phases is the continuing merger of life sciences, which includes medicine with information technology. His first phase was the development of infrastructure to acquire genomic data at unprecedented scale. And phase 2 was triggered by the million-fold reduction in cost of sequencing, which has allowed us to study human genetic variation in very large populations and to sequence beyond organisms of known economic importance. For example, the 10,000 plant genome initiative and the 1,000 plant transcriptome initiatives, both of which he has led. So I will invite Prof. Wong to share his screen, and then we will get going with the final presentation of the day. Thank you.
Okay. So are you projecting the slides or do I just share my screen?
Yeah, you need to share your screen again, please.
Okay, no problem. Then I'll bring it up here. Okay, now how do I share screen? Share screen. Here we are. Share screen.
Yeah, okay, perfect. So it's coming through now.
Can you see it now?
It's powering up. Yes, so you now need to, it's showing us several slides. So what you need to do is to, yes, perfect.
Does that work now?
This works. Go ahead, please.
Okay, so I want to first thank the organizers. I've been sort of informally advising them on their plant sequencing for the last few years. It's kind of been an interesting experience because it reminds me of like a quarter century ago doing the same thing with my friends in China. And that ended up with the Beijing Genomics Institute. So that was a huge success. I really wish the best for the African BioGenome. So today I want to talk about both the Earth BioGenome and the 10,000 Plant Genomes Initiative. They were both sort of officially launched in 2017. There's a couple of papers, I guess one is in Science, one is in Nature. So there was an announcement for the Earth biogenome in 2017. And then the 10,000 plant genome was announced at the International Botanical Conference in Shenzhen in 2017. Shuzhen and I announced that. And we got Dennis Normili to write up an article in Science thereafter. So they're both ambitious projects, ultimately to sequence all the plants and animals known to science. And a lot of things have happened since 2017. Two things, most important. One is the expectations for the quality of a genome sequence have dramatically gone up. So the first thing that happened, and it started around 2018, the DNA Zoo came online. And so this is a database of primarily mammalian genomes that, because of high-C technology, they were able to extend the scaffolds to a chromosome level. And actually, to this day, my colleagues at BGI just updated the number. There's about 336 animal genomes are released on DNA Zoo and only 21 plants. It turns out that high-C is somewhat more difficult to run in plants, but nevertheless, there are expectations of very large contigs and scaffold. Then in, I guess this one was 2022, the human community published the first telomere to telomere assembly. This takes advantage of the long reads from Pacific Bioscience and Oxford. And the idea is now you can actually sequence through the heterochromatins, the telomeres, the centromeres, which when the first human genome was done a quarter century or more ago, simply couldn't be done. And so that's also raised the expectations for assemblies. But then on the negative side, much more negative, of course, as you're very well aware of, there was the pandemic starting in early 2020. And for like 3 years or more, there were no plant or any shipments of any samples around the world. This particularly hurt us with 10,000 plants because we, unlike, say, the Darwin Tree of Life in the UK, where they sequence everything in the UK and don't have to worry about getting through customs, a lot of our stuff is ultimately, if 10 kp, will come from around the world. And so basically, we were shut down for 3 years in a row. Now, despite all that, so here's a state of the progress. This is an up-to-date chart. I'll have to explain how this works. So this is going by taxa. And, at the order, family, genus, species level. The red indicates what has not been done. And what I'm showing here are all the genomes that, to our knowledge, have been sequenced and assembled, not necessarily published. So a lot of these genomes are not published, but we're good friends with the people at the Darwin Tree of Life in the UK. And of course, we took everything that was at NCBI, representing the stuff that's already been published. And of course, this blue stuff is from BGI. And one thing I should comment, is a little subtlety, is the red part shows what has not been done, and that's the pure percentage. You can see, obviously, as you get to finer detail, like the species level is 90s, high 90% not done. At the order, we're almost done for most taxa. And the way I draw these things, you got to think that at the order, family, or even genus level, it's entirely possible that BGI and NCBI DTOL will have done the exact same order, same family, same genus. So in order not to double count, what I'm showing there is the relative contribution. In other words, if they've both done the exact same number of orders, then it will have an equal weight on this chart. So the BGI versus NCBI DTOL thing is an indication of relative contribution, but the total amount is the correct amount. And if you then, this is broken down by taxa. If you break it down overall across all plant, there's an interesting plot. So BGI, 10,000 plants, has, if you look at what has been done at both at the order level and to a lesser extent at the family level, BGI has done almost as much as the rest of the world. On the other hand, if you look at the genus level, it's a much smaller proportion, and you can't see what's going on at species levels, even less. And that reflects the fact that our project is focused on phylogenetic diversity. We're more concerned with getting a representative of every order, and then, of course, every family, and then eventually every genus, and so on. Whereas, particularly, one of the other major producers, the Darwin Tree of Life, is focused on sequencing plants within the UK. And so that gives a different bias in how these things turn out. So anyways, so that's the state of the progress of the sequencing. But again, I would emphasize that this is not necessarily all published. And if you want an overall number for the total number of plant genomes that are at least assembled, if not necessarily published, it's something like 3,000 to 4,000. And we know this because we've been spending the last little while, I'll tell you why in a little bit later, we've been gathering together all the sequenced and assembled genome as part of a larger consortium to generate a big paper on it. But so the number, so you should appreciate, is approximately, it's over 3,000, and that's non-redundant counting, okay? So next question is publications. So what has been going on in terms of publications? So it's, of course, difficult to publish a genome and just say, here's the genome. Well, you could do that. There are, I guess, data publications. GigaScience has them, and Nature Science will have them. But most people want to publish papers that find something interesting. And of course, that ensures you get into a better journal. And so many of our publications have been focused on horizontal gene transfer. For people who don't appreciate that, for the longest time, of course, everybody assumes that genes are inherited parentally from your mother, your father. And I guess around 20 or 30 years ago, the bacterial people realized that horizontal gene transfers from unrelated species were quite common. And I guess in these days in the bacterial world, it's not uncommon to see a genome where something like 20% of the genes will be horizontally transferred. It also throws into a big wrench in the whole idea of a phylogenetic tree. But for eukaryotes, this is considered rare. And for the longest time, people thought that would be next to impossible because the genes would have to get across two membranes, both the outer membrane and the nuclear membrane. But in recent years, we have been finding that this can still happen at a much lower rate than in prokaryotes, but nevertheless, it happens. And we are finding as we sequence more and more genome that it's almost difficult to find any genome where you don't at least have a handful of genes that are very clearly came into the genome from a horizontal transfer. And so some of the papers that we've been publishing, we're finding that one paper I'll show you briefly is that one of these transfer from a soil bacteria was probably responsible for terrestrialization. In other words, the transition of green plants from water to the land. And more recently, in unpublished results, we're finding that plant immunity is also likely have come about from horizontal gene transfer. And so more generally, the field is now moving towards not just looking at the existence and occurrence of horizontal gene transfer, but at least from our end, we're now looking at the mechanisms by which these genes can transfer some foreign organism. So they include endosymbiosis, which we call EHGT, or very long word, nucleocytoplasmid large DNA viruses. These are recent discovery, these ginormous viruses that are physically as big as a bacteria and whose genomes are as big as a bacteria. And then the most recent one that we have now in review is phage-mediated horizontal gene transfer, where bacteria viruses, phage, are involved in helping the gene get inside. So just to show you, the first paper this came out in 2019, so our early paper, this was in Cell, where we published, I guess, the first genome. So I never pronounced this right. It's my colleague, Michael Malconian, is the algae expert. And he calls it, I just call it Zygnema, and I'm happy with that. Anyways, this is the sister group to the land plants. And when we sequenced these, we found that there's a particular gene family called GRAS, which was already associated with terrestrialization. And we found that those were suddenly introduced in this taxa through a horizontal gene transfer from soil bacteria. And this made a lot of sense because these particular algae, they live in what Michael calls a subaerial or terrestrial, where they sometimes live in a dirty pond or a small lake, and then the pond dries out, and then they live in dry land for a while. So they oscillate between a dry land environment and a sort of a muddy pond. And so it makes sense that this is sort of a species in transition, and it'll be in constant exposure to soil bacteria that live in the same environment. So that was an early result. And encouraged by that, we've been looking for more and more instances of horizontal gene transfer. And not just us, many other people are finding it. So as I said, it's now getting hard, in fact, to find any species that does not have at least a few horizontally transferred genes. So a more recent discovery that we made, which is unpublished, is still in review, is this another group of algae. And when we look at these, so this is the typical thing you do, you draw a big genome, you show where it is phylogenetically, and then you draw a figure like this. All of these papers have this sort of the same structure. First you sequence a new clade in the phylogeny, and then you look phylogenetically as you see the tree there, and you look, is there a sudden burst of a gene family? And so what we see here circled in red is a sudden burst in gene families, and this is a gene family that is known to be involved in critical for plant immunity. And what we see is that not just one gene, but many related genes just suddenly came into appearance. And so that's interesting and suspicious. And so to show that this really is the gene for plant immunity, because the standards for publication have been going up, this is sort of an issue, is it wasn't sufficient to do just the protein homology by sequence comparison. We did some 3D structural work with AlphaFold and whatnot just to show that these proteins that we're finding in these algae have the same 3D structure as the Arabidopsis version, where there had been experiments done. And more importantly, we convinced some colleagues to do a heterologous expression of these genes in, I guess, tobacco and with agrobacteria-mediated transient expression, just to show that these algal genes really are doing what we think they are doing for plant immunity in a land plant. And this is partly because I guess the standard says, interesting, back in 2019 when we did the Cell paper, it wasn't necessary to do these kinds of experiments, but people are now raising the bar. So in addition to showing evolutionarily that something appeared and it may be a horizontal gene transfer, they want you to do functional experiments to show that it really does what you think and what you claim that it's doing. So it's just the standards going up, not just for the quality and of course the sequence has to be now chromosome level contigs, and you have to prove things. So the standards for publication, understandably, are still going up. And then, of course, the other thing that was interesting, of course, you have to show the phylogenetically that these really are more closely related to, in this case, they were related to phage-mediated or bacteria. It's always hard to know when you have a phage gene, whether it came from the bacteria or whether it's intrinsic to the phage. But what was very interesting when we started looking into this particular transfer is that adjacent to where we see it on the algal genome, we always see a trace of the phage sequence and all the phage components. And so that made us look, I don't have that figure here, is we started looking for these phage residues all over the land plants, and they turn out to be everywhere. And so that suggests that, and in fact, we even started looking for these phage remnants in animal genomes, and they turn out to be certainly present in birds and other animal genome. So what it opens the possibility to do, because phage is a natural vector for a gene that would be, the phage would then take, well, here's the picture that we have in mind. So we call it phage-mediated horizontal gene transfer. So you have phage starting with some sort of a domain, which is sitting in some bacteria, I guess in this case. Then phage infects it. There's some recombination event, so it pulls out of the bacteria. And then let's see, then the phagocytosis and endocytosis, you can look at that. I guess, I don't know if I'm leaving. Yeah, I can post some of these slides or something. I don't mind posting some of these slides, but I may have to take out some of the industrial slides later if you guys want to see it. I don't want to dwell into it, partly because it's still under review. But basically, the idea is very complicated. But there, if you imagine the advantage having a phage carry it's a vector for which you can use to get the gene inside, not just the exterior membrane, but inside the nuclear membrane. And so this is something that we're still trying to get it through review, so that's why I'm a little bit cautious about going through everything. But it's been an endeavor, because this has been about 2 or 3 years work since the pandemic, trying to get all the details to satisfy the reviewers, and the level of proof that is required is substantially more than what we got away with in Cell back in 2019, which is an interesting observation. And this has caused problems for us, because really what we're releasing here is this paper is only meant to, I don't know, maybe about half a dozen genomes. And meanwhile, we're sitting on close to 1,000 genomes. So you do the math, this is a big problem. if we keep trying to publish paper, we can. I mean, we've done it before. We've gotten into big journals before. I'm sure we keep plugging away, we can get into big journals. And for the young kids who are involved in the project from a career perspective, it's obviously to their benefit to publish in a major journal. The problem is if we keep putting in this amount of effort just to publish a half dozen genome, we've already got a backlog of close to 1,000 genomes. It's clearly not a sustainable path to publication. I think this is going to be a problem for every one of these big genome projects, whether it's the African BioGenome, or the Darwin Project, and certainly us, is if we are successful, we simply cannot publish a few dozen genomes at a time. There's just not enough. And also, even if we could crank out the papers, the journals won't take that many papers. They just can't. And they'll get more and more diluted. So out of frustration, this happened last year at the most recent International Botanical Conference, which was held in Madrid. So out of frustration, I was having a hallway discussion with my good friend Michael Barker and lamenting the fact that everybody doing these production, we've got a backlog of genomes that we haven't been able to publish because the publication standards keep going up. And yes, you can release the data, but there's obvious reasons for the kids who are doing the work why we want to get publications for them. And so we both were commenting that this increase in expectations for the quality of the genomes, where if it's, I just had a commentary, there was just, I don't know if it's been mentioned in your conference just last week on, I guess, Wednesday or Thursday, Frontiers in Science just published the latest flagship paper from the Earth BioGenome, and I have a viewpoint there. And my point is that whatever their so-called gold standard is nonsense. It's an arbitrary line in the sand. They don't ask for telomere to telomere. They don't ask for haplotype resolve, particularly for polyploidy, which is common in plants. And I would argue that we should be doing pan-genomes because, of course, no one genome from one individual will capture the genetic diversity of a population within a species. So what we really need to do is telomere haplotype resolved pan-genomes, and that's grossly beyond the capability of, beyond budget. Well, maybe technically capable, but it's way beyond the budget of what we could hope to do for any of these projects. So to be blunt, the so-called gold standard that the Earth BioGenome people talk about, to me, is an arbitrary line in the sand. And I've lived through this because I was involved in sequencing the first human genome, and we set an arbitrary line in the sand, which in the end wasn't the final goal anyways. So it's just something important to remember. And I think the more important thing, and even the journals understand, is that the publication standards should be based on what conclusions are you trying to draw. And the funny thing is, you don't need chromosome level assembly to learn a lot of things. So to show that a gene was horizontally transferred, you don't need chromosome level assemblies. The only reason why the reviewers are holding out to that standard, they figured, well, for half a dozen genomes, not entirely unreasonable to hold these guys' feet to the fire and make them generate higher quality genome. But that's the thinking. In terms of the discovery of the horizontal gene trend, it's totally unnecessary. Similarly, Mike Barker had been doing some calculation looking to compare synteny, and he wanted to compute all the synthetic blocks across the green plants. And he's already determined you don't need very large scaffolds to do this. And you can go on and on and on. There's a lot of things where you don't need chromosome level assembly. And of course, there are obviously things where you want telomere to telomere, but there's a range of different questions that can be answered. And it is just silly to hold ourselves or anybody else's feet to the fire when things don't require. And the only reason why that happens is because you're only submitting half a dozen genomes. So the plan was to get together with everybody else who has these unpublished genomes and do something big where, because it's such a large data set, the reviewers cannot say, oh, you must push everything to chromosome level, telomere to telomere. I mean, it's easy, it's annoying. It's easy for reviewers to say, you must do this, this, and that. You know, I'll tell you, when I got the 1,000 plant transcriptome published on the cover of Nature, we had a big impasse because the reviewers said, you should make them full genomes, not just transcriptomes. You know, that's insane. But this is the kind of thing that goes on review. So the way to get around that, you are vulnerable if you publish a small number of genomes. But if you publish a large number of genomes and you have an interesting discovery, they back off. And so we are going to do a paper with all available genomes from not just ourselves, not just published, with Darwin people, with other people, and Fei Wei and other friends, and the Canadians, et cetera, where we're just going to throw out anything that has a reasonable assembly quality, and just show there's a number of things that we can show. So Mike is convinced that he can compute all the synteny, and we've got at least like 70 to 80% of all the orders, and half the families out there. Is it complete? No, but when we published the first human genome, I don't know, a quarter century ago, that wasn't complete either. It wasn't only until a couple of years ago that we did the telomere assembly. So it was like 20 some odd years between when we claimed that we had a finished genome and then we actually did it. And even now, you still have to do the pan-genome. So it's just silliness to have to set an arbitrary standard. So we're just going to try to break that and try to publish something so to demonstrate, because horizontal gene transfer is so common, and I suspect that when we go taxa by taxa, many of the major evolutionary transition, whether it be terrestrialization or plant immunity or whatnot, my bet is that a large fraction of them is not going to come from some gene duplication. They're going to come from a horizontally transferred gene, which evolved in some microbe, whether it be a bacteria or even a virus or something. And so that'll shock people. And we already have anecdotal evidence that that will be the case. And so that would be the punchline or the thing for that paper. The other thing, of course, having said that, people do expect better and better assembly. That's fair enough. And one of the problems that I face at 10 kp, which Darwin does not have, is because the funding for 10 kp came from BGI and not from some granting agency. And BGI justifies it as a sort of a publicity vehicle for their sequencing technology. And their MGI technology, as you know, is a short read technology similar to Illumina. And so if we need long reads, we have to pay for PacBio or Oxford. And they don't want to do too much of that for pretty obvious reasons, not the least of which now that's going to an American company, and there's this trade war with China and US. Anyway, so what we have done is we have developed our own Nanopore technology. And this is, there'll be more announcements in another couple of months about all this going on. So let me just tell you roughly what's going on with the plant 3 kp. So one of the things for the 3000 plant genome, we call it plant 3 kp, but these are now assembled genomes, and they're going to be covered. I showed the semicircle charts that I show you of all the distribution. That's the taxa that we have covered. One of the first things that we realized that we would have to do, we needed to have a consistent annotation. So first of all, we wanted to re-annotate all 3000 genome, because if we take what is in the database, whatever, is wildly inconsistent. And then if you start doing that, you also have to worry about the compute time, because annotating 3000 genomes is not a trivial thing. And so we have been designing and testing a new pipeline for both repeat and protein coding regions. So for the repeat, the main issue is the traditional method for what we call repeat masking, dating back to Arian Smith in Seattle. I know Arian back in those days when he was developing repeat maskers. You run something like what he calls repeat modeler, which builds the ancestral consensus sequence for the repeats. And then you run repeat masker to look for all the degraded instances of the repeat. And repeat modeler is actually an extremely slow process. And so what we intend to do is we're going to take and build sort of a library of known repeats, taxa by taxa, so that we do not actually run repeat modeler on every single genome, all 3000, because that would be too slow. The other thing that's, in fact, more interesting and more important is when it comes to annotating the exons for at least the protein coding genes. Traditionally, you have your hidden Markov model, ab initio methods, and they've never been very reliable. And so people rely on RNA-seq. And I guess the gold standard these days is something like called Breaker 3. And more recently, people using artificial intelligence neural network have shown that you can actually use the latest neural network methods. Is an algorithm called Tiberius, published just last year in bioinformatics. And they were able to demonstrate that without using RNA-seq, you can actually get an annotation which is maybe even better than Breaker 3, which uses RNA-seq. So we are in the process, but of course that was trained for human genomes, the mammalian genomes. And so we are in the process of retraining it right now for plant genomes, all the way from angiosperms all the way to algae. There will have to be taxa-specific training. And so this is very encouraging, because when you look at the availability of RNA-seq, there's like 30 or 40% of the plant genomes out there, they didn't even bother doing RNA-seq. So we're not in a position to go and run, thousands of RNA-seq experiment on species we don't even necessarily have access to. So the beauty of this is we can get a consistent annotation. It'll be faster, and we don't have to rely on inconsistent to non-existent RNA that we may otherwise have to generate our own data sets. So this will be a big boon. We're still benchmarking the pipeline. We'll publish something on that before we go ahead with the whole thing. But this is an important issue, because then it allows us to do a consistent and a much faster annotation of thousands and thousands of genomes. And then so that means that where we see differences in patterns and what's evolved and what's not involved, it won't be an artifact of the inconsistent availability of RNA-seq, which was a big concern. Okay, so lastly, I wanted to show you the new BGI platform. So this is basically similar to Oxford Nanopore. I'll let the company, BGI of course has a public and a private entity. So BGI Research, where I'm affiliated, is for a nonprofit, but there's also a commercial arm which does MGI sequencers and does diagnostics, etc. And so this will be, at the moment, this I believe is under BGI Research. I'll let the company figure out how they'll deal with patent issues. But the current N50 read lengths are about 30 to 50 kilobases. It goes up to about 100,000 if you do it right. The accuracy, single reads are 98%, and then the consensus is 99.995. So it's getting pretty decent. I think what's important is the two instruments. Last year, it was at about 85 gigabase per chip, and they were reluctant to do things like a tree genome. Now, the latest model that just came out, it's about 4 or 5 times higher throughput. And in fact, one of the things there was an interesting paper, a news article in Nature, saying that there are a lot of rare trees where the only available specimen is at a Buddhist monastery in China. So it turns out all these Buddhist monks for thousands of years have been lovingly taking care of all these trees, and some of them that may be the only place they're available. So I sent out our guys to go talk to a bunch of monks to see if we can get all these obscure trees into the pipeline. And we wouldn't have done that last year because their throughput is limited, but now with the new instrument, it's reasonable. So there's a little corporate video I'll show you. Let me see if I can blow this up. So it's a chip like everything else. You have a little drop of all your DNA, goes on. And then there are a gazillion nanopores that it goes through. I'm not at liberty to say how they do the detection. There's IP issues involved, obviously. So I have no idea what the commercialization plans will be. I'll find out more when I go to BGI in the end of October. But as long as we're running it within BGI Research and all for this open access project, it's a non-issue from the patent lawyer perspective. If we start selling the instruments, that becomes a different issue. I'll let the patent people figure that out. But so if you want more information, we just published a paper in GigaScience. It's a telomere to telomere assembly of an African wild rice. And so this just showed up in GigaScience. You can Google for it, and that will give you more detail. And so I think this thing, it does combine with some Hi-C and some Hi-Fi data, but it's no different than everybody else that does combination of Oxford and Hi-Fi. And so we basically replace Oxford nanopores with this, what we call Cyclone C. And depending on the particular application, we may or may not include PacBio's Hi-Fi and of course Hi-C. So it's a hodgepodge thing like everybody else. And so that's about it. I'm done. So I just wanted to acknowledge, all this work is done through BGI. So the BGI Research, the leader these days is Xuzhen. who's his deputy is Gu Ying. And then under the plant work is right now under Wei Tong. And then the people actually overseeing the details are Wang Sibo, who's doing mostly algae. There's not listed Lu Yang, who's doing the bryophytes, and Sunil, who's doing the, I guess it's vascular plants and angiosperms. And we have a lot of collaborators, Michael Melkonian for the algae, Doug Soltis, Pam Soltis, Sean Graham, East Van Peer, Bernard Goffinet, and the bryophytes, and many more that I cannot list. So I thank you for listening.
Okay, Dane. So we have time for one burning question before we close the session. I see, thank God, you has quite a few questions in the chat. So I think Kane has answered quite a few of these questions in these last two slides. So maybe if there's anybody else besides thank God, before we get to thank God, if there's anybody else who would like to ask a question. Go ahead, thank God.
Yeah, thank you, Craig. So I hope we can trash this in the few seconds or minutes, because this is the last presentation. Thanks again for coming. It's nice having you. It's in one of our workshops. I have a number of questions. I think you've answered some, but you can just look at the charts and kind of try to glean from it and see if you can respond. But you've answered the first one that the cyclone is now available, but I'm not quite sure if it's available in any institution in Africa. You've also, I can also see that you've published some paper already with it, so that kind of again answers it.
Thank God we have.
I don't know what the rollout is yet. I'll have to talk to them in person.
Yes.
I will be going in October on a big trip, and then I'll talk to them in person. So I don't know what exactly the rollout plans is. I suspect it's right now. I have exclusive access to the platform for the 10KP. I'm not at liberty because I don't honestly know what the rollout plan is.
No, we have one in the genomics platform.
Okay.
So we're working with MGI and DJI to test cycle in our lab.
Yeah, okay, over to you, Craig, so that we can close on time. But again, I think we can chat more about this offline and try to catch up on this. Thank you so much for coming. Over to you, Craig.
Okay.
Just thank you, thank God, and thanks, Dane, for the presentation. And with that, we come to the end of the first day of our symposium. We had an excellent group of presenters throughout the day. And so we welcome everybody back, to those online and in person. We will start tomorrow morning again at 9 o'clock. And we're going to have a very productive day tomorrow. We look forward to having our students and early career researchers present for us. And our Kane is a panda. Thank you very much for that.
Thank you. Bye-bye. Thank you so much.
came with the we have got